कि सेशन के अलावा हमारे पास बाहर एनआईबी की तरफ से एक स्टॉल लगाया गया है जिसमें हमारी विभिन्न गतिविधियों के से रिलेटेड हमारे पैम्पलेट्स, बुक्स वगैरह इत्यादि रखी गई है। इसी स्टॉल पे आईपीसी की तरफ से भी कुछ इन्फॉर्मेटिव मटेरियल्स रखा गया है, तो कृपया आप स्टॉल ज़रूर विज़िट करिए। हमारे संस्थान की कुछ एक्टिविटीज़ हमने पोस्टर्स के माध्यम से भी प्रस्तुत की हुई है, वो भी हॉल में हमारे सारे पोस्टर्स लगे हुए हैं। बीच में किसी भी वक्त जब भी आपको समय मिले सेशन से, तो कृपया वहाँ भी हो आइएगा। जलपान की व्यवस्था नीचे एट्रियम में की गई है जो ग्राउंड फ्लोर पर है, और चाय इत्यादि तो हमने पूरे टाइम के लिए हॉल नंबर 6 के बाहर उसका इंतज़ाम कर रखा है, तो काइंडली फील फ्री टू हैव दैट। एंड क्योंकि अभी थोड़ा वक्त था हमारे पास, हमने एनआईबी से रिलेटेड एक वीडियो पिछली बार हमने आधिकारिक रूप से लॉन्च किया था, जो हम चाहते हैं कि जब तक सचिव सर हमारे बीच उपस्थित होते हैं आप उस वीडियो का आनंद लीजिए। थैंक यू।
बायोफार्मास्यूटिकल्स अथवा बायोलॉजिकल्स में विश्व स्तर की अत्यंत प्रभावी, जीवन रक्षक, लक्षित तथा महत्वाकांक्षी श्रेणी की दवाएं शामिल हैं और भारत इस क्षेत्र में निरंतर प्रगति कर रहा है। चूंकि ये दवाएं अत्यंत संवेदनशील किस्म की होती हैं, इसलिए इनके उत्पादन और इनकी मार्केटिंग में नियामक गुणवत्ता मानकों के सख्त अनुपालन की आवश्यकता है। ये कहने की आवश्यकता नहीं है कि जब राष्ट्रीय जैविक संस्थान एनआईबी जैसी विधिवत रूप से मान्यता प्राप्त राष्ट्रीय प्रयोगशालाओं द्वारा ऐसी दवाओं का परीक्षण और इन्हें प्रमाणित किया जाता है, तो स्वास्थ्य देखभाल प्रदाताओं और रोगियों का इन पर भरोसा बढ़ जाता है। राष्ट्रीय जैविक संस्थान की स्थापना जनवरी 1992 में स्वास्थ्य और परिवार कल्याण मंत्रालय के संरक्षण में एक स्वायत्त संस्थान के रूप में की गई थी। इस संस्थान ने अपनी वैज्ञानिक यात्रा झंडेवालान नई दिल्ली में स्थित एक अंतरिम सुविधा से शुरू की थी और अब ये NIH USA द्वारा डिजाइन की गई सेक्टर 62 नोएडा उत्तर प्रदेश में स्थित अपनी स्थायी सुविधा से राष्ट्र की सेवा कर रहा है। भारत सरकार की एक राष्ट्रीय वैधानिक प्रयोगशाला NIB को केंद्रीय औषधि प्रयोगशाला और केंद्रीय चिकित्सा उपकरण परीक्षण प्रयोगशाला के रूप में अधिसूचित किया गया है, जो राष्ट्रीय और अंतर्राष्ट्रीय निर्माताओं द्वारा जैविक उत्पादों से संबंधित नियामक गुणवत्ता मानकों का अनुपालन किया जाना सुनिश्चित करता है। यह गैर मानक गुणवत्ता वाले उत्पादों की पहचान करता है और उन्हें बाजार में पहुंचने से रोकता है और सार्वजनिक स्वास्थ्य को बढ़ावा देने में महत्वपूर्ण भूमिका निभाता है।
राष्ट्रीय जैविक संस्थान बायोलॉजिकल्स की क्वालिटी टेस्टिंग करता है ताकि पब्लिक हेल्थ सिस्टम को सेफगार्ड किया जा सके। हमारा स्टाफ लगन से काम करता है हमारे इंस्टीट्यूट के मैंडेट को अचीव करने के लिए। मैं धन्यवाद देना चाहूँगा मिनिस्ट्री ऑफ हेल्थ एंड फैमिली वेलफेयर, CDSCO तथा अन्य स्टेक होल्डर्स का, जिनका हमें लगातार सपोर्ट प्राप्त होता रहता है। साथ में मैं हमारे सभी पूर्व निदेशकों का भी धन्यवाद देना चाहूँगा और फ्यूचर में हम और बायोलॉजिकल्स को ऐड करना चाहेंगे अपने पोर्टफोलियो में, ताकि फ्यूचर के चैलेंजेस को हम टैकल कर सकें।
ड्रग्स एंड कॉस्मेटिक्स एक्ट और उसके तहत बनाए गए नियमों, इंडियन फार्माकोपिया अथवा लागू अंतर्राष्ट्रीय नियामक मानदंडों में निर्धारित प्रावधानों के अनुसार सुप्रशिक्षित वैज्ञानिक और तकनीकी कर्मचारियों द्वारा अत्याधुनिक प्रयोगशालाओं में बायोलॉजिकल्स का परीक्षण किया जाता है। परीक्षण की गुणवत्ता सुनिश्चित करने के लिए वैश्विक स्तर पर ज्ञात परीक्षण मानकों जैसे ISO 17025 और परीक्षण के बाद उत्पन्न अपशिष्ट के निपटान के लिए बायो वेस्ट प्रबंधन नियमों का अनुपालन किया जाता है। संस्थान के कई वैज्ञानिकों को सरकारी विश्लेषक और चिकित्सा उपकरण परीक्षण अधिकारी भी अधिसूचित किया गया है। NIB अंतर्राष्ट्रीय संदर्भ मानकों के विकास, प्रयोगशालाओं और विनिर्माण इकाइयों के निरीक्षण और महत्वपूर्ण परीक्षण मापदंडों के विधि सत्यापन और उसके बाद मोनोग्राफ विकसित करने के लिए अंतर्राष्ट्रीय संगठनों जैसे WHO, NIBSC UK और विभिन्न अन्य अंतर्राष्ट्रीय निकायों जैसे NACO, ICMR और IPC के साथ सहयोग करता है। NIB ने भूटान, दक्षिण अफ्रीका, बांग्लादेश जैसे अन्य देशों को बायोलॉजिकल्स की क्वालिटी कंट्रोल टेस्टिंग और हीमोविजिलेंस के विभिन्न क्षेत्रों में विशेष सेवाएं प्रदान की हैं। नमूना प्राप्ति और रिपोर्ट डिस्पैच इकाई SRRDU, NIB और इसके हितधारकों जैसे विनियामक निकायों, निर्माताओं, सरकारी चिकित्सा आपूर्तिकर्ताओं आदि के बीच सैंपल प्राप्त करने और मूल्यांकन के बाद परिणाम रिपोर्ट करने का कार्य करती है। बायोलॉजिकल्स को मूल रूप से 3 मुख्य श्रेणियों में विभाजित किया जाता है: डायग्नोस्टिक्स, थेराप्यूटिक्स और प्रोफिलैक्टिक्स। NIB ट्रांसफ्यूजन संचारित रोगों जैसे HIV, HBV, HCV, सिफिलिस आदि के लिए रैपिड टेस्ट से लेकर मॉलिक्यूलर टेस्ट तक इन विट्रो डायग्नोस्टिक्स और सुरक्षित ब्लड ट्रांसफ्यूजन के लिए ABO और RH ब्लड ग्रुपिंग के उपयोग में लाए जाने वाले रिएजेंट्स तथा दुर्लभ प्रकार के ब्लड ग्रुपिंग रिएजेंट्स और ब्लड ग्रुपिंग जेल कार्ड या जेल का मूल्यांकन करता है। भारत दुनिया में मधुमेह से प्रभावित दूसरा सबसे अधिक प्रभावित देश होने के नाते NIB ने CLSI दिशा निर्देशों के आधार पर ग्लूकोमीटर, ब्लड ग्लूकोज टेस्ट स्ट्रिप्स और ग्लूकोज रिएजेंट्स के क्वालिटी कंट्रोल टेस्टिंग के लिए एक समर्पित और अपनी तरह की पहली प्रयोगशाला स्थापित की है। थेराप्यूटिक्स के क्षेत्र में NIB सक्रिय रूप से प्लाज्मा डिराइव्ड उत्पाद जैसे ह्यूमन एल्ब्यूमिन, इम्यूनोग्लोब्युलिन, कोएगुलेशन फैक्टर्स आदि, रिकॉम्बिनेंट DNA डिराइव्ड थेराप्यूटिक्स जैसे इंसुलिन, इंसुलिन एनालॉग्स, ग्रोथ फैक्टर्स, साइटोकाइंस, विभिन्न एंजाइम्स जैसे स्ट्रेप्टोकाइनेज और फर्टिलिटी हार्मोन्स की गुणवत्ता सुनिश्चित करने में कार्यरत है। NIB विभिन्न थेराप्यूटिक एंटीबॉडी की भी गुणवत्ता सुनिश्चित करता है जैसे ट्रास्टुजुमैब, रिटुक्सिमैब, जिनका उपयोग कैंसर के उपचार आदि में भी किया जाता है। इसके अलावा वैक्सीन के क्षेत्र में NIB को BCG, CCRV, MMR और IPV जैसे टीकों का मूल्यांकन करने में विशेषज्ञता प्राप्त है और हाल ही में कोविड-19 के टीकों के लिए भी विशेषज्ञता प्राप्त कर ली है। NIB की प्रयोगशालाएं टेस्ट परफॉर्मेंस के मूल्यांकन को और मजबूत बनाने के लिए यूरोपियन डायरेक्टरेट फॉर द क्वालिटी ऑफ मेडिसिन EDQM फ्रांस, WHO जिनीवा, नेशनल सीरोलॉजी रेफरेंस लेबोरेटरी NSRL ऑस्ट्रेलिया, NIB पुणे, क्रिश्चियन मेडिकल कॉलेज वेल्लोर जैसी विभिन्न अंतर्राष्ट्रीय और राष्ट्रीय बाहरी एजेंसियों द्वारा किए जाने वाले एक्सटर्नल क्वालिटी अश्योरेंस स्कीम EQAS विभिन्न प्रोफिशिएंसी टेस्टिंग में नियमित रूप से भाग लेती है, जिसके परिणाम उत्साहजनक हैं और अंतर्राष्ट्रीय प्रवीणता मानदंडों के अनुसार 100% सामंजस्य के साथ संतोषजनक हैं। संस्थान में सेंट्रलाइज्ड अत्याधुनिक सहायक प्रयोगशालाएं हैं, जिनमें स्टेरिलिटी टेस्टिंग प्रयोगशाला, बायोएसे प्रयोगशाला और पशु सुविधा शामिल हैं। NIB की पशु सुविधा 1.16 एकड़ में फैली हुई है, जो बायोलॉजिकल्स के गुणवत्ता नियंत्रण मूल्यांकन के हिस्से के रूप में महत्वपूर्ण इन वीवो परीक्षण करती है। ये पशुओं पर परीक्षण के नियंत्रण एवं पर्यवेक्षण के प्रयोजनार्थ समिति CPCSEA के मानदंडों के अनुसार विभिन्न राज्यों के विभिन्न संस्थानों को छोटे प्रयोगशाला जानवरों की आपूर्ति भी करती है। NIB ने भारत की पहली हाई थ्रूपुट कोविड-19 सैंपल टेस्टिंग प्रयोगशाला स्थापित करके लगातार 24 घंटे 7 दिन काम करके 2.5 लाख से अधिक कोविड-19 के संदिग्ध सैंपलों का परीक्षण करके कोविड-19 महामारी के खिलाफ देश की लड़ाई में अपनी अग्रणी भूमिका निभाई है। यहाँ कोविड-19 डायग्नोस्टिक किट और VTM का भी परीक्षण किया जाता है। इसके अलावा NIB ने कोविड-19 टीकों के गुणवत्ता नियंत्रण परीक्षण के लिए सुविधा स्थापित की है। संस्थान ह्यूमन इंसुलिन और इंसुलिन लिस्प्रो हेतु राष्ट्रीय संदर्भ मानकों के विकास के लिए एक नोडल संगठन है और इनका एकमात्र भंडार गृह भी है। भारत सरकार की मेक इन इंडिया पहल का समर्थन करते हुए शोधकर्ताओं और स्वदेशी निर्माताओं को HIV, HBS, AG, HCV और सिफिलिस के परफॉर्मेंस पैनल उपलब्ध कराए जाते हैं। NIB में क्वालिटी टेस्टिंग और ऑक्यूपेशनल हेल्थ प्रैक्टिसेस को सुनिश्चित करने के लिए एक स्वतंत्र गुणवत्ता प्रबंधन इकाई भी है जो सभी आवश्यक मान्यताओं और प्रमाणन का कार्य देखती है। सुरक्षा को NIB में सर्वोपरि महत्व दिया जाता है। संस्थान ऑक्यूपेशनल हेल्थ सेफ्टी के लिए वैश्विक मानकों का पालन करता है और इसका परिसर दिव्यांग जन अनुकूल है, जिसमें दिव्यांग जनों की आवश्यकताओं का पूरा ख्याल रखा गया है, जिसके लिए इसे गर्व है। बायोलॉजिकल्स की टेस्टिंग से संबंधित नैतिक मुद्दों की इंस्टीट्यूशनल ह्यूमन एथिक्स कमेटी, एनिमल एथिक्स कमेटी, बायोसेफ्टी कमेटी द्वारा समीक्षा की जाती है और इनके अनुमोदन के बाद ही परीक्षण शुरू किया जाता है। इसके अतिरिक्त प्रधानमंत्री कौशल विकास योजना के अनुरूप NIB बायोलॉजिकल्स के क्वालिटी कंट्रोल टेस्टिंग के क्षेत्र में छात्रों के साथ साथ उद्योग के तकनीकी कार्मिकों को प्रशिक्षण प्रदान करता है। देश में रक्त सेवाओं को मजबूत करने के उद्देश्य से NIB ब्लड सेल, राष्ट्रीय स्वास्थ्य मिशन NHM के सहयोग से ब्लड बैंक अधिकारियों को प्रशिक्षण प्रदान करता है। NIB वर्ष 2012 में शुरू किए गए भारत के हीमोविजिलेंस कार्यक्रम का राष्ट्रीय समन्वय केंद्र है और रक्त चढ़ाने और रक्तदान से संबंधित प्रतिकूल प्रतिक्रियाओं को एकत्रित करने और उनका आकलन करने और उनके घटित होने और पुनरावृत्ति को रोकने के लिए अंतर्राष्ट्रीय हीमोविजिलेंस नेटवर्क का सदस्य भी है। NIB के प्रशासनिक, उपार्जन, वित्त और इंजीनियरिंग विभाग के निरंतर सहयोग से NIB एक वैज्ञानिक संस्था की आधारिक संरचना की सभी आवश्यकताओं को पूरा करता है। इसमें 7.5 मेगावाट का इन हाउस जेनरेटर के साथ 100% पावर बैकअप है, जिससे बायोलॉजिकल्स का निर्बाध परीक्षण सुनिश्चित किया जाता है। संस्थान में इसके व्यापक प्रशिक्षण कार्यक्रमों और सम्मेलनों में भाग लेने वाले छात्रों, प्रशिक्षुओं और प्रतिनिधियों के लिए हॉस्टल और गेस्ट हाउस है। NIB, जिसकी देश और दक्षिण पूर्व एशिया क्षेत्र के लिए सभी बायोलॉजिकल्स की टेस्टिंग के लिए एक रेफरल प्रयोगशाला के रूप में कल्पना की गई थी, वो देश में बायोलॉजिकल्स के क्वालिटी कंट्रोल के क्षेत्र में एक पथ प्रदर्शक बनने और अंतर्राष्ट्रीय बेंचमार्क को हासिल करने संबंधी अपने लक्ष्य को पूरा करने की दिशा में प्रयासरत है।
आदरणीय सचिव सर हमारे बीच पर आ चुके हैं। कृपया तालियों से उनका जोरदार स्वागत करें। Dr. Jaideep Mishra sir, please. Shri Rajiv Vadavan sir. Dr. Harish. नमस्कार। हमारे सभी विशिष्ट अतिथिगण को राष्ट्रीय जैविक संस्थान की ओर से हमारा हार्दिक अभिनंदन। कार्यक्रम शुरू करने से पहले मैं अनुरोध करना चाहूँगी हमारे निदेशक डॉक्टर अनूप अन्वीकर सर से कि वे डाइस पे आए हमारे माननीय केंद्रीय स्वास्थ्य सचिव जी का बुके देके सम्मानित करें। हमारे दूसरे अतिथि डॉक्टर गोपाल कृष्ण जी। श्री राजीव वदावन जी के लिए ओके। श्री जयदीप मिश्रा जी। धन्यवाद सर। राष्ट्रीय जैविक संस्थान जन स्वास्थ्य रक्षा के लिए प्रतिबद्ध। आज के इस शिखर सम्मेलन के लिए यही है हमारी संक्षिप्त और छोटी सी पहचान, पर बहुत ही महत्वपूर्ण। आज का ये कार्यक्रम जो एजेंडा था, शुरू करने से पहले जो अप्रत्याशित बदलाव इस एजेंडा में हुए आपको अवगत करा दूं। पहले इस शिखर सम्मेलन की अध्यक्षता के लिए हमारे बीच आने वाले थे माननीय स्वास्थ्य मंत्री डॉक्टर मनसुख मांडविया जी और माननीय केंद्रीय स्वास्थ्य परिवार कल्याण राज्य मंत्री सुश्री डॉक्टर प्रवीण पवार जी, पर किसी आधिकारिक कारणों की वजह से वो हमारे बीच उपस्थित नहीं हो पाए, पर उन्होंने अपने बिजी शेड्यूल में से हमारे लिए, आप सब के लिए कुछ वीडियो मैसेजेस भेजे हैं जो हम कुछ ही देर में अभी यहाँ पे प्ले करेंगे। पर सबसे पहले मैं अभिनंदन करना चाहूँगी हमारे विशेष अतिथि माननीय आदरणीय सर सचिव सर श्री राजेश भूषण जी, जिन्होंने हमारे इस सम्मेलन की अध्यक्षता को स्वीकार करके इसकी गरिमा को और उज्जवल किया है। सर, आपका इस समारोह में हमारे लिए आना, हम आपका हार्दिक अभिनंदन करते हैं। इस सेशन की शुरुआत में हमने जब ये एजेंडा बनाया था तो हमारे जो विचार थे वो ये थे कि हम जो 3 महत्वपूर्ण भूमिकाएं हमारे संस्थान निभाती है, 3 संस्थाओं के साथ, चाहे रेगुलेटर्स हो, इंडस्ट्री हो और एकेडमिशियंस हो, तो वो हम सारा इस शिखर सम्मेलन में दिखाना चाह रहे थे। और ये हमारा प्रयास था कि हम किसी तरह से उसमें एक बेहतर योगदान दें। इसलिए हमारे सेशंस 3 सेक्शंस में डिवाइडेड हैं। पहले हम रेगुलेटर्स की बात सुनेंगे, उसके बाद इंडस्ट्री और आखिर में जो सबसे महत्वपूर्ण सेशन रहेगा हमारा एकेडमिशियंस के साथ, क्योंकि हमारा मानना है कि एक अच्छी नींव जो धरोहर हमें भविष्य के लिए चाहिए वो एकेडमिशियंस हमें दे सकते हैं। पर सबसे पहले कि जैसा सनातन काल से चली आ रही परंपरा है कि हम इस समारोह की शुरुआत दीप प्रज्वलन से करेंगे। दीप प्रज्वलन हमारी संस्कृति की एक अलौकिकता को दर्शाता है और इसका मूल उद्देश्य ये रहता है कि अंधकार से प्रकाश की ओर हमें जो ले जाए और उसकी प्रार्थना हम करें। तो मैं अनुरोध करना चाहूँगी हमारे सचिव सर से कि कृपया दीप प्रज्वलन के लिए। जी सर।
शत्रुबुद्धिविनाशाय दीपज्योति नमोस्तुते। दिव्यादिव्यारे दीपत्कार कानी कुंडलमोतीहार दिव्याला पाहून नमस्कार तिळाचे तेल कापसाची वात दिवा उजेड पडे विष्णू पाशी माझा नमस्कार सर्व देव देवतां पाशी।
धन्यवाद। अब मैं अनुरोध करना चाहूँगी हमारे निदेशक डॉक्टर अनूप अनवीकर जी से कि कृपया आके आप सबको संबोधित करें।
धन्यवाद रश्मि जी, नमस्कार। डॉक्टर मनसुख मांडविया जी माननीय केंद्रीय स्वास्थ्य एवं परिवार कल्याण मंत्री, डॉक्टर भारती प्रवीण पवार जी माननीय केंद्रीय स्वास्थ्य एवं परिवार कल्याण राज्य मंत्री तथा श्री राजेश भूषण जी सचिव स्वास्थ्य एवं परिवार कल्याण मंत्रालय, इनके मार्गदर्शन में राष्ट्रीय जैविक संस्थान का 31वां स्थापना दिवस इस राष्ट्रीय शिखर सम्मेलन के रूप में मनाया जा रहा है। इस सम्मेलन के जनक माननीय स्वास्थ्य सचिव श्री राजेश भूषण जी, श्री गोपाल कृष्णन जी स्पेशल सेक्रेटरी, श्री जयदीप कुमार मिश्रा जी AS एंड FA, श्री राजीव वधावन एडवाइज़र कॉस्टिंग मिनिस्ट्री ऑफ हेल्थ एंड फैमिली वेलफेयर, मेरे कलीग डॉक्टर हरीश चंदर, और इस सदन में उपस्थित मेरे काफ़ी मेंटर्स डॉक्टर अमूल्य पांडा जी, डॉक्टर साहू और बहुत सारे कलीग्स। अपार्ट फ्रॉम दिस कि कलीग्स फ्रॉम CDSCO, IPC, industry, procurement agencies, CDLs, then academicians, policy makers and my very own NIB colleagues तथा अन्य सभी stakeholders, मैं आप सबका इस शिखर सम्मेलन में स्वागत एवं सादर अभिनंदन करता हूं। इस सम्मेलन का फोकस क्वालिटी ऑफ बायोलॉजिकल्स है। राष्ट्रीय जैविक संस्थान मार्केट रिलीज से पहले बायोलॉजिकल्स की क्वालिटी टेस्ट करके पब्लिक हेल्थ की सेफ्टी इंश्योर करता है। NIB को टेस्टिंग के लिए 2011 से NABL के ISO 17025 के डिफाइंड स्कोप के लिए एक्रीडिटेशन प्राप्त है। बायोफार्मा का सीधा इम्पैक्ट पब्लिक हेल्थ पर होता है, इसीलिए सरकार कैपेसिटी बढ़ाने के लिए निरंतर प्रयासरत है ताकि क्वालिटी मेडिसिन और डायग्नोस्टिक्स जनता तक अफोर्डेबल तरीके से उपलब्ध हो। जहां एक और भारत ग्लोबल बायोफार्मा लीडर बनने की राह पर है, वहीं प्रोडक्शन के साथ साथ इनोवेशन के मामले में भी इंडस्ट्री का लगातार विकास हो रहा है। इस सिनारियो में हमें इंश्योर करना है कि बायोलॉजिकल मेडिसिन्स तथा डायग्नोस्टिक्स क्वालिटी एश्योर्ड हो तथा रेगुलेटेड हो। आपको ये जानकर खुशी होगी कि हाल ही में हमारे संस्थान को टेस्टिंग के क्षेत्र में अपनी उत्कृष्टता के लिए क्वालिटी काउंसिल ऑफ इंडिया द्वारा स्थापित देश का पहला प्रोफेसर एस.के. जोशी लैब एक्सेलेंस गोल्ड अवार्ड मिला है। हम हमेशा कार्यरत रहते हैं हमारे इंडस्ट्री कलीग्स के साथ मिलकर काम करने के लिए, उनकी अगर कोई समस्याएं हैं उनको ठीक करने के लिए। एक डिमांड थी हमारे फार्मा कलीग्स की कि भाई NIB बहुत ज़्यादा सैंपल साइज़ रखता है, अपने सैंपल्स को टेस्ट करने के लिए बहुत ज़्यादा फीस कर लेता है, बहुत ज़्यादा समय लगाता है। हमने वर्क किया इस चीज़ को और हमने सभी स्टेक होल्डर्स के साथ बातचीत की, हमारे एक्सपर्ट कमेटी में विषय लगा और इट वाज बैक्ड बाय साइंस। ऐसा नहीं कि आपकी डिमांड को हमने फुलफिल किया, इट वाज बैक्ड बाय साइंस और हमने सैंपल साइज को अप टू 60% कम किया है। कुछ केसेस में टेस्टिंग फ़ी अप टू 40% कम करी है, जहाँ पे जो टर्न अराउंड टाइम 42-45 दिन का रहता था डायग्नोस्टिक में 5वें-6ठे दिन रिपोर्टें निकलने लगी है। हम अपनी एंड में शनिवार को भी काम करने लगे हैं, जो हमारा 5 दिन वैसे वर्किंग वीक होता है। और इसके अलावा हमने पूरा एविडेंसेस देखे, हमारी बहुत लोगों वाली एक्सपर्ट कमेटी है उसके सामने विचार रखा और आगे भी हम निरंतर प्रयास करते रहेंगे आपके साथ मिलके जो भी कोई दिक्कतें हैं उसको सॉल्व करने की। इसीलिए इस शिखर सम्मेलन का हमने आयोजन किया है। ये पूरा डाइवर्स है सर एरिना, देर आर पीपल फ्रॉम ऑल वास्ट एरियाज जिनका भी रिलेशन NIB के साथ है। आपको अगर कुछ सुझाव है हमारे वर्किंग के लिए, हमारे फंक्शनिंग के लिए, दे आर मोस्ट वेलकम। आपकी कोई शिकायतें हैं हम उसको भी आज सुनने के लिए यहाँ पर है। आप अगर आज नहीं बता पाएंगे आप बाद में भी हमें बताएं, हमें बड़ी खुशी होगी वो सुनने में और उसको सॉल्व करने में। पब्लिक हेल्थ के फ्यूचर के चैलेंजेस और डिमांड्स को पूरा करने के लिए हम हमेशा तत्पर हैं। मैं हमारा मंत्रालय, CDSCO, IPC और आप सभी स्टेक होल्डर्स का आपके निरंतर सहयोग के लिए आभार व्यक्त करता हूं। अंत में मैं मंत्री महोदय, मंत्री महोदया तथा सचिव महोदय का पुनः आभार व्यक्त करता हूं। जय हिंद।
धन्यवाद सर। कार्यक्रम को आगे बढ़ाते हुए जैसा कि सर ने आप सब लोगों को बताया, आज हमारी 32nd फाउंडेशन डे है। 31 वर्ष हम हमारी संस्था का स्थापना हो चुके हैं। मुझे बहुत खुशी हो रही है क्योंकि हमारे काफ़ी एक्स डायरेक्टर्स, पुराने लोग, सारे लोग आज हमारे साथ इस समारोह में आए हैं। हमारे संस्थान के जो प्राइम मैंडेट्स रहे हैं, जैसे कि जब इसकी स्थापना हुई थी एक विजन के साथ हुई थी कि जो जैविक उत्पाद जो हमारी कंट्री में आते हैं या हमारी कंट्री में बनते हैं उनकी गुणवत्ता सुनिश्चित की जाए। इसके अलावा नेशनल और इंटरनेशनल लिंकेजेस हो और इंडस्ट्री को पूर्ण सहयोग मिले। इस पूरे मैंडेट पे काम करते हुए 31 सालों का जो सफ़र है और आज जिस पड़ाव पे NIB आप लोगों के सामने है उसको दर्शाता हुआ तथा एक संक्षिप्त सी टाइमलाइन दिखाते हुए हमने एक कॉफ़ी टेबल बुक बनवाई है, जिसका आज सचिव सर के कर कमलों से हम विमोचन कराना चाहेंगे। हमें काफ़ी खुशी है कि आज हमारी इस वर्षगांठ की सेलिब्रेशन हमें आप सब लोगों के साथ मनाने का सौभाग्य प्राप्त हुआ है। आप सब लोगों का एक बार फिर हार्दिक अभिनंदन। मैं अनुरोध करना चाहूँगी टीम से कि वो बुक्स लेकर आ जाएं। ठीक है। ये पुस्तक हम अभी स्टॉल पे भी डिस्प्ले पे रखेंगे। हम चाहते हैं कि हमारी तरफ से एक रिटर्न गिफ्ट की तरह आप कृपया एक प्रतिलिपि अपने साथ ज़रूर लेके जाएं। अब इस इनॉग्रल सेशन की सबसे महत्वपूर्ण अंश की ओर बढ़ते हैं और मैं अनुरोध करना चाहूँगी आदरणीय सचिव सर से, जो कि हमारे संस्थान के गवर्निंग बॉडी और जनरल बॉडी के अध्यक्ष भी हैं, कि कृपया डाइस पे आएँ और सभा को संबोधित करें।
The Director of National Institute of Biologicals, Dr. Anoop Anvikar, my colleagues in the Ministry, Special Secretary Shri Gopala Krishnan, my other colleague in the Ministry, Additional Secretary and Financial Advisor Shri Jaideep Mishra, the Joint Secretary and Advisor Cost, Shri Rajiv Vadhavan, who also looks after drug and regulatory issues, Dr. Harish Chandar representing NIB, ladies and gentlemen. I also see a lot of old faces which bring back old memories. I was associated with this ministry as director, looking after food, drug, regulation, international relations, medical education, and a lot of other issues from 2000 to 2006. Those were the years when NIB was undergoing a transformation and upgradation in its physical infrastructure. Most of the labs were getting substantially and significantly upgraded, and a lot of new skilled manpower was getting recruited. So therefore, my satisfaction and my happiness is that much greater when I see lot of young, energetic faces here, led by a young and energetic director. And I wish you well on your 31st Foundation Day. I am also extremely satisfied to know that for the entire day today, you would be deliberating with various stakeholders on the different aspects of quality control of biologicals in this national summit. I also congratulate everyone. This is something that Anoop just mentioned, that you have won the first Professor S.K. Joshi Gold Award. So this is something that you must keep up going ahead. Ladies and gentlemen, I wanted to draw your attention to the fact that when we have gathered here today, we also are chairing the G20 group of nations. The G20 presidency is with India today, and this presidency will continue till the end of 2023. And within the presidency, there are various tracks. There is a finance track, उसमें अलग से negotiations member countries के बीच में होते हैं. There is a health track, there is a environment and climate change track. So in the health track, the three priorities that India has put on the table for G20 countries are dealing with health emergencies, one of which is still, we are in the midst of that health emergency, which is the COVID-19 pandemic. The second priority that we have put forward, and this is why I am sharing this with you, the second priority is how do we ensure equitable access to quality VTDs, vaccine, therapeutics, and diagnostics. And the third priority is how do we proceed towards universal health coverage using digital solutions. So while we interact and deliberate on ensuring quality of biologicals, more efficient testing, science-based protocols, so on and so forth, engaging with regulators, engaging with industry, we should not lose sight of the fact, why are we doing all this? अगर हमने समय घटा दिया टेस्ट करने का तो क्यों? अगर हमने ये कहा कि हमें इतनी ज़्यादा क्वांटिटी में सैंपल नहीं चाहिए, कम चाहिए, तो क्यों? Eventual aim is not to make the process simpler. eventual aim is not only be governed by science, eventual aim is to promote that those products are available in the market and they are available for everyone and they are available at a prescribed level of quality. अल्टीमेट एम तो वो है, लक्ष्य तो वो है, इसलिए आप ये सब काम कर रहे हैं, इसीलिए आपको वो इनाम भी मिला है जो अभी मिला. तो हमें ये सोचना है, गोइंग अहेड, we are a country which is popularly called the pharmacy of the world. Every fourth child in the world is inoculated using an Indian made vaccine. So we are virtually the vaccine capital of the world. But what are we doing and what should we do more going ahead to ensure equity and access? तो ये चैलेंज है, ये चुनौती है, और उसमें क्योंकि मल्टी स्टेक होल्डर एक्सरसाइज है ये, तो रेगुलेटर्स को भी काम करना है, जो क्वालिटी टेस्टिंग करते हैं उनको भी करना है, जो मैन्युफैक्चर करते हैं उनको भी करना है, और जो प्रोग्राम मैनेजर्स हैं मुझ जैसे लोग या मेरे मित्र जो उस तरफ बैठे हुए तीन, हमारा भी रोल है. तो ये एक महत्वपूर्ण चीज़ है जो हमें अपने दिमाग में रखनी चाहिए एज वी गो अहेड. I also believe that institutions like NIB cannot exist in isolation. So they need to interact on a regular basis, not only with the national regulators, that is CDSCO, Central Drug and Standards Organization. They should also be dealing with the Indian Pharmacopoeia Commission on a regular basis, and also interacting with the industry again on a regular basis. And they must keep their channels of communication open with people who have worked in the field and are working in the field, who can contribute domain knowledge. Dr. Gupta is one such example. He's just joined us. We welcome him. So NIB will remain a vibrant organization only when they keep all these channels of communication open. We must also not lose sight of the fact that in today's global world, until and unless we build collaborations, we will not progress. So NIB should actively explore collaborative arrangements with similar other organizations in G20 countries. And if you need any help of the ministry to take those MOUs or agreements forward, that is something that, Rajiv, we should be pushing on a proactive basis. So would Gopal help in this exercise. So please do not lose sight of the fact that while within the country you may be doing exceptionally good work, you must build networks. You must be part of electronic networks, physical networks of institutions that go beyond the country boundaries. Particularly during this period of India's G20 presidency, please ensure that you have some kind of agreement or MOU with similar organizations in G20 countries. I would also like to take this opportunity to place on record that during the last 1.5 years of, or almost 2 years now, of COVID pandemic and its management, NIB set new standards. NIB for the first time acquired the know-how and the skill set to be a central drug lab, CDL, for vaccines. That work should be taken forward. You should not keep it limited to paper, saying कि हमको मिल गया, उसके बाद आप क्या कर रहे हैं वो भी आपको देखना चाहिए. But this is a credit-worthy achievement and we must appreciate it. We must also appreciate the kind of quality control testing that you established for COVID diagnostic kits. जब मैं VTD की बात करता हूँ तो उसमें diagnostics is a critical component. वो भी आपने पहली बार किया during the COVID crisis. and obviously COVID vaccine is something that I've just pointed out. So these activities should not become one-off कि एक बार कर दिया तो बहुत अच्छी बात है, लेकिन उसको फिर आगे ले जाने की ज़रूरत है. So to sum up, I congratulate you for all your achievements. I had forgotten, you have also started your PhD courses. So we must congratulate you for that also. And I should again, before closing, suggest that whatever collaborations that NIB has today with the regulator on one hand and with the industry and academic institutions on the other must be strengthened. And we wish that you would establish new standards of quality control in the country, and you would play a leading role in networking with other such globally established institutions. So I once again wish all of you well. मेरी शुभकामनाएं आपके साथ हैं आपके 31वें फाउंडेशन दिवस समारोह के अवसर पर, और मेरी शुभकामनाएं इस नेशनल समिट के लिए भी हैं कि इससे कुछ कॉन्क्रीट चीज़ें निकलें जिसको फिर आगे ले जाने में मंत्रालय और NIB मिलके काम करें. बहुत बहुत धन्यवाद. जय हिंद.
धन्यवाद सर. जैसा कि मैंने पहले बताया कि किसी आधिकारिक कारणवश हमारे बीच मंत्री जी उपस्थित नहीं हो पाए हैं, पर हमारे पास उनके वीडियो मैसेजेस हैं. तो मैं सबसे पहले IT विभाग से अनुरोध करना चाहूँगी कि हमारे केंद्रीय स्वास्थ्य राज्य मंत्री सुश्री डॉक्टर भारती प्रवीण पवार जी का मैसेज प्ले करें.
नमस्कार. हमारे देश के लिए अच्छी क्वालिटी के बायोलॉजिकल्स की अवेलेबिलिटी इंश्योर करने के लिए नेशनल इंस्टीट्यूट ऑफ बायोलॉजिकल्स NIB के 31 साल पूरे होने पर बहुत बहुत बधाई. किसी भी देश के लिए एक मजबूत हेल्थ केयर सिस्टम का होना महत्वपूर्ण है. भारत सरकार माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के दूरदर्शी नेतृत्व में भारत के लोगों की सभी स्वास्थ्य आवश्यकताओं को पूरा करने के लिए विभिन्न नई तकनीकों की सहायता से नई पहल कर रही है. NIB एक ऐसा इंस्टीट्यूट है जिसकी क्वालिटी बायोलॉजिकल्स को रिलीज करने में महत्वपूर्ण भूमिका है, जिससे इफेक्टिव और सेफ प्रोडक्ट की अवेलेबिलिटी इंश्योर होती है. इन प्रोडक्ट्स में वैक्सीन्स, ब्लड प्रोडक्ट्स, एंजाइम्स आदि शामिल है, जिनमें कॉमन कोल्ड से लेकर कैंसर तक की बीमारियों और स्थितियों के खिलाफ लड़ाई के आवश्यक टूल्स हैं. जैविक की गुणवत्ता की गहन और निष्पक्ष मूल्यांकन करने के लिए NIB यूनिक पोजीशन में है. रॉ मटेरियल्स और रेडी प्रोडक्ट्स का इवैल्यूएशन करके NIB यह इंश्योर करने में मदद करता है कि बायोलॉजिकल्स हाईएस्ट क्वालिटी के भी है और पब्लिक यूज़ के लिए सुरक्षित भी है. NIB गुड मैन्युफैक्चरिंग प्रैक्टिसेस को बढ़ावा देने और बायोलॉजिकल्स की क्वालिटी और सेफ्टी को इंश्योर करने के साथ साथ प्यूरिटी, पोटेंसी और स्टेबिलिटी के लिए बायोलॉजिकल्स प्रोडक्ट का टेस्ट करता है. NIB इसके लिए अन्य रेगुलेटरी एजेंसीज और इंडस्ट्री पार्टनर्स के साथ सहयोग करने में भी महत्वपूर्ण भूमिका निभाता है, जिससे बायोलॉजिकल्स की क्वालिटी और सेफ्टी को इंश्योर करने के लिए इंटरनेशनल एफर्ट्स में भाग लिया जा सके. बायोफार्मा इंडस्ट्री नए युग की ऑटोमेशन टेक्नोलॉजीज को अपना रही है. रेगुलेटर्स के साथ टेस्टिंग लैब्स को भी इन इमर्जिंग टेक्नोलॉजीज और प्रैक्टिसेस के साथ तालमेल रखना चाहिए, ताकि जीवन रक्षक दवाओं को बाजार में तेज गति से लाया जा सके. लंबी अवधि के विकास के लिए हमें R&D और इनोवेशन, API में सेल्फ रिलायंस, क्वालिटी स्टैंडर्ड्स को उन्नत करने, डिजिटलाइजेशन, सिंप्लीफिकेशन ऑफ रेगुलेशन और एक्सपोर्ट की दिशा में बढ़ने पर ध्यान देने की आवश्यकता होगी. मैं यहाँ उपस्थित इंडस्ट्री रिप्रेजेंटेटिव्स को सुझाव देना चाहती हूँ कि वे अपने प्रपोजल्स के साथ आगे आए और विभिन्न बायोलॉजिकल प्रोडक्ट्स के निर्माण में हमारे स्वदेशी उद्योग को मजबूत करने के लिए आने वाली बाधाओं पर चर्चा करें। मैं सभी स्टेक होल्डर्स से अनुरोध करती हूँ कि वे एक ही मंच पर काम करें ताकि विभिन्न चरणों की कड़ाई से निगरानी करने के नए बायोलॉजिकल्स को लाने और कुछ एक्टिविटीज़ का रेपुटेशन कम करने में संबंधित मुद्दों पर चर्चा की जा सके। मुझे पूरा विश्वास है कि यह सहयोग देश के हेल्थ केयर सिस्टम को मजबूत करेगा, जो हमारे आदरणीय प्रधानमंत्री जी का विजन भी है। मैं फिर एक बार बहुत बहुत शुभकामनाएं देती हूँ। धन्यवाद।
बहुत बहुत आभार प्रकट करना चाहती हूँ मैं इस वीडियो मैसेज के लिए मैम का। अब मैं अनुरोध करना चाहूँगी आईटी विभाग से कि वो माननीय स्वास्थ्य मंत्री जी का वीडियो भी प्ले करें।
नमस्कार। नेशनल इंस्टीट्यूट ऑफ बायोलॉजिकल्स द्वारा आयोजित इस क्वालिटी बायोलॉजिकल्स के राष्ट्रीय शिखर सम्मेलन में यहाँ उपस्थित सभी एक्सपर्ट्स और स्टेक होल्डर्स का स्वागत करते हुए मुझे अत्यंत प्रसन्नता हो रही है। जैसा कि आप सब लोग जानते हैं कि वर्तमान समय में महामारी ने हमें एक बड़ी शिक्षा दी है कि हमें अपने देश के हेल्थ सिस्टम को और अधिक मजबूत करना है। अब पारंपरिक केमिकल दवाओं के साथ बायोलॉजिकल दवाएं एक चॉइस ऑफ थेरापी के रूप में उभरी है। पिछले कुछ वर्षों में कोविड-19 महामारी के कारण मेडिकल इमरजेंसी में देखा गया है कि हमारी बायोफार्मा और डायग्नोस्टिक इंडस्ट्री न केवल हमारे देश के लिए बल्कि विश्व स्तर पर पब्लिक हेल्थ आवश्यकताओं को पूरा करने के लिए रणनीतिक संपत्ति यानी कि स्ट्रेटेजिक एसेट्स साबित हुई है, जिसने यूनिवर्सल ब्रदरहुड के कथन वसुधैव कुटुंबकम को भी सार्थक कर दिया है। एफोर्डेबल हेल्थ केयर के साथ विश्व स्तर पर पब्लिक हेल्थ में सुधार करते हुए भारतीय अर्थव्यवस्था के इस क्षेत्र ने पिछले कुछ वर्षों में तेजी से वृद्धि दिखाई है और नए बायोलॉजिकल्स की सेफ्टी, क्वालिटी और एफिकेसी सुनिश्चित करने के बारे में रेगुलेटरी चुनौतियों का भी सामना किया है। यह एक सुखद संयोग है कि यह आयोजन ऐसे समय पर हो रहा है जब भारत ने माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के समग्र रूप से मानवता को लाभान्वित करने के लिए मौलिक मानसिकता में बदलाव, शिफ्ट इन फंडामेंटल माइंडसेट टू बेनिफिट ह्यूमैनिटी एज ए होल के आह्वान के साथ G20 की अध्यक्षता संभालने के साथ फ्यूचर रेडी एंड रिलिजियंट ग्लोबल हेल्थ केयर इकोसिस्टम बनाने के लिए भारत की प्रतिबद्धता व्यक्त की है। हील इन इंडिया और हील बाय इंडिया के उद्देश्यों को पूरा करने के लिए यहाँ मौजूद सभी स्टेक होल्डर्स को अपने अपने क्षेत्र में न केवल भारत के लिए बल्कि पूरी दुनिया के लिए सेफ, इफेक्टिव और क्वालिटी बायोलॉजिकल्स उपलब्ध कराने के कॉमन लक्ष्य की दिशा में काम करना चाहिए। मैं संस्थान को क्वालिटी ऑफ बायोलॉजिकल्स से संबंधित विभिन्न मामलों पर चर्चा करने के लिए भी स्टेक होल्डर्स को एक मंच पर लाने के लिए बधाई देता। यह समिट भारत में वर्तमान में प्रचलित क्वालिटी एश्योरेंस अप्रोचेस में गैप एनालिसिस के लिए एक बेस प्रदान करता है। यह देश के बायोफार्मास्यूटिकल्स और इन विट्रो डायग्नोस्टिक इंडस्ट्रीज के इंफ्रास्ट्रक्चर और टेक्नोलॉजीज को अपग्रेड में मदद करेगा और पब्लिक हेल्थ को बढ़ावा देने और विश्व स्तर के उत्पादों को विकसित करने की क्षमता में भी वृद्धि करेगा। मार्केट में स्पूरियस और नॉट ऑफ स्टैंडर्ड क्वालिटी वाली दवाएं रोगियों और दवा निर्माताओं दोनों के लिए गंभीर और प्रतिकूल स्थितियां पैदा करती है। एनआईबी ने स्पूरियस और नॉट ऑफ स्टैंडर्ड क्वालिटी दवाओं से पब्लिक हेल्थ को बचाने में महत्वपूर्ण भूमिका निभाई है। एनआईबी शायद देश में एकमात्र सेंट्रल ड्रग्स लेबोरेटरी है जो विभिन्न प्रकार के बायोलॉजिकल्स की टेस्टिंग करती है और साथ ही देश में निर्मित या देश में आयात किए जा रहे बायोलॉजिकल प्रोडक्ट की क्वालिटी पर पैनी नजर रखने के लिए नेशनल रेगुलेटरी अथॉरिटी सीडीएससीओ को महत्वपूर्ण सहायता भी प्रदान करती है। बायोलॉजिकल प्रोडक्ट के सभी पैरामीटर्स की टेस्टिंग यह सुनिश्चित करती है कि कोई भी टेस्ट पैरामीटर छूटा नहीं है जिसके कारण वह प्रोडक्ट फेल हो जाए। एनआईबी जैसी लैब की टेस्ट रिपोर्ट मार्केट में आने वाले बायोलॉजिकल्स की क्वालिटी के ट्रेंड को एसेस करने में नेशनल रेगुलेटरी अथॉरिटी को मदद करती है। और सबसे महत्वपूर्ण बात यह है कि फेल होने वाले प्रोडक्ट्स के प्रकार के साथ साथ जिन पैरामीटर्स पर यह प्रोडक्ट फेल होती है उनकी भी जानकारी ऐसी रिपोर्ट से एनआरए को लो क्वालिटी या सब स्टैंडर्ड दवाइयों के मार्केट में आने से पहले ही उनकी क्वालिटी एश्योर करने के तरीके तैयार करने में भी मदद मिलती है। एनआईबी यह सुनिश्चित करने में महत्वपूर्ण भूमिका निभा रही है कि केवल क्वालिटी बायोलॉजिकल प्रोडक्ट ही हेल्थ सिस्टम तक पहुंचे, जिससे हमारे प्रधानमंत्री जी के सभी के लिए क्वालिटी हेल्थ एंड वेलनेस फॉर ऑल के मिशन को सुनिश्चित करने को मजबूती प्रदान की जा सके। मुझे यह जानकर प्रसन्नता हो रही है कि एनआईबी ने रिसर्च प्रोग्राम्स भी शुरू किए हैं जिनके माध्यम से बायोलॉजिकल्स की क्वालिटी में सुधार के लिए रेगुलेटरी रिकमेंडेशंस तैयार करने में मदद मिल सकती है। जहां एक और देश बायोफार्मास्यूटिकल्स के लिए ग्लोबल मार्केट को लीड करने के लिए अपनी कमर कस रहा है, वही एनआरए को रेगुलेटरी इंफ्रास्ट्रक्चर को भी मजबूत करने और नए बायोफार्मास्यूटिकल्स की क्वालिटी एश्योर करने के लिए ठोस तैयारी पर बल देना चाहिए। टेस्टिंग लैब्स को रेगुलेटरी अप्रूवल के लिए आए नए बायोफार्मास्यूटिकल्स के बारे में जानकारी दी जानी चाहिए ताकि ऐसे प्रोडक्ट्स की टेस्टिंग का स्टैंडर्डाइजेशन पहले से तैयार किया जा सके। एनआरए को नए प्रोडक्ट के पैरेलल प्री रिलीज क्वालिटी टेस्टिंग शुरू करने के लिए उपयुक्त तरीके तैयार करने चाहिए, जिससे कि रेगुलेटरी अप्रूवल प्रोसेस में तेजी लाई जा सके। भारत लो कॉस्ट स्किल्ड मैन पावर और यूएस एफडीए और डब्ल्यूएचओ जीएमपी कंप्लायंट वेल एस्टेब्लिश्ड मैन्युफैक्चरिंग बेस की एक महत्वपूर्ण संख्या को संचालित करता है और दुनिया भर में लाखों लोगों को एफोर्डेबल और लो कॉस्ट जेनरिक लाइफ सेविंग मेडिसिन की आपूर्ति करता है। यही कारण है कि भारत को दुनिया के फार्मेसी के रूप में पहचाना जाता है। भारत फार्मास्यूटिकल प्रोडक्शन के मामले में वॉल्यूम के हिसाब से दुनिया भर में तीसरे स्थान पर है और 200 से अधिक देशों में उनका निर्यात करता है। हमारी इंडस्ट्रीज का देश और दुनिया भर में मानव जीवन बचाने में सक्रिय और सफल योगदान रहा है, क्योंकि भारत इस क्षेत्र में अग्रणी भूमिका में है, इसलिए हमारी इंडस्ट्रीज की जिम्मेवारी और बढ़ जाती है कि वो क्वालिटी कंट्रोल को प्राथमिकता दे। WHO के सस्टेनेबल डेवलपमेंट गोल के अनुरूप क्वालिटी हेल्थ केयर के माध्यम से एंश्योर हेल्दी लाइव्स एंड प्रमोट well-being for all at age so that no one shall be left behind। इस संकल्प को साकार करने के लिए मेरा सुझाव है कि NIB को भी माननीय प्रधानमंत्री श्री मोदी जी के भारत के हेल्थ इंफ्रास्ट्रक्चर को प्रोत्साहन देने के लिए अथक रूप से काम करने की दृष्टिकोण की दिशा में काम करना चाहिए। इस दिशा में निरंतर कार्यरत रहने से सभी नागरिकों को क्वालिटी और एफोर्डेबल हेल्थ केयर सुनिश्चित हो सकेगा। भारत जैसे बड़ी आबादी वाले देश के विभिन्न हिस्सों में बायोलॉजिकल्स के क्वालिटी कंट्रोल टेस्टिंग के लिए समर्पित NIB जैसे और संस्थान स्थापित किए जाने की भी मुझे आवश्यकता लगती है। वर्ष 2020 में दुनिया अप्रत्याशित कोविड-19 महामारी की चपेट में आ गई थी, लेकिन आज हम पीछे मुड़कर भारत सरकार के प्रयासों पर नजर डालते हैं। चाहे उपलब्ध संसाधनों और बुनियादी ढांचे का उपयोग करके प्रभावी ट्रैकिंग और ट्रेसिंग करना हो, एक्सटेंसिव टेस्टिंग करनी हो, डायग्नोसिस और टीकाकरण अभियान चलाना हो, तो हमें सरकार के इस प्रबंधन पर गर्व का अनुभव होता है। आप यह भी भली भांति जानते हैं कि इस महामारी को समय पर और प्रभावी तरीके से हराने के लिए हमारी स्ट्रेटेजीज और एक्शन प्लान्स की विश्व स्तर पर सराहना की गई। NIB ने कोविड-19 महामारी के कारण उत्पन्न आपात स्थिति के दौरान रिकॉर्ड समय में देश की पहली हाई थ्रूपुट लैब स्थापित करके और अपने रेगुलर स्टैट्यूटरी टेस्टिंग के अलावा 24/7 रोगियों के कोविड-19 नमूनों की जांच करते हुए यह भी सुनिश्चित किया कि मार्केट में क्वालिटी बायोलॉजिकल मेडिसिन की कोई कमी न हो। बायोसिमिलर मार्केट में मौजूदा उछाल ने बायोलॉजिकल मेडिसिन की सेफ्टी और क्वालिटी एश्योर करने के लिए अत्याधुनिक इक्विपमेंट उपयोग को आवश्यक बना दिया है। यदि ऐसे प्रोडक्ट्स स्वदेशी रूप में विकसित किए जाते हैं तो आम आदमी के लिए उपचार अधिक एफोर्डेबल हो जाएंगे और साथ ही हमारा पब्लिक हेल्थ सिस्टम और मजबूत हो जाएगा। रेयर और नेग्लेक्टेड डिसीज के इलाज के लिए वर्तमान दवाओं सहित नई बायोलॉजिकल मेडिसिन के स्वदेशी विकास को बढ़ाने, जीन थेरापी, स्टेम सेल थेरापी और पर्सनलाइज्ड मेडिसिन जैसे नए प्रोडक्ट कैटेगरीज पर इनोवेशन के लिए इंडस्ट्रीज, एकेडेमिया और रेगुलेटरी नेटवर्क को साथ में काम करना होगा। इनोवेशन को बढ़ावा देकर बेंच टू बेड साइड कंसेप्ट को सपोर्ट करने वाले पैरेलल रेगुलेटरी ओवरसाइट के साथ इंडस्ट्रीज के सहयोग से एक रिसर्च इकोसिस्टम तैयार करने की आवश्यकता है। हमें रेगुलेटरी अप्रूवल प्रोसेस को सरल बनाकर इंडस्ट्रीज के लिए एक स्टेबल और सपोर्टिव रेगुलेटरी एनवायरनमेंट बनाना चाहिए और अपने ग्लोबल पीयर्स की बराबरी के लिए इंटरनेशनल रेगुलेटरी बॉडीज के साथ कोलैबोरेट करना चाहिए। क्वालिटी कंट्रोल लैब्स में नए बायोलॉजिकल्स की टेस्टिंग के लिए अपनी क्षमता बढ़ाने के लिए पर्याप्त फोरसाइटनेस होनी चाहिए ताकि ऐसी नई मेडिसिन जनता के लिए जल्दी से जल्दी उपलब्ध हो। NIB ने एकेडेमिक्स और रिसर्च के क्षेत्र में जो पहल की है वह निश्चित ही माननीय प्रधानमंत्री जी के जय जवान, जय किसान, जय विज्ञान और जय अनुसंधान के विजन को चरितार्थ करती है। यह प्रशंसनीय है कि एनआईबी ने इस क्षेत्र में ट्रेन ह्यूमन रिसोर्स की आवश्यकताओं को भी महसूस किया और राष्ट्रीय कौशल्य विकास कार्यक्रम की दिशा में पहल की है। मैं एनआईबी से यह कहना चाहूँगा कि इस विशेष क्षेत्र में क्वालिफाइड ह्यूमन रिसोर्स तैयार करने के लिए ट्रेनिंग प्रोग्राम को और मजबूत करें। अपने संबोधन के समापन पर मैं एनआईबी के 31वें स्थापना दिन तथा आज आज़ादी के अमृत महोत्सव के मौके पर एनआईबी के सभी कर्मठ कर्मचारियों को बधाई देता हूँ, जिन्होंने अपने अथक प्रयासों से बायोलॉजिकल्स के क्वालिटी कंट्रोल के क्षेत्र में विशिष्टता बनाए रखते हुए संस्थान को राष्ट्रीय और अंतर्राष्ट्रीय मंच पर प्रतिष्ठा दिलाई है। मुझे आशा है कि आप न्यू एंड हेल्दी इंडिया को बनाने में सेफ, इफेक्टिव और क्वालिटी बायोलॉजिकल्स की दिशा में निरंतर कार्य करते रहेंगे। आप सबको मेरी ओर से हार्दिक शुभकामनाएं। धन्यवाद।
Can I have Dr. Charu on stage? Dr. Harish? Okay, thank you. So I think सचिव सर को कहीं जल्दी पहुंचना था, हम ऑलरेडी थोड़ा सेशन लेट कर रहे हैं। डायरेक्टर सर के ने सर बोल के गए हैं कि कार्यक्रम आप चालू रखें, तो उनकी एब्सेंस में मैं डॉक्टर हरीश से निवेदन करूंगी कि मंच पे उपस्थित हमारे सभी वरिष्ठ अतिथिगणों को मोमेंटोज भेंट करके सम्मान करें। We have first श्री सी गोपाल कृष्ण सर। सर एनआईबी की तरफ से एक छोटी सी भेंट आपके लिए। श्री राजीव अदावत सर। श्री जयदेव मिश्रा सर। आप सब लोगों का हार्दिक आभार। अब मैं अनुरोध करना चाहूँगी डॉक्टर हरीश चंद्र, जो कि डिप्टी डायरेक्टर क्वालिटी कंट्रोल एनआईबी के हैं, कि वे डायस पे आएँ और अपने सभी अतिथिगणों का आभार प्रकट करें।
Hi, good morning to all of you and namaskar. On the behalf of the National Institute of Biologicals, Noida, Ministry of Health and Family Welfare, Government of India, it is my profound privilege and great honor to extend a vote of thanks for the National Summit on Quality Control of Biologicals. I must mention our deep sense of appreciation and gratitude for our chief guest, Honorable Cabinet Minister, Dr. Mansukh Mandavia, for his precious time, motivational message, and for extending support to NIV. Thank you, sir. A heartfelt thanks to the guest of honor, Dr. Bharati Praveen Pawar, Minister of State, for her message, motivation, and support to NIV. Thank you, ma'am. I'm very thankful to our honorable guest, Shri Rajesh Bhushan, Health Secretary and Chairman of the governing body of NIB, for sparing his precious time from busy schedule for the summit and grace the occasion. His knowledge, wisdom, understanding, experience, drive, and energy always guide us. We will certainly engage institutes from G20 countries for quality testing of the biologicals for collaborations. Thanks again, sir, for your guidance. I express my heartfelt thanks to our Honorable Director, Dr. Anup Anvikar, for guidance and motivation in all our efforts for making this meeting a success. I express my sincere thanks to all participants and representatives from industry, academia, research institutes, CDSCO, IPC, and other government agencies who have come from all the parts of country and shown a keen interest for their active participation. My sincere thanks to all the officials from the Ministry of Health and Family Welfare, Shri Gopal Krishnan ji, Shri Jaideep Kumar Mishra ji, and Shri Rajiv Vadhavan ji. Thank you all for gracing the occasion. Thanks to all staff of NIB, event management team, and staff of Vigyan Bhavan, and also CISF for their contribution to the National Summit on Quality Control of Biologicals. Thank you. Namaskar.
धन्यवाद सर। अब मैं सबसे आग्रह करना चाहूँगी कृपया खड़े हो जाएं। नेशनल एंथम हम चलाने वाले हैं। आईटी विभाग।
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
मैं निवेदन करना चाहूँगी सबसे कि अब कि हम थोड़ा सा डिले कर रहे हैं, तो अभी चाय के लिए सबकी व्यवस्था, जलपान की व्यवस्था नीचे एट्रियम में की गई है और हम ग्राउंड फ्लोर एट्रियम पे हैं और हम 11:30 बजे अब नेक्स्ट सेशन शुरू करेंगे. We'll just have 15 minutes more and please be seated by 11:10. 11:20. Yeah. Sorry. Kindly be seated. We need to start the session shortly. I welcome you back. Dr. Maheshwari, इनका तो टॉक है मैं अब सबको बुला रही हूँ एक साथ। I welcome you all for the first interactive session of the day. As we informed you all in the inaugural session that we'll have 3 sessions today, and we are beginning with the one where the focus will be on regulators. We are very pleased to introduce our chair for this session, none other than Professor Atul Goyal, Director General of Health Services, Ministry of Health and Family Welfare, Government of India. Sir, please, I invite you on the stage. With him, I would like to invite Sri Rajesh Maheshwari ji, CEO, National Accreditation Board for Certification Bodies. Sir, please join the stage. I request all the speakers to be on stage. Dr. Gaurav Pratap Singh from IPC, Dr. Rubina Bose from CDSCO. We have Dr. Dimple Kasana, Director, Central Research Institute, Kasauli. Then we have Dr. Mala Chandra, DGM, Haryana Medical Supplies Corporation. So in a way, we are covering CDSCO, IPC, Government Medical Supplies Corporations. So we are looking forward to a very good interactive session. I would like to now hand over the dais to our chair, Professor Atul Goyal, to start the session.
So we begin with the session. It's about assuring quality of biologicals in India. Fortunately or unfortunately, we are in an era of biologicals. Like we had once an era of stents, devices. Today we are in an era of biologicals. So they are being used in every field, starting from immunotherapy as far as connective tissue disease is concerned, to cancers, and they are being tried in other fields also. For example, allergic reactions and dermatitis. So till the era lasts, we need to ensure the quality of the available products, both the imported as well as the indigenous. So we start with the first session that is being taken first, talk that is to be taken by Dr. Anup Anvikar, Director NIB, and he needs no introduction. And he'll be talking about quality testing of biologicals. He is the one who is responsible for the quality testing as it is from the government sector. Dr. Anup.
Thank you, sir. Thank you very much. May I have the first slide, please? We have been hearing since the morning, there is absolute need of making quality and affordable commodities available to all the strata of the community. The Sustainable Development Goal number 3 talks about ensuring the healthy lives and promote well-being for all at all ages. That does not mean that the quality medicines or the commodities will only be available to the rich people of the community, everybody. So that is why we have gathered here, and there are all the stakeholders, including the policy makers. I was talking to the director CGHS just a few minutes back, and I told her that you are an important stakeholders of ours, because the biologicals which are going to the CGHS or whatever system, they should be all quality assured. And when we talk about the target 3.8 of the SDG, it talks about the universal health coverage, and it focuses on the importance of the quality of the essential commodities. Next slide, please. Since there are variety of the stakeholders in the gathering, I would like to define the biologicals, then why the quality testing of biologicals is important, and why this meeting, and then couple of minutes about our institute, what are the challenges faced for regulation of the biologicals and the way forward. And of course, I will only be enumerating couple of things in the way forward, and we expect that we all will form the roadmap together. My team will be noting down the discussions, and that slide will be populated at the end. Next slide, please. So, biologicals are the substances which are derived from the natural sources, and they are used for preventing, diagnosing, and treating various diseases. The humans have been using the biologicals since the millennia, but their importance has really increased, and we have been manufacturing them, and we are conducting all sorts of scientific research on the biologicals in the recent years. And there are different terminologies like biologicals, which are the innovator products, biosimilars, which are in line with the generic products, and there are biobetters also, so which are the improved version of the existing biologicals, and they are the future of the medicine because they have the targeted efficacy and tolerability. When we talk about chemotherapy for a cancer, so along with the cancer cells, they will also destroy the normal cells, but the biologicals, they will specifically act on the target cells so that there are not much side effects, and their production is really challenging compared to the traditional drugs. If we take the example of say aspirin on the left hand side in that figure, so it is a small molecule manufactured by a chemical reaction of two different molecules, and its size is hardly 20 atoms. When we talk about a typical monoclonal antibody, its size is more than 20,000 atoms, and it's like comparing a bicycle with an aeroplane when we talk about a chemical entity and a biological. Next slide, please. These are few examples of the biologicals like vaccines or the blood products, monoclonal antibodies. There was one, the trastuzumab or tocilizumab was pretty common name during the COVID-19 pandemic. Then there are gene therapies, recombinant proteins, various types of diagnostics. It's an exhaustive list. This is just to quote certain examples. Next slide, please. So why we need to emphasize on quality control of biologicals? We should be little more careful while dealing with the biologicals. Why? Because these are large and complex and heterogeneous mixtures of different types of agents, and their source is natural one. So there is variability of that source as well, and generally they are made up in genetically engineered cells, and those cells will also have their own variabilities, and which will again make our biologic variable, and their manufacturing as well is a little complex process, and there could be many challenges during that. So we really need the quality assurance program for the biologicals. And lastly, the handling of the biologicals not only during the manufacturing process but also during their transport and storage is a challenging thing, and this even makes our drug surveys little difficult. Next slide, please. National Institute of Biologicals, so we were created around 31 years back. It's an autonomous institute of the Ministry of Health and Family Welfare, and we are into quality testing of different types of biologicals, and we are also into research and training. We also run one important program, the Hemovigilance Program of India. We are ISO 17025 accredited for the specified scope of biologicals, and we are also a WHO collaborating center. Next slide, please. So, for testing of the biologicals, we have different types of laboratories like the labs for testing of the diagnostics, vaccines, anti-sera, biotherapeutics, and there are certain central facilities to support them. Like whenever any biological is to be released or while it is tested, sterility is an important component. If the biological is not sterile, then we are actually infecting the person. Then we have many other facilities. I won't go into all the details. Next slide, please. So, we carry out the quality testing of biologicals for regulatory purpose. So, before their release in the market, then also for procurement purpose. So, we also have many procurement agency representatives here. What the procurement agencies do, before the procured product is released to the different governments, they conduct a quality check for that and they send it to our institute. And we are also a notified central drug laboratory as well as medical device testing laboratory. And our scientists are gazette notified as the government analysts as well as the medical device testing officers. Next slide, please. This is our portfolio of different biologicals which are tested here. So, we have been gradually increasing our portfolio, we are expanding, and currently we are testing around 302 to be precise, types of biologicals. And on the right hand side in the table we can see there are different types of products like blood products, then different diagnostic kits, enzymes, hormones, recombinant proteins, vaccines, etc. And I would like to reiterate here that we would like to engage with all of you to make the processes simpler if you feel that they are complicated. And we have recently rationalized the sample requirement for over 200 types of biologicals, and we have worked extensively in reducing the turnaround time as well as the cost in case of certain diagnostics. Next slide, please. And we would like to further work with you to expand our portfolio to add more and more biologicals so that we are future ready. As you can see in the graph, I do not have the pointer. So, we are testing around 2000 batches of different types of biologicals per annum. Although we have a capacity to test say around 5000 or 7000 batches per year. So, we would like to have more batches even voluntarily, and we will work with our regulator colleagues, and today also we can have certain preliminary discussions regarding that. And as we can see, around 2.7% of the biologicals on an average they were reported as not of standard quality, and we are actually preventing such biologicals from entering the market. Now, which are the parameters on which these biologicals are failing? So, in case of diagnostics, it could be sensitivity, specificity. In case of the biotherapeutics, it could be potency or any say transfusion transmitted infection, or it could be a pyrogen test, or it could be solubility or protein content, sodium content, and many other things. So, they look to be really smaller parameters, but when it goes into the body, it will have serious repercussions. Say if we talk about say for example any blood product, and if it is producing agglutination in a dilution in which it is not supposed to produce, what will happen when it goes to the body? So this is a really serious affair. And on the right hand side there is a table, it is really very difficult to read. So these are the different classes of biologicals which were declared, certain batches of them were declared as not of standard quality. Actually we do not want to emphasize on that, that's why the font is smaller. Next slide please. So we are also into research and training at NIB, as Secretary Sir told in the morning. This year we started a PhD program, and currently we have 12 students enrolled with us, and you will be happy to know we have got one seat reserved for our industry colleagues. And through this gathering we would like you to nominate personnel for conducting training with us. So we think that NIB, all stakeholders, industry colleagues should work together. We should send our students to industry, industry can send their students to us, and we can really strengthen the quality of the products which are used in India. Recently, we also started our own postdoctoral fellowship in line with ICMR, CSIR, etc. We have also started a certificate course on biologicals, which will help to create trained manpower for the industry. Sir, 30 seconds more. So, next slide, please. So, talking about the regulation challenges, so the biologicals are diverse, raw material is from the biological sources, there are no reference standards available for many biologicals, then transportation requirements, and in case of diagnostics, since the incidence of many infectious diseases is declining, courtesy our DGHS sir, so we find it difficult to find the cases with those infections, say for example HIV, HBV, and it is difficult to get the panels. Hence the QC testing is challenging. Next slide, please. This is my last slide. So the way forward, we need to standardize the QC testing even before the marketing approval so that there is no time lost. So for this, we will need support from all our industry colleagues. Then we will need to harmonize the testing methods, particularly in case of biosimilars, which will help all of us. Then we definitely need research to improve QC testing of biologicals, and we will need to replace the manual assays with automated ones so that there are objective results, no confusions at any end, and we are also saving time. And we will need to strengthen our QC testing as well as regulatory procedures, and of course capacity building is really important. Next slide, please. So thank you, and we invite you all to actively participate in this meeting so that we get more and more inputs from you. Thank you so much. And I'm sorry, sir.
Sorry, actually you told me you will take 8 minutes. So probably you assessed yourself wrong. Anyway, doesn't matter. Any comments or questions would be over lunch, I guess. So I would invite the next speaker, that is Shri Rajesh Maheshwari, CEO, National Accreditation Board for Certification Bodies. He'll be talking about accreditation, assuring quality, regulatory compliance, and facilitating trade.
Thank you, Chair, and thanks to NIB for inviting QCI, NABCB to this very, very important event. I have a short presentation, and if my colleagues in the IT cell can quickly run through it. Coming to the next slide, please. I'll be presenting how the national accreditation system is helping the entire ecosystem, whether it's the government, regulators, industry, or any other stakeholders. So if you look at the international trade scenario, we have the trade organizations or the those bodies which are governed by the international trade agreements like a TBT agreement or SPS agreement, which is WTO, and we have the national regulators here in CDSCO and other regulators within the country. And for any product, process, or service, we have standards, international standards like ISO, IEC, ITU, Codex, lots of other international bodies, and then we have the national standards body which lays down the standards. And we have the national accreditation system, which is linked to the global system and the international bodies like ILAC or IF. And this is how any product or process needs a conformity assessment, and the traceability of measurements, the metrology, which is NPL in India, or for radiation standards BARC, and then BIPM at the international level. Next, please. So if you look at the entire ecosystem for quality, we have the standards bodies, we have the conformity assessment bodies like certification bodies, inspection bodies, testing laboratories, calibration laboratories. And to have the confidence in the conformity assessment, we have the accreditation system in place. Next. If you look at the Indian ecosystem, as I already mentioned, the national standards body, QCI is established by the government as the national accreditation body. NPL is there, and we have the Department of Legal Metrology for the legal metrology aspect. Next. I'll skip this just to tell you that we were established in 1997. We are an independent autonomous body under the Department of Commerce and Industry, and the mandate was to establish the accreditation systems within the country and help the Indian industry establish quality when it comes to both the domestic and international markets. Next. So this is how we are structured. We have 4 accreditation boards. NABCB is one. NABL, everyone knows about it. NABH, everyone knows about it. NABIT is another board, and we'll talk about how this accreditation system functions in the country. And we have other divisions within QCI. Next. Yeah. So what is accreditation? It's an international system for having confidence between the economies on the conformity assessment results. And accreditation is a third-party attestation, so anybody who doesn't have any interest, user, supplier, buyer interest, so that gives a assessment of that body that it's competent to perform that conformity assessment, whether it's a test, inspection, or certification. Next. So when you talk of conformity assessment, when you talk of testing, inspection, certification, so there are, as you all know, first party, second party, third party, but as the risk increases, and definitely in biologicals the risk is very, very high, so the third party conformity assessment is the most preferred route globally. Next. And if you look at the international trade agreements, I was referring to the technical barriers to trade, the TBT agreement of WTO or SPS agreement or the UNEC recommendation, everyone talks about the word accreditation. That means when you are verifying compliance of a product or a process to a standard or a regulation or any industry spec, it needs to be done through accredited bodies, and that's where the confidence is coming around the world on accreditation system. Next. So we at NABCB, it's I think lot of schemes, and that is why you can't see it. 5 broad areas that we give accreditation to. One is management system certification, whether it is 9001, which is very, very simple, quality management systems, 22,000 food safety, or maybe 13485, which is medical devices quality management system. So we give accreditation to these certification bodies which are providing these certifications. Then we have the product process certification bodies. We have the bodies which are certifying the skills and competence of person. So we call it personal certification. We have the inspection bodies, and we have the validation verification bodies. And all these are as per the international standards. And when I talk of validation verification, of course, everything is important for this sector. So when a new product or a new process and you want to validate against the limitations, against the assumptions, against the methods, so validation verification bodies. And verification is against claims. So if a product or a organization is making some claims, so it can be done through the accredited verification bodies. It's a new standard which has come, 17029, which came in 2019, and it's applicable to all industry sectors, including biologicals. Next. NABL, you all know about it. So they are the another constant board under QCI, and congratulations again to NIB for getting that Professor S.K. Joshi Award. First award you got it in this year. And they gave accreditation to the testing, calibration, medical laboratories, PT providers, reference material producers. And reference materials is one of the key elements. You were talking about the controls. Proficiency testing is another one. Not only accreditation, but these two are other areas which are of quite relevance to the biological sectors. Next. So this is how the system exists, and both NABCB and NABL form part of the international system. And ultimately it is to provide confidence, trust, assurance to all stakeholders, which is the government, regulators, consumers, purchasers. And every level there is an assessment happening. We face international peer evaluations, and then we evaluate the competence of these accredited bodies. Next. So I have my stopwatch running here so I can see that. So this is how we are internationally recognized. We are recognized, there are 2 international bodies, International Accreditation Forum, that's for the certification validation part, and ILAC is for the testing, calibration, medical inspection part. And these 2 international bodies are also destined to merge. Decision already been taken, and by 2024 this will become one single international organization. And there are different regional bodies. So we fall, India comes under Asia Pacific region. So we are members of APAC. We are not only members but signatories to the multilateral recognition arrangement. So we get internationally peer evaluated every 4 years. So does NABL. And once we are successful in our evaluations, any accredited reports or certificates issued under our accreditation will have an international equivalence and acceptance. Next. So this whole concept is about 7-8 decades back, just emerged after the Second World War. And now the whole objective is anything which is tested, inspected or certified once should be accepted everywhere. Now we are all talking of technical barriers. Nobody talks about tariff barriers. And that is why this is all more important. And when you look at G2G agreements in the morning inaugural session, the Secretary Health was also mentioning about having bilateral agreements. So this will help the NIB or any other institution or any other government body to have bilateral or multilateral agreements. Every G2G agreement, whether it's a bilateral or multilateral agreement, these days have the components on standards, conformity assessment and accreditation. And since NIB is a premier institute, I would suggest that not only we should adopt international standards, but we should take a lead in development of the international standards also. Next. So this is how it is structured. There are 5 recognized regions, regional bodies: AFREC for Africa, EA for Europe, APAC for Asia Pacific, IAC for both North and South Americas, and then we have AREC for the Gulf countries. And these are all internationally peer evaluated, and then we are evaluated, and then we evaluate the competence of these conformity assessment bodies. This is how the whole chain works. Next. If you look at this map, all blue ones are the countries which are signatories to the MRAs. And if you look at Europe, America, Asia, only part is Africa and West Asia that you can see. Next. So now how does this MRAs at accreditation level helps? It helps, as I mentioned, if the government or the regulator wants to have G2G agreements, conformity assessment is very, very important. And this becomes the basis of accepting each other's conformity assessment results, accredited conformity assessment results. And that's where the national accreditation system has an important role to play. Next. We are recognized by many international certification scheme owners in diverse fields, ranging from food to environment to aviation to other areas like forests also. Next. Within the country, we are recognized by almost all government ministries, departments, regulators. In fact, CDSU and their medical devices regulation prescribe NABCB accreditation as a prerequisite for the notified bodies. Food Authority in their food safety auditing regulation prescribes that. CPCB prescribes that. PNGRB for oil and gas sector. They have an MOU with us. They rely on our accredited bodies. Bureau of Energy Efficiency. So all those regulators, they rely on us. Industry heavily relies on the accredited bodies. Next. So these are some of the benefits, main technical competence and international equivalence and acceptance. I would suggest that you go to the 2 sites given at the bottom, publicsectorassurance.org and business-benefits.org. Globally, you can see how countries have benefited, sectors have benefited around the globe on using accreditation. Next. I skipped 30 seconds. So last slide. How does this sector? Management systems, you can use it for the manufacturers, processors, suppliers, vendors, anyone. If you are sourcing your products from your vendors, then product process certification of your vendors through our accredited bodies. Inspection, if any consignment, public procurement consignments, or maybe any inspections of facilities that accredited inspection bodies would do. Validation verification, I've already mentioned about testing of products. Any product needs to be tested, accredited laboratories, even reference producers or PT providers. Medical testing, once a human is sampled, I mean say human being is subjected to a particular biological drug or a medicine, then if his sample needs to be tested during clinical trials, that can also be done. And most important, certification of persons. We have lots of people working in it and at different levels. If the certification and of their competence skills is required, it is done through our NABCB accredited personal certification, but it's all as per international standards. Thank you. I think this is it.
Thank you, Mr. Maheshwari, for a good enlightening talk on the NABL, NAB assessment and certification. Next to on pharmacopoeia, and I would invite Dr. Gaurav Pratap Singh to talk of role of IPC in setting pharmacopoeia standards. That we are all aware of the term pharmacopoeia, and at times we still have certain products which are not part of Indian pharmacopoeia and yet are purchased by institutions as well as national programs. So we need to understand about the importance of pharmacopoeia standards.
So thank you, sir. And I thank NIB also for giving us this platform to present our work. So can we have my slide? So I'm Dr. Gaurav, working as a Principal Scientific Officer in IPC. Next one. So just like NIB, IPC is also an autonomous institute under the Ministry of Health and Family Welfare. And this IPC was established in the year 2009. And this year we have completed 14 years of our existence. So there are basically three bodies which are guiding us. First one is the general body, and next one is governing body. These are basically policy making bodies, and chairman of these bodies is Secretary Health and Family Welfare. Our scientific work is reviewed by the scientific body, and currently Dr. V.M. Katoch, who is the former Director General of ICMR, is chairing this scientific body. Within the IPC, we have two sections. One is IPC Secretariat, that is responsible for the development of monographs, and laboratory activities are supported by IP Laboratory, which includes the verification and validation of monographs as well as the development of reference standards. Next one. IPC has several mandates. First one is the publication of Indian Pharmacopoeia, which includes the monograph standards of the drugs which are currently marketed in India. Second one is the development of reference standards, and these reference standards are used in the monographs to assess the quality of the medicines. We also publish the National Formulary of India, that is NFI, and IPC is also the National Coordination Center for the Pharmacovigilance Program of India, that is PBPI, which is a WHO recognized program. IPC also organizes several skill development programs and trainings for the stakeholders, including industry, regulators, analysts, and for students also. And the last one is the national and international collaborations. So we collaborate with the other organizations which have similar mandates. Next one. So Indian Pharmacopoeia, as all of us know, that it is a book of standards, drug standards, as per the provisions of this Drugs and Cosmetic Act. So the monographs which are prescribed in the IP, they are official in nature, and they are used by the stakeholders, including industry as well as by the regulators, to assess the quality of the drugs which are marketed in the country. Next one. So this is the monograph development process. So the process starts with the selection of the drugs for which there is a need to develop the monographs. So there is a specific criteria based on which IPC decides to develop the monograph, and we source the specifications for these monographs from the manufacturers, and based on their specifications we develop the draft monograph, which is published on IPC website for inviting public comments. And we also circulate these draft monographs to the stakeholders for their comments, and once we receive these comments, these comments are discussed in the expert working groups, and the final monograph is then approved by the scientific body before its publication in the IP. Next one. So this is the criteria for the inclusion and exclusion of monographs. So IPC usually prefers to include the drug monographs which are given in the national health programs, which are used in the national health programs of India, different programs, and the drugs which are included in the national list of essential medicine. And the drug must be approved by the CDSCO. We do not include any drug that has not been approved in India by the CDSCO. And if any other drug that is suggested by any expert or industry, they are also considered for the development of monographs. For exclusion, we usually exclusion is usually based on two criteria. That is, if the drug has been banned in India or it has become obsolete. That is, if it is not currently being in use. Next one. So these are different expert working groups of IP. So currently we have around 23 expert working groups which guide IPC on different classes of medicines. For biologicals, we have separate working groups like recombinant DNA products, there is a separate working group. For blood products, there is a separate working group. For vaccines also, we have a different working group. Next one. So this is the brief history of IP additions. So the first edition came in 1955, and last year on 1st of July, the 9th edition, that was IP 2022, was released, and it became official from the 1st of December 2022. Next one. So this is the basic structure of IP standards. Within the IP, you can find that each volume will have, initially it will have some blue pages. These are known as general notices. So the text that is given in the general notices, it is mandatory and it is applicable to all the pharmacopoeia texts. Then we have general chapters also. So there are, per se, these general chapters are not mandatory, but if these chapters are referred in any individual monograph, then they become the part of a standard. Then we have general monographs. So in the second volume of IP, you will get, initially there are general monographs which are given for various dosage forms like tablets, capsules, injections. And so the requirements of general monographs, they are mandatory to comply. Then we have specific monographs, which are product-specific monographs. So these monographs are developed based on the approved specifications. And any drug product that is marketed, it should comply with the specifications of these specific monographs throughout its shelf life. Then the next part is the reference standards. So the quality of any drug should be assessed based on the methods and specification given in the specific monographs, along with the use of these reference standards, and which can be sourced from the IPC. Next one. So this is the current status of the monographs. So currently we have 3,152 monographs in IP, and the last IP 2022 edition, it has included 92 new monographs, 12 new general chapters, and many of these monographs, they have also been revised. And within the IP monographs, we have different categories of monographs like formulations. There are around 1,200 formulations, then APIs, more than 900 APIs. And in the categories of biologicals, there are different, these products are categorized among different categories like vaccines, RDNA products, blood products, likewise. Next one. So IP reference standards and impurity standards, they are developed by the IPC laboratory, and they are available for sale and used by stakeholders. They can go through our website and they can order these standards. So IPRS, they are developed through a collaborative approach, and our laboratory, they are accredited by ISO 17025 as well as ISO 17034, and our laboratory is also accredited as per ISO 17043 for proficiency testing provider. So these standards are authoritative in nature, and they are used to assess the final quality of the product if there is a case of doubt or dispute. So currently there are 663 IPRS and 427 impurity standards. They are available from IPC, and you can you can visit our website to order any of these standards. So among the different pharmacopoeias, we are on the third number. USP and European Pharmacopoeia, they have more number of standards, but we are the third one when it comes to the number of IPR reference standards that are available for any pharmacopoeia. There are other standards like vaccine standards. They are, IPC actually authorized CDL Kasoli for supplying these NRS, National Reference Standards for vaccines. And we have also authorized IMTech Chandigarh, CSIR IMTech Chandigarh, for supplying the reference microbial cultures. Next one. With the help of the Ministry of Commerce and Ministry of External Affairs, we are proposing to different countries that the IP should be recognized by other countries also. So currently this is the status, like currently IP has been recognized by 4 countries. First one was the Ghana in West Africa, then Afghanistan was also has also recognized, and later on Nepal and Mauritius, they have also recognized IP within their legislation in their pharmacy acts, along with the USP and BP. They recognize the IP standards. So and we have MOUs with the British Pharmacopoeia as well as US Pharmacopoeia to collaborate on the processes of setting standards. IP has an observer status in the EDQM, so we participate in the meetings which are conducted by Pharma Europa. And recently, IP has also been inducted as a pilot member in the PDG. PDG is a pharmacopoeia discussion group of 3 pharmacopoeias. One is the US pharmacopoeia, then European pharmacopoeia and Japanese pharmacopoeia. And the mandate is to harmonize the journal chapters and the excipient monographs within these pharmacopoeias. So this is a pilot project for next 1 year, and we are participating in this project. Next one. So these are different organizations with which we are collaborating. So various CSIR labs, they are helping us in developing the monographs in different categories of drugs. We participate. We have MOUs with the USP and BP. Next one. So these are the areas where IPC and stakeholders, they can collaborate. Industry stakeholders, they can share their specifications for the development of any new monograph, which they consider that it should be a part of IP. And they can also donate their candidate materials for the development of reference standards. And also we can collaborate for organizing any joint seminars, workshops, and trainings. Next one. So I would like to thank Ministry of Health, Family Welfare, CDSCO and their labs, NIB, CSIR labs, and various pharma companies for helping us in the development of these standards. Next one. Thank you.
Thank you for finishing well in time. So I would invite the next speaker, the regulator from CDSCO, Dr. Rubina Bose, to discuss about regulation of biologicals in India.
Thank you, sir. Thank you, Chair, and good afternoon to all. And it's really proud and I'm privileged to be here for this in this summit. So while talking about the regulations of the biologicals, I would, so can we go to the next slide? Because we have a very limited time. So I would like to start with it. So we'll go through the slides very first, just to save the time. So can summarize initially, the biologicals are considered as new drugs as per the new drugs and the clinical trial rules under the Drugs and Cosmetics Act and rules for the purpose of the regulation. And so when it is new drugs, it means that it has not been used in the Indian population to a significant extent to establish the safety and the efficacy. So as it was already said that these are very highly complex heterologous medical products, and hence there has to be specific consideration in respect of the development of the chemistry manufacturing control data, non-clinical data and the clinical data, which will provide a clear understanding of the regulatory landscape for their development and approval in a scientific manner. So these products are required to be characterized and manufactured in compliance with the good manufacturing practices. The manufacturing processes for every biological products are to be validated, defined and controlled adequately to ensure batch-to-batch consistency. I would emphasize again for the regulatory standpoints. And this is important even before the drug is marketed. So it is important that adequate data on the quality, safety and efficacy. In general, these are to be generated before the approval to establish the safety and effectiveness of this product throughout the product life cycle. So I would now go to the slides very first. Please, next slide. So this is what I said about the introduction. The next slide, please. Some of the definitions which are well known, two are from the WHO's, and one is from the biosimilar guidelines, the guidelines and the new drugs clinical trial rules, where we define similar biologics as comparable to the reference biologics which is already approved in the country or innovator's product. Next. So these are the various bodies which are also along with CDSCO are responsible for the regulations. We can go to the next, very specific to the biologicals. Next slide, please. So the specific to these, there is the other than CDSCO who is responsible for giving the permission for the. So it starts with the in the stepwise manner. If we consider, then it's the starting with the manufacturing, giving the manufacturing license for test and analysis or for the preclinical development, and then so here along with the state drug controllers are also responsible for initially licensing a facility for the manufacturing of the test batches, either for the preclinical development stage, and then comes the finally when the final manufacturing licenses are also given, then also the state drug controller along with the CDSO are responsible for the joint inspection and licensing, and otherwise the CDSO is also responsible for the import and registration of all the biological products, and also the for giving the permission for the clinical trials. So all cases the pre-approvals are required for any phases of the clinical development. So in the preclinical stages, this is the RCGM, the review committee on the genetic manipulation of the under the Department of Biotechnology are responsible for the research and development at the preclinical evaluation stage up to the preclinical stages, and also for the import of the strains or for the control of the hazardous substances, and specifically the genetically modified or living modified organisms. The genetic engineering appraisal committee of the Ministry of Environment and Forest and Climate Change are also responsible. And then we also have the Central Drugs Laboratory, so the CDL Kasoli for lot-to-lot release as well as NIB. So next slide, please. So these are the regulatory guidelines. We go to the next. So regarding the dossier requirements, the dossier requirements which is in the SUGAM online follows exactly the CTD modules, the common technical documents as per the ICH, where the module 1 is on the overall summary, the module 1 is on the administrative information, module 2 being the overall summary of the non-clinical, clinical and the CMC part, module 3 which requires a detailed requirements starting from the developmental history, from the strains, the cell lines, the cell banks, the entire characterization. So it requires, there is a specific requirement of capturing the entire developmental history of the cell bank, master cell bank, working cell bank and the seed trace, as well as all the stages starting from the next stage from expansion, or in case of the biosimilars from expression in the specific vector. So it needs a complete characterization as I mentioned above. Along with this, then the development of the manufacturing processes stepwise for all the purification steps up to the purification and finally the product. So and the complete validation of the analytical procedure is a requirement, as well as validated assay methods are essential for establishing this. So this what is indicated in the CMC, and it really requires a complete validated process as I mentioned earlier. So then there is a module 4. The slides are there. So there is a module 4 which overall is a non-clinical summary, and the module 5 is a clinical trial data, so which is a summary of all the phases of the clinical trials. So we can go to the next slides, please. So as we say that in case of if we go in a specific manner, then as I already mentioned about the chemistry, then the non-clinical, there are various toxicological data, which is a specific toxicological data or a specific pharmacological data which are needed to establish the safety efficacy in the animal models and sometimes in the relevant species, but it depends from indigenous products which are to be manufactured in the country where there are extensive studies are needed, and there can be some relevant species studies or an abbreviated toxicological study when the product is already, the safety of the product is established, or in case of the biosimilars. But in case of the vaccines also the animal challenge studies is important, and also the study is required in the specific age considering the specific immunogenicity is also essential when there is a vaccines to be developed. So that is what is the non-clinical part. Going to the next clinical part. So, so here this is also the detailed, and where a product is to be used for a longer time. There are additional studies, or when it is to be used in the special population, like the different specific population, then it has to be studied in those populations, and such we call them as a developmental reproductive studies, which are also needed additionally. So next. Coming to this clinical trial phases, as we have said that the first phase 1, 2, 3 are as such required. Sometimes it is important to have a traditional design, like a placebo-controlled randomized study is actually needed, but in some cases this is also what we have seen during the COVID. Also the adaptive designs are acceptable, where we can have the phase 1 by 2 or phase 2 by 3 as per the requirement, and then they can be gradually from one phase to another. The study can go as per the plan and the design, and it has to be statistically significant, and also it is required to be in the, so while the phase 1 is being planned in the healthy subjects, the phase 2 being planned and in a smaller number of people in general, and in case of the vaccine always it has to be a age de-escalation study, and in case of the phase 2 it can be in case of vaccines definitely in several hundreds, and also it's to be in the specific age group, the dose interval, those are to be captured. And what is required is in the phase 2, normally while in the phase 1 there has to be a 3 dose is being expected. The last dose in the phase 2 should be lesser than the dose should be less than the last dose of the phase 1, and in the phase 3 it is being captured, it is being done in several thousands. In case of vaccines it is expected, whereas in the case of the biosimilar there are different guidances. We can put, so next slide please. So this is the pathway which goes in the SUGAM online, the different levels of review, and along with the subject expert committee, the evaluation is being carried out. So they are responsible for the evaluations of the clinical trial protocol, as well as for the safety evaluations in case of the periodic safety update reports, which is the post-marketing. Yes. So quickly we go through the slides only, right, sir? So next slide, please. Next slide. So these are all the different application process. Next slide. So this is I'll finish on this. So the accelerated approval process and the expedited review process, which are the two which are under in the NDCT rules, have enabled to do the various approval process in an expedited manner or in an accelerated approval manner, considering the severity of the disease, prevalence of the disease, and if there is a life-threatening disease, where the approval could be carried out even based on some surrogate markers, or on the basis of where phase 2 trial there is a sufficient safety and efficacy data, then such approval could be carried out. So these are the regulatory enablers which have helped us in reviewing various approvals. Next please. So this the processes which has been, next please, next, and these are the biosimilars which I have already said. It is next. So where there is an the abbreviated procedures are followed with a pharmacokinetic study and a phase 3 study in usual. Next, please. Okay. Next. Thank you, sir. Thank you, sir.
Acha. Well, actually, there is a small issue. I and my Shuri have to leave, so we'll be leaving the session in between, and we'll leave it to Dr. Anup to carry on. And I think there is some problem in today's teaching. I think Dr. Y.K. Gupta must remember, even during my times, we used to have something like Pressy writing, which is a forgotten art. So at times, even I am used to taking 1-hour lectures, and Dr. Y.K. Gupta must also have taken those lectures in AIIMS. So sometimes when you have to put those lectures together in the form of a talk in a conference or a session, it becomes a little tough. And I generally avoid that completely. So I'll take leave along with Mr. Maheshwari. Before that, I'll invite Dr. Dimple Kasana for the next talk on vaccines, ensuring quality in vaccines. Dr. Dimple Kasana is director of CRI Kasoli, and while I'll not be there to listen to your talk, I apologize for that.
So pardon us, but we don't want you to leave without the gift. So can I get it? I would request our director, Dr. Anup, for Goel sir, to present a memento for Professor Goel. Thank you, sir, for your session. Since Maheshwari sir is also leaving, I guess. Yeah. Thank you, sir, for joining this session. Gaurav, group photo. Over to you, ma'am, please.
Good morning, everyone. Dignitaries on the dais, my dear colleagues and friends. I'm here on behalf of Central Research Institute Kasoli. We are manufacturer of vaccines and antisera. I've been asked to talk about the quality management system in vaccine production. Of course, this is a work which we do in and day out, and quality management system is integral part of any manufacturer who's making vaccines as such. So we do comply with all the regulations. So with this, I start with my presentation. So why do we say we need a quality management system? We need a quality management system because first and all, it's a regulatory requirement. Like whenever there's a regulatory requirement, you have to comply with it. So that is why that's the first and foremost thing, but that is not the only thing why we have a QMS. Second and the most important part is it builds and assures the quality, safety and efficacy in the product what we are making. That itself gives us a assurance, or the end user when they're using it, they are assured of the quality and that's why they use it. So that is important for us to get their faith in us, or to get that belief that what we are giving them as a product is the worth using it. So that's another requirement why we are going for it. As far as the regulatory requirements are concerned, we have the different frameworks. These are the national frameworks and the international frameworks. In the national frameworks, we have the Drugs and Cosmetic Act, which kind of forces us to follow these procedures. So it's like a policing there, which asks us to follow this, and we follow this. By the Schedule M quality system, which actually talks about the GMP. Second is the Indian Pharmacopoeia. My colleague had just emphasized on this Indian Pharmacopoeia as such, so I'm not going in detail here. The international regulations, if you are going to export, you have to follow those international regulations, like WHO regulation, it could be the European regulations, USFDA, or any which which particular country you aim to supply it. Since I'm talking about the QMS in vaccine, so I just would like to talk about certain definitions here. So what we mean by QMS as a definition, it's a management system which is a wide-ranging concept covering all matters that individually or collectively influences the quality of a product. Or we can also say that it is a totality of the arrangements made with the objectives of ensuring that products are of quality required for their intended use. So it's a model for pharmaceutical quality system that can be implemented throughout the different stages of a product life cycle. So in all these three things, we are just saying the same thing, that whatever you are manufacturing, you have to be assured of the quality, and that quality is a constant process which will be checked at each step you are taking into that while you are manufacturing it. So as we talked about the QMS, I'm also talking about the vaccine and what we understand by vaccine. So as per definition, it's a biological preparation that consists of either a whole organism or a part of it against which immunization is to be achieved. All of us know there are 4 types of vaccine. I'm not going in detail of it, but broadly we have live vaccines, we have killed vaccines or inactivated vaccines, we have toxoid vaccine, which we are making the diphtheria, pertussis and tetanus. DNT are the toxoid part, and the subunit vaccine. So the process of the vaccine manufacturing involves the upstream process and the downstream process. Those who are familiar with the terms, they would know it. Those who are not, they can see the first 2 steps are made up of that is known as the upstream, and the below 3 steps are the downstream. That is, first and foremost, you select the strain out of which you want to make the vaccine, and then of course you have to grow that microorganism. And later on that is a downstreaming process. You have to isolate and purify the microorganism. You have to inactivate that organism, and then you formulate the vaccine. And then there's a quality control and the lot release. These are the journal and steps. And if we go in details in that, as you said, in each step, the quality checks are maintained. So right from the selection of the raw material selection to the final product when it goes to the end user, in between is the process of the manufacturing. So it starts when you're selecting your raw material, and once you've selected your raw material, you're assured about the quality, you go for the production part, in which the detailed parts I shall elaborate again. But once the final product is also made, then also your responsibility is not finished, your quality checks are still there. That is till the time it uses that is the transportation part, that you assure that it is transported at a cold chain or whatever temperature required for that particular vaccine. So this is all the 3 stages in broadly: before manufacturing, during manufacturing, and post-manufacturing part of it. So this is the 3 parts that you saw. The raw material part, that is pre-manufacturing stage. In process QC, when you are manufacturing the vaccine, and the final product QC, that is when you are sending it to the end user. So these are the various sub parts of it. When they are selecting the raw material, you have to select the air quality, water quality, the chemical models, or the production material. You also see in the process when you're doing the staining test, inactivation test, physical test, biological activity test, and when your final product is made, you check the quality of that product which you have made by physical test, identification test, stability studies, and the VVM monitoring. VVM is basically to see the stability part when you're transporting it, that is post-production, that it remains whatever product you have made, the end user using the same product. It has not changed. Next, please. So this is a diagram which just says that how QMS is inbuilt in your whole process, right from the selection, that is R&D portion, right? When you're doing the R&D till you inactivate the product, in each stage, the QMS is inbuilt. That is in the helm of the affair. You see in the center is a QMS. So whenever you're manufacturing or selecting or doing R&D or producing the product or even discontinuing it, your QMS is at the heart of it. It is maintained. And each step you're doing the process control, you're doing the change controls, and you're doing the CAPA. This is for each step. Each different step, like when you're doing your R&D, when making the product, you're doing the technology transfer, or you're commercially manufacturing, or when you're inactivating the products. So these 3 steps would always be there. That is why the QMS is not a standalone procedure. It is a part and parcel right from the beginning of the idea, of the conception of the idea, till the time the user uses it. Next, please. This is again, I have given the detail of it, the strengthening of QMS and vaccine production. These are the various steps of the vaccine production and how each step has to follow the QMS qualification. Whenever they're manufacturing, not even a single step would go where the quality checks are not maintained. The first 3 diagrams, as you can see, that is your seed, and then from you grow it. After growing, you harvest it, you purify it, you make a bulk, then in the bulk you add your additives, your adjuvants, stability factors. And after addition, here also QMS has been checked, and then of course it goes the filling part of it. After filling, you have to do the examination, that is a naked eye examination, or there are tools available if you want to use that. And then of course the labeling part of it. Label, of course, you know, you have seen the vaccines, you know the label contains all the details about the manufacturer. It tells what is the product, what is the route of administration, what is the expiry. All those things are checked. And then of course the packaging, and then it finally reaches the person who are intended to use it, or to the clinician, or directly to the patient as far as the conditions are. Next please. So this is again the same diagram which tells you how, like if it's in our facility we have selected the raw material. The material comes to the warehouse. The warehouse, it's been picked up for the quality checks. The quality checks are maintained, and once they are sure about the quality, then only it goes for the production part of it. And once the production is done, the various stages of transportation where again the cold chain is maintained, and then it goes and reaches to the end user. Next please. The topic actually say how do you strengthen it. Of course the quality checks are there, the books are there, everything's there, you have your SOPs. But when you say the QMS, you have to see that your SOPs are being followed. You have the SOPs available for each step, but those steps have to be followed. Like if there's a parent seed when you're selecting, you're thinking about a new vaccine. There are many of my friends here who would like to make a vaccine. They're going in a field of vaccine manufacturing, or at least they intend to do that. So for that, they have to have a parent seed first. When they have a parent seed, they have to check the origin, history, its source, certificate of authentication, absence of advantageous agent. As the name says, advantageous agent, I don't know why they say that, because I feel it is actually checking that there's no contaminant in that. So that's, how does it advantage? I don't get it, but that says so. So it's you check for the absence of the contaminants, the quantity, and the characterization. In the characterization, you see all aspects is physical, biochemical, serological, molecular, physiobiochemical. And once these tests are done, these are not one-time tests. After parent seed, then you have a master seed, and then of course you have a working seed. You make master seed and working seed in your own facility wherever you want to make that vaccine. And again, the same steps are repeated. Regarding the origin, history, these are one-time steps. But when you do the characterization, that you do each time you are taking a working seed out of it. So these are the steps which you are doing one time, and these are the steps which you are doing every time a working seed is made for the vaccine purpose. Next please. So these are the various steps. I'm not too sure if you'd like to go in the details of each step, but it again says if you want to just break down each step and then talk about the details of it, that how QMS has been maintained. Of course, right from the seed strain that you do the integrity of the container, the purity part of it, the labeling of the thing. You would like to check the sterility of the media if you're inoculating the seed in that media. You would like to check at the stage of harvesting by identity, yield, and absence of contamination. During purification process, again you will check for the yield and the absence of contamination. Next please. Then the detoxification and inactivation part. Our QMS team would identify the specific toxicity, any irreversibility, absence of contaminants in the pH. Stabilization and preservatives are added, and once they are added, they are also not without the check of QMS. There is a qualification. And the stabilization, you do the quantification of stabilizers, you do the quantification of preservatives also. Next please. Yes sir, 30 minutes. The last few slides sir. Then of course these are the characterization of vials. When the vials which you're using for your vaccine, you have to do the sterility, pH, potency, volume, and identity test, along with the preservative, stabilization, and the endotoxin content test. Next please. Next please. This is of course the same thing we have talked about. The QC is during storage, transfer, and use, and your role does not finish once you have manufactured it. Your role is still there till the time it reaches the end user, so you have to maintain that cold chain. And you have to do the stability studies. This is a stability study during product development, during licensing, and also after licensing. That is an ongoing stability study. So as I said, it is an ongoing process. It does not stop at any step which we take. Next please. These are of course the particular example given for our own institute. The steps are same as we already enumerated for vaccine production, and of course the various checks which we incorporate. The QA control right from when the seed is received, that control is with the division which handles the quality part. Under their control only the seed is released. Even the detoxification or purification, that also needs a quality checks. Your sterile filtration, formulation, and even when the product in the market, the role of quality management team does not finish there. They are checking it at every step. And these are the products what we are making, the vials which you see, we can making the DPT vaccine, we are making TD, anti-seras, and that's all from our institution, and that's the end of it. Thank you so much.
So we have been talking about the importance of quality assurance, the regulations, the monographs, the QMS since morning. But one of the most important links between the public health system and the manufacturers is the procuring agency, which makes the bulk of the purchases for the public health system. So that is why the procurement agencies are very important stakeholders in the quality assurance process. So I now invite Dr. Mala Chandra, who is the DGM of the Haryana Medical Supplies Corporation Limited, and she will talk from the perspective of the procurement agencies as far as the QMS is concerned. Over to you, Dr. Mala.
Thank you, Dr. Anup, for inviting me here. यहाँ उपस्थित सभी अतिथिगण को मेरा सादर प्रणाम. And I would like to thank and congratulate the whole NIB team on their foundation day. 31st foundation is a great achievement. And first of all, I will, before start, as Dr. Anup also said, why quality? What is quality? We all know. Then why and how? In my pharmacology, I'm an MD pharmacologist. In the first day, we learned that a patient is coming to me with a disease. Yes, and we are giving a drug. That drug has its side effects. But due to poor quality, mera mareez, my patient should not get another disease. For example, if the injection is not sterile, it is having a bacteria or a fungus, we are giving him a new disease, that is the fungal disease, and it will lead to fatality. And that is a punishable crime. And therefore, it is all our moral responsibility to supply and issue the drugs with a good quality. We all can purchase, we can afford the branded drugs, right? But being a part of my government institution, we are here, designated here, to provide good quality drug to my poor patients also coming to the government hospitals. They're very poor. They can't afford the drugs, branded drugs, or even can't they purchase the most important drugs from outside. They're fully dependable on our dispensaries and my hospitals. So that's why a different department was set up by Haryana government for the procurement and the supply chain management, ensuring the quality, stocks, and other things. That is Haryana Medical Services. Please, the slides. It was established in 2014, and we have an essential list of, that is almost 400 drugs to 500 drugs, that we make every effort that should be available 24/7 in my hospitals. And we have other purchases also, that is including the other, that we purchase on demands basically. That is maybe the hospital bed sheet, it may be the program medicine, it could be anything. So the basic funda is कि my drugs, every item, even the bed sheets, even the gloves, even the syringes, including all the medicines, we all get it tested before in our NABL accredited impaneled labs, and our biologicals are only tested from NAB since beginning to ensure the quality. चाहे वो टेस्ट हुआ हो, पहले नहीं हुआ हो, even sometimes we get pressure कि मैडम ये तो डोनेशन से आया है, why we are getting it tested? We need not to do the payments. तो क्यों करना? कागज ही तो पूरा उनको लगता है. Sometimes people think it is just a formality of the policy to complete the paper. A quality report is a paper कि वो लगेगा और payment हो जाएगी. And when we are buying from the government agencies, suppose government bodies, CPSU है, other bodies also, then also we get it tested before distributing it to my patients. Before ensuring, because जैसे कहा कि क्वालिटी आपने निकाला ड्रग अपनी मैन्युफैक्चर ने, इट इज़ ऑब्वियसली गुड क्वालिटी होगा, बट व्हेन इट इज़ कमिंग टू माय हॉस्पिटल, इट इज़ कमिंग थ्रू मेनी रूट्स. टेम्परेचर है, ट्रक में आ रहा है, कैसे आ रहा है, बारिश में आ रहा है, कैसी पैकिंग है, दैट इज गोइंग टू अफेक्ट द क्वालिटी ऑफ एनी ड्रग. सो वंस it is in my warehouse, it is tested, then only it is distributed to our hospitals. So for that, we have a mandate in our policy. We have a HMSL purchase policy on which it actually works. And two skeletons are of Haryana Medical Corporation. That is our policy. And the second is our DPMU portal. That is the online portal on which we are connected through every hospital, our ministers, our stakeholders, everybody is connected through a portal. Because इतना बड़ा state है तो हम daily mails पे और इस पे तो हम बात करके नहीं चल सकते offline में. तो we have a drug. I will discuss in the next slides how our portal works. And please next slide. That is our portal. That is a drug procurement and management unit. It is our backbone basically. ये हिल जाता है तो सारे हिल जाते हैं. बहुत we get sudden calls. मैडम ये दवाई नहीं दिख रही हमें, ये नहीं दिख रहा and all that. इसमें क्या है कि स्टार्टिंग फ्रॉम डिमांड फ्रॉम आवर हेल्थ फैसिलिटीज़ कि हमें ये ड्रग इतने अमाउंट में चाहिए, एनीथिंग वी हैव डिमांड इन आवर पोर्टल. अकॉर्डिंगली वी परचेज दैट अमाउंट बाय गोइंग मैनी डॉक्यूमेंट्स, tendering procedure, everything. And everything is on the portal. That is to maintain any transparency, and it is under monitoring of direct our health minister. And it is very honored that our health minister, Honorable Anil Vijji, he is always with us to ensure the quality. He never outdates कि quality test नहीं होगा. हमेशा ही इज ऑलवेज ऑन माई साइड. चाहे वो कोई भी प्रेशर रहा हो, चाहे वो हमारा कोविड पेंडेमिक रहा हो, पहले हर ड्रग टेस्ट हुई है देन ऑलवेज. चाहे वो एम्फोटेरिसिन रहा हो, चाहे वो कुछ भी रहा हो, लेकिन वो टेस्ट होने के बाद ही गया है. वो अप्रूवल देते ही नहीं है कि विदाउट टेस्टिंग तो बटेगा ही नहीं है सामान. तो इट इज़ वेरी प्राउड कि वी आर वर्किंग विद सच हायर अथॉरिटीज़. नेक्स्ट इज अर पोर्टल. पोर्टल में एवरीथिंग इज देयर, स्टिल द पेमेंट ऑफ आर फॉर्म्स, ताकि उनको भी दिखता रहे कि कहाँ पे ड्रग लाई कर रहे हैं, वेदर इट इज़ इन क्वालिटी टेस्टिंग पाइपलाइन में है या कहीं पे भी है, स्टॉक्स में है, कहीं भी है. दैट इज़ ऑल विजिबल इन द पोर्टल. एवरी हेल्थ फैसिलिटी कैन सी आवर पोर्टल और वो देख सकता है कि कौन सी ड्रग कितनी है, क्वालिटी में है तो कितने दिन में हमारे पास आ जाएगी. नेक्स्ट प्लीज. दैट इज़ आवर पॉलिसी. पॉलिसी में हमारा मैंडेट है. वी हैव आवर अदर इम्पैनल लैबोरेटरीज ऑल्सो, NABL, बट इन बायोलॉजिकल्स, सेराज, वैक्सीन्स, this is a mandate in our policy itself. It's written that it will get tested from NIB only. Our next is pass or fail. If it is passed, then it is passed in the portal only. Then it is available to all the specialists. They can see the stock. But in case it is declared as not of standard quality, then it is blocked in our portal, and that is not available for any stakeholder to see that drug. वहीं पे ब्लॉक हो जाएगी, वो जाएगी ही नहीं. दैट इज़ वाई द पोर्टल इज़ ए मैंडेट. ऑफलाइन में गलती हो सकती है बट पोर्टल में ऑटोमेटिकली वो ब्लॉक हो जाएगा, वो आगे दिखेगा ही नहीं किसी को. नेक्स्ट. और एक बहुत स्पेशल कि हमारा ये है पॉलिसी में कि वी हैव अ पॉलिसी कि द फर्म और मैन्युफैक्चरर कैन कंटेस्ट कि आपका एक एनपायरल रिपोर्ट से टेस्ट नॉट ऑफ स्टैंडर्ड क्वालिटी आया है, प्लीज़ हमारा रीटेस्ट करवा दीजिए. बट विद NIB they cannot challenge. We refuse their challenge that it is already has been tested from a renowned NIB, so it could not be retested. That is, once it is declared not of standard quality by the NIB, it is a not of standard quality and they have to replace that product. Next, biologicals. We have learned already a lot, so I will skip that slide. Now, importance. It is a very good platform for the quality of biologicals, as biologicals are target on the special target cells. So their quality needs to be maintained at most priority. That, and especially when it is leads NIB, when we send the samples, even sometimes we have an issues. Recently the sample was received on Sundays, so we were very worried कि पता नहीं courier वाला पहुँचा पाएगा कि नहीं. There are practical problems we face actually. Then we recently calling Dr. Gauri, really successfully, thanks to her, she takes the calls on even non-working days. She helps us a lot. The NIB team whole कि मैडम सैंपल पहुँचा कि नहीं कहते. मैडम don't worry, our every staff is trained to receive the sample at the correct temperature. अगर वो correct नहीं होगा, that is returned to us. So this is a very important part in quality इंश्योरेंस कि टेम्परेचर जब पहुंचा ही नहीं वो सही टेम्परेचर पे तो होगा ही नहीं. दैट इज़ नॉट द फॉल्ट ऑफ मैन्युफैक्चरर. that is the transport. So there are many minor factors जो हम कभी देख ही नहीं पाते, that will be rejected. So every step is taken care of कि वो जो है सही टेम्परेचर पे रिसीव हो, टाइमली रिसीव हो. So they have facilities on non-working days also. Thanks to the team, they support us so much. And next slide. Next is my acknowledgement to everybody from NIB and especially Dr. Rashmi and the whole entire NIB team for taking care of us for our minor requirements during accommodation, transport and everything. Thank you. Jai Hind.
I congratulate the Haryana State Medical Supplies Corporation for such stringent policies for quality checks for not only biologicals but also for other commodities. And I am sure all other medical supplies agencies must also be following such best practices. And now I am realizing that this one day is not enough for a stakeholders meeting, and next year when we organize such a meeting we should have a full session on just procurement agencies. So this is one of the take home lessons for me, madam. So thank you very much.
अगर कोई यहाँ पे from the manufacturer side है या firms है, I would like to request them all please for money don't compromise on the qualities. चाहे वो पैकिंग हो, पैकिंग बहुत इंपॉर्टेंट होता है. गोली निकलती है, घुर जाती है, पेशेंट कंप्लेंट करता है, उसको खाने को ही नहीं मिलती बेचारे को दवाई, वो पाउडर हो जाता है. सो प्लीज़ इट्स माई हम्बल रिक्वेस्ट, सिर्फ लोएस्ट रेट के लिए L1 के लिए क्वालिटी कॉम्प्रोमाइज़ ना करें. आप अपना स्टैंडर्ड बढ़ाइए, वी हैव एनफ मनी, वो हो जाएगा अरेंज. L1 के लिए क्वालिटी नहीं कॉम्प्रोमाइज़ कीजिए, ये हमारा एक मॉरल ड्यूटी है, एथिकल. सो प्लीज़ मेरा हम्बल रिक्वेस्ट है कि डोंट कॉम्प्रोमाइज़ ऑन द क्वालिटी. Thank you.
Thank you, Dr. Malla, for being this vocal. But our industry colleagues, they definitely take ultra care to ensure that the packing is also absolutely perfect. But then really good that you raised this issue. Packing is also very, very important. Yes.
तो मेरे अपने पेशेंट की तरफ से रिक्वेस्ट है, जिनका इनकम टैक्स से आई एम गेटिंग माय सैलरी, उनके लिए प्लीज डोंट कॉम्प्रोमाइज द क्वालिटी. दे शुड गेट गुड मेडिसिन्स.
We are really running out of time, but still we will take a couple of 2 or 3 questions. Definitely we are going to have 2 full-fledged sessions on the regulatory aspects of the biologicals, followed by the session on industry, academia, connect. But if so we 5 people are sitting here, so if you have any questions for us, they are most welcome. So while questioning, kindly introduce yourself and then you can ask your question. And if we find at the end of 2 or 3 questions that the time is not enough, then you can just write them on a slip and hand it over to any one of us, whoever is wearing this type of badges. So we will take care that they are deliberated upon and replied also. Yes, Dr. Rajiv Tandon. So he is the director of the Research Triangle Initiative.
Thank you, Dr. A great meeting today. Just want to congratulate you and all. My question is that we've been hearing a lot about nutraceuticals, and when I read your definition of biological, it appeared that nutraceuticals also can fit within the biological framework. Is the regulatory pathway for nutraceuticals going to be the same, or will it be somewhere else?
Using that only.
Okay. Needs to be quality assured. So, Madam Mala needs to take care of it.
अभी हो गया? क्या? हाँ. So can you hear me?
So that I was just predicting that the first question will be directed finally towards me only, the regulator. So was not ready for the questions. Anyways, I was thought that the session is over. Anyway, sir, thank you, sir. Thank you for the questions. No issue.
There will be more opportunities for the regulatory questions
Sure,
in
sir.
subsequent
Sure,
sessions.
sir. What I was saying is that definitely the nutraceuticals, though from the similar origin, are not regulated as per the vaccines or the biotherapeutics, as we were discussing throughout these days. So these are definitely in a different part of it, and we don't regulate it as presently under the Drugs and Cosmetics Act and rules. Though there is a FDA has a requirement under the food and the USP also, but right now. It is still regulated under the FSSI, and we have not yet planned any, so it's not regulated under the DNC Act and rules, so the same regulation doesn't apply to the nutraceuticals.
It's Dr. J.P. Prasad.
मेरा क्वेश्चन नहीं, मेरा एक रिक्वेस्ट है CBSEO से, तो blood product testing.
Request है.
For HIV, hepatitis, TBI testing. So no pharmacopoeia has given for this product testing. We are doing as per Drug and Cosmetic Act. So this we have requested in 2010 and 11 when our director Dr. Jyotna Soke, then Director Sundar Singh was the DCGI. When Dr. Singh became a director of NIV, again we have requested, but that has not been changed. So this is the request. Kindly do it. Because reason behind that, ki unnecessary we are wasting the kit and doing the and in international pharmacopoeia there is no they have not given in any pharmacopoeia, even in the IPC. Number one. And one question from Mr. Gaurav, ki so far as testing protein concentration of blood product is concerned, we are doing by the Geldal method. Suppose any product having the stabilizer as glycine or any amino acids, we will do the testing by the Geldal method, that will affect the protein concentration. So there should be another method like a biuret method in the pharmacopoeia.
Recently we have changed the method also, if in the IP 2022.
No. Okay. Not for this biuret test, test they have not given this method.
So there were, I think, 5 or 6 methods they were given.
They have given, but not for the blood product.
Okay.
That's why I am especially asking for the blood product.
Okay, that we will look, sir. We will.
In case of immunoglobulin, they are adding stabilizer amino acids, so they affect the concentration of protein.
Okay, sir.
Any other
Sir,
question?
sir,
Yes, please.
one question, sir.
Yes.
So, I hope I'm audible. And I just want to say that you had a concern, Dr. Anup, about the panel preparation. This is an ongoing process, and NIB was established. The first thing we did was to prepare the panel. And I think drug control and DGHS had given an order that we could go to any hospital or only government hospital, and we had tied up with them. If there were any positive samples for HIV, HBsAg, or HBV or HCV, they would telephone us, or we would contact them, and we would send our staff to get. And then we used to characterize. That characterization takes time, and we have to be a part of external quality control system, be a part of it, and also conduct for others. So I think that that could be renewed again, and you will have the gold mine with you, which is a wastage for the others.
No, absolutely right, madam. This is a very important issue, and in fact, I have offered collaboration to many other labs, and we are ready to help them to for their capacity building to prepare the panels and also to start the QC testing.
Yes, and it can be done because we have been doing it earlier.
We all should work together, and it should. Yes, please.
Sir.
We will have one more question after this. Okay. Then our second session will start.
Sir, my name is Rishi Kumar. I'm from Indian Pharmacopoeia Commission, and I basically belong to Haryana. So my question is to Dr. Mala Chandra regarding quality of medicine. That it is very impressive to note that from them that they are checking the quality of medicine before distributing it to the patients. So my question is related to pharmacovigilance. Sometimes if some medicines
So lack of quality or lack of efficacy, or some medicines are of poor quality. Then what you do with that manufacture of such medicines? Are you blacklisting them, or are you reporting such events to the Pharmacovigilance Program of India? What is your stand on the, such reports?
I would like to intervene before Dr. Mala replies. First thing is that if a product is reported as a not of standard quality, then there is no question that it will enter the public health system. After that, I do not think there will be anything to be done with that, but then I will go with what.
First of all, if it is declared not of standard quality by the labs, any lab, NIB, या फिर empanel labs, then it is blocked in the portal. It has not been distributed, still lying in our warehouses. It is not available, it is not visible in the portal for any stock. It is there blocked, and the manufacturer is bound to replace the entire quantity. First. Second, if the same drug is not declared not of standard quality 3 times, आपने supply किया, again not of standard quality, again, और आपने 3 batches दिए थे तीनों 3 instances, anything, then it is blacklisted.
So what about, do you have any mechanism for reporting of lack of efficacy drugs? Say for example, it passes the test. Your test has been passed in the laboratory, but the drug is not acting actually on the patient. Then what is the?
Then in that case, we get complaints from our clinicians. Suppose recently we are dealing with the complaint of his injection Misoprost. That is not effective, contractions nahi aa raha hai. Like they report us through emails, phone calls, anything. Mainly we take emails, then also we block the drug. And then also the manufacturer is bound to replace that drug. And if, regarding the adverse reactions, it may be shivering and all that. They are reported, their officers are there, pharmacovigilance, from that I was there in the field, then I was reporting to the PGMR Chandigarh. उनकी टीम में अपनी pharmacovigilance की, Dr. Vikas Mehdi की, then we report there the side effects. But if there is any complaint, efficacy है या severe कोई reaction है, then again it is blocked. That is called as administrative block.
Okay, ma'am. Can you please help us to establish, because I'm from Pharmacovigilance Program of India. So from this platform, I request you to kindly issue a letter to all your healthcare professionals who are using your drugs to report such events to Pharmacovigilance Program of India. And there are various tools are available for reporting of such events, like helpline number is there. It's very simple to note, 1800-180-3024, which is 30 days and 24 hours a day. It can be remembered like that, 30 days 24 hours a day. This is the helpline number. Additionally, there are various ADR monitoring centers all across Haryana as well as India. So they can report adverse event to the hospitals, ADR monitoring centers. Additionally, we have some mobile app also. So through your platform, if this message passes to the healthcare professionals, I hope that healthcare professionals who are using the medicines can report such events. Sometimes such events are not being reported by the healthcare professionals, and ultimately it will strengthen our healthcare system. This is my request.
You can directly upload on that portal.
I would like to intervene here, and I am really overwhelmed by the types of discussions which are happening here, and there is real need of having such meetings at frequent intervals. I am sorry we will not be able to take up more questions because our next chair, Professor V.K. Paul, Niti Aayog member, is on his way, and he is going to reach this venue within a couple of minutes, and he, along with Dr. Y.K. Gupta, will be chairing this session. So over to you, Rashmi ji.
Yes, so we have to have the felicitation round right now. I'll request you, now you being the chair of the session, I would request you to kindly felicitate. We can start with Dr. Mala Chandra. We have Dr. Rubina Bose. We have Dr. Dimple Kasana. have Dr. Gaurav. Thank you all for joining this session. You can proceed to your chairs, and within 1-2 minutes, we'll set up the dais for our upcoming session. Meanwhile, I can let you know about, like I said in the morning, the second session now will focus on industry, and we'll see the regulatory aspects of biologicals from the viewpoint of industry. The session will be chaired by Dr. V.K. Paul, member Niti Aayog, needs no introduction, and Professor Y.K. Gupta sir. We fondly know him from AIIMS New Delhi. Currently, in AIIMS New Delhi, he has been there since 1983, and currently he's president of AIIMS Bhopal and Jammu. We have many interesting topics currently being in the line. We'll be starting with the Atmanirbhar IVD industry at the point of inflection by Shri Jatin Mahajan, Secretary ADMI. And then we have Shri Rajendra Kumar Suri with us, CEO DCVMN, who will be telling us about the regulatory flexibilities, the vital need for pandemic preparedness and response, a manufacturer perspective. Followed by him, we have Dr. Jitendra Sharma, CEO AMTZ. He'll be telling us about bridging innovation and medtech infrastructure. Then we'll have Shri Vijay Kumar, Head Portfolio Solutions and External Affairs, Roche India Private Limited. He'll be telling us more about enabling policies, promoting diagnostic medical devices. And last but not the least, we have Dr. Shalini Menon with us, the country medical lead and head general medicine, Sanofi. She'll be talking about biotherapeutics, strategies, and challenges. We are pleased to have Dr. Paul with us right now. Can I please request Dr. Paul and Dr. Y.K. Gupta to kindly come on stage. Next, I request all the speakers to take their places on the dais. Can I have them on the dais? Shri Jatin Mahajan, Shri Rajendra Kumar Suri, Dr. Jitendra has come, Dr. Jitendra Sharma, Shri Vijay Kumar, and Dr. Shalini Menon, please. Over to you, Dr. Paul.
Thank you. Namaskar. Good afternoon. Apologies for the delay, but we'll go straight into the session, and I'll make my observations after the session. So we are going to discuss the regulatory aspects related to biologicals, and to address this, we have 4 interventions on IVD industry, enabling policies, biotherapeutics, and strengthening quality management. So, इसको शुरू करते हैं. I'll request Jatin Mahajan, Secretary ADMI, to kindly make your intervention, Jatin. Thank you very much. You are Managing Director at J. Mitra & Co. and Secretary Association of Diagnostics Manufacturers of India. You have 55 patents at J. Mitra and a business across 45 countries with 72 products. Please, over to you. We are discussing the regulatory aspects related to biologicals on this auspicious day of National Summit on Quality of Biologicals. Thank you.
Thank you, Chair, and good afternoon, everyone. It's indeed a great honor and a pleasure to be part of the first National Summit on Quality of Biologicals. I would also like to congratulate NIB and its team on its 31st Founders' Day. Next slide, please. So, basically, I would like to highlight diagnostics in two quotes. And these quotes are very pertinent quotes to set the context and also to highlight the relevance, significance of diagnostics in today's context, in today's times. The most crucial step towards healing is having the right diagnosis. Next slide, please. And if the disease is precisely identified, a good resolution is far more likely. So what I want to communicate is that the diagnostic sector has come as an age, and earlier it was under the carpet or behind the curtains, but now during the pandemics, the importance and what value it adds in diagnosis has been realized by one and all. So what I would like to communicate is now diagnostics as a tool is used for prevention and prediction to save lives, and at the same time it is to improve the quality of lives. Next slide. I'd also like to talk about the market size of the diagnostics. Pre-COVID 2019, we were around 100 billion Indian rupees. And post-COVID, it is supposed to be growing at a rate of around 7.5 to 8% per annum. And we are expected to be, by 2027, we're expected to be around 175 billion Indian rupees. And these data have been taken from Invest India websites. Next slide, please. Also, there is another interesting WHO data which I'd like to share.
Can you repeat that? Can you just repeat the estimate?
Previous slide, please. Post-COVID, we are growing at a rate of around 7.5 to 8% per annum, and by 2027, we will be around 175 billion Indian rupees. And this data is from Invest India website.
Okay, thank you.
Okay, thank you. Next slide, please. So another interesting data, which is the average life expectancy of an Indian human being, which is increased by 19 years in the past 40 years. And this has been made possible due to the investments in diagnostics and healthcare capacity building. Next slide, please. So as we all know, and it's a fact, that 70% of the clinicians or therapeutic decisions are based on these diagnostic tests. Next slide. So to give you a practical example, in COVID times, we have all seen that, diagnostics has been used as a very, very potent tool in the fight against the pandemic. In fact, if you look at the strategy which has been deployed globally, it is the 3T strategy, which is the testing, tracking, and then treatment. So, my dear friends, diagnostics is one of the most important, vital elements and strategy in the fight against any particular infection or disease outbreak. Next slide, please. So let me also talk about some of the past growth factors which have been influencing the growth of the industry, and these factors are relevant today and even tomorrow also. So first of the factor is the advancement in the technology aspect. Then there is also an uprise or uptake in the insurance policies, which has led to the rise of the insurance sector. Then the other aspect is the changes in the lifestyle of people, which has led to an increase in the chronic disease. Also, there is an emergence of the diagnostic service labs, be it the centralized labs, be it the chain labs, be it the standalone labs, be it even the blood banks, which are all facilitating or influencing the growth of the diagnostics. I mean, I'm also saying that manufacturers is also contributing. It's a very healthy cycle. A lot of manufacturing companies are now entering this space because it's a very nascent and a very it's a sunrise sector to be in right now. And also, there is an interest by our people to do preventive health checkup, so that what I said, prediction and prevention is going to have a better quality of life. Next slide, please. So, needless to say, I've spoken about IVD, but let me also talk about the current healthcare overall scenario. And as what the Secretary of Health had mentioned in the morning, we are the pharmacy of the world. We are the third largest medicine manufacturing by volume. We are one of the largest manufacturers of vaccines in the world. We are ranked 7th globally as far as the medical value tourism business is concerned. And we are the 4th largest market in Asia after China, Japan, and Korea. Next slide, please. Now, the point here is, which brings me to the topic of my presentation, is where is IVD industry today at a point of inflection? So, basically, I'll have 3 points here, which are actually going to influence, or rather I would say fuel the growth of the sector. First one is the policy aspect. Next slide, please. The second one is innovation and the entrepreneurial spirit of our people. And the third one is the enhancement of the enhancement of the futuristic medical technologies. Now, these are the 3 elements which are the 3 pillars which will actually lead the growth of the industry into the next level. Next slide, please. So let me just briefly explain about what are the policy aspects that I am referring to here. So first one is the NEDL, which is the National Essential Diagnostic List, where the first country in the world to have this NEDL. And if it is actually implemented, it will actually blow up the Indian diagnostics market. Then the second one is the Indian Medical Device Policy. The third one is the trade shows, the Indian MedTech Expo, which is going to happen shortly. It is going to build up the brand India and also showcase some of the finest technologies which India has launched. The Export Promotion Council, because India will not manufacture for India itself, but it's going to manufacture for the world. So this is why it's very important to set up the Export Promotion Council. Then we have the IVD park. So I would suggest that, since the numbers will increase, we should have IVD parks. Then we already have PLI schemes. Then we have a future-ready India 2047 plan. How will India look at when we reach 100 years, when we complete 100 years. And then we are looking at making India self-reliant. Then we're looking at better connections between the industry and academia, because globally, this is one of the factors which is actually accelerating the pace of new product launches globally. So this is something that we need to strengthen here in India, and also we need to strengthen the lab systems at the tertiary level under the NHM schemes. Next slide, please. Now, when we look at the innovation part and as well as the entrepreneurial spirit, if you look at the data here, we have more than 5,300 startups, which talks about how vibrant our ecosystem is right now. And we have all the necessary support infrastructure in place, be it the funding part, whether it's a seed stage funding or whether it is a bridge stage funding. If you see the data here, and again it's taken by this Inc. 42 report, which is going to be which is available publicly online. So it talks about the increased, at the primary level and at the later stage also. And then the tremendous work, the wonderful work which is done by BIRAC. I mean, excellent programs which is facilitating this technology transfer and also the startup, proliferation of startups in the diagnostic space. Then we look at also our neighbor's loss is our gain. So we have now gained this kind of a visibility that India is very vibrant and we are attracting a lot of investments now. And if the timing, if we are able to capitalize on this, we will be one of the manufacturing superpowers of the world. And then right now the domestic production is around 20%, and there is a lot of scope for further expansion from 20% gradually to a higher number here. So this is the innovation and entrepreneurial spirits that we have, but yes, it is on a positive side, and we see a lot of innovations happening right now. Next slide, please. Now, when we look at futuristic technologies, these are technologies which have huge potential, and it will unlock a lot of potentials which probably we are not experiencing right now. So first one is CLIA, that is chemiluminescence assay. Then we have the predictive and preventive genetic test. Then we have the MI, the AI, the virtual reality, the neurotechnologies. Then another thing that we have seen during the pandemic times is the point of care testing, which is the home testing. Now this market was explored when we had the home test for COVID-19. But if we look at the potential it has with respect to the critical tests, whether it's HIV, hepatitis B, hepatitis C, dengue, the vector-borne disease, whether it's dengue, chikungunya, or any other test, the market is on a very positive side. The market is very huge, waiting to be tapped. So these are some of the futuristic technologies that would enable the IVD sector to go to the next level of growth. Next slide, please. So now this is what is India at 2047, 25 years down the line. This is what the forecasting and prediction would be, that, with the deployment of the three pillars that we spoke about, the government aspect, then second one is the innovation and the entrepreneurial spirit, the third one is the future emerging technologies, which I just spoke about. So if that thing is intact, then we will, it is expected that we will garner 12 to 15% of the market share. Right now, globally, we are not even more than 1%, or maybe less than 1% right now. Then we will be home to 25 emerging latest technologies of the world. And as we speak also, a lot of new technologies have emerged out. Even during pandemic, we have seen the PCR, which was relegated to a high-end research lab, came on the front, and it became the first line of testing, which was unheard of. So a lot of innovations are happening in this space. And some of the technologies where I think that we will be leaders in is basically the cancer oncology side. Then we have the critical components, which is the need of the hour. Then we have the medical technology, the ultrascan aspect. Then we have the molecular, the next generation sequencing. So roughly what I mentioned earlier was that, it's a very vibrant space that we are in right now. The market is growing at 7.5 to 8%. And so 5 years, this is what I'm sharing, that as per the estimates by Invest India, by 2027, we'll be 175 billion Indian rupees. Imagine what will be the size when we look at 20 years down the line. So with this, due to time constraints, I would like to conclude, and I would like to thank NIB for giving me this opportunity to share my views on making, on Atmanirbhar IVD diagnostic sector at a point of inflection. Thank you all. Thank you.
Thank you, Jatin. Thank you for demonstrating the possibilities. Thank you so much. May I now request Shri Rajinder Kumar Suriji to present before us on the topic of regulatory flexibilities. Over to you. Shri Suri is CEO of Developing Countries Vaccine Manufacturers Network, established in 2000. Over to you, sir.
Thank you so much.
I request all our speakers to confine to about 7 to 9 minutes, a little more crisp, so that the lunch can be accomplished by 2:15 or so. Thank you.
Thank you very much, sir. In fact, it is my proud privilege to be here, and I must thank NIB for inviting me for this prestigious session. As Honorable Secretary Health in the morning said, it is a story of access and equity. And what I would like to take you through is a story of developing country vaccine manufacturers where India has played a distinct role. And particularly, talking about the manufacturer's perspective in terms of regulatory flexibilities, and that of course is a vital need for pandemic response and preparedness. Next slide, please. Yeah. So before we delve into the aspect of regulatory flexibilities, I will very quickly give you a brief background of what DCVMN and its capabilities are, because for some of you, the name DCVMN may be new. It is Developing Countries Vaccine Manufacturers Network, which comprises all the vaccine manufacturers that you know of, including Serum Institute of India, Biologically, Bharat Biotech, Panacea Biotech in India, but also in all other countries, including Korea, Indonesia, Brazil, China, and everywhere else. So what we do is that we are a network of 15 countries comprising of 43 member companies, and we are supplying almost 170 countries globally. In fact, before COVID, it was 3.5 billion doses, which was the manufacturing capacity, and I will just share with you what has transformed over the period of time. Next slide, please. So during COVID, we deployed all the platforms, vaccine platforms that were available, whether these were viral vector, whether these were nucleic acids, VLP, or inactivated viral vaccines. So all the vaccines have been used effectively during the COVID. Next slide, please. In fact, I am very happy to share with you that there were several international technology transfers with developing country vaccine manufacturers, including AstraZeneca with Serum Institute of India and several other companies in the world. J&J having a tie-up with BioE, and of course Pfizer USA with BioVac in South Africa, and Moderna with Samsung Biological. Next slide, please. When we see, south-to-south collaborations, you will see very interesting picture emerging where Sinovac China had several partnerships in Brazil and Indonesia and other places, and similarly Sinopharm China had in Morocco and in Dubai. Whereas Serum Institute of India, very recently also you must have heard about a collaboration in Africa with Aspen, and also Bharat Biotech having internal within India with Indian Immunological for the drug substance production. Next slide, please. All this has resulted into an outcome which is amazing story. A case study, in fact, from 0 dose in December 2020, we reached more than 13 billion doses in end of 2022. And this was possible only through several factors, which I'll just share with you. Next slide, please. I am happy to share with you that out of this, 7.4 billion doses, which is more than 60% of the total global production, has been contributed by developing country vaccine manufacturers. And as you can see, the Tiranga glowing high, 1/3 of the global vaccine production came from India and deployed under the very leadership of Dr. V.K. Paul, who is sitting here. And thank you very much, sir, let's give a big hand to that. So this was a tremendous contribution by India, acknowledged worldwide, what India has really demonstrated leadership in the vaccine field. Next slide, please.
Now, what went into this were three important factors, and of course, technology was one of them. We have been discussing and talking about technology all through the day. There was another important aspect which was at-risk manufacturing by the manufacturers, and I will delve into that a little bit. And of course, the third, the most critical part was the regulatory flexibilities. Next slide, please. Now, these regulatory flexibilities, I must say that we have to say kudos to all the regulatory agencies globally for rising to the occasion and taking several initiatives and active steps to beat the unprecedented challenging situation arising out of the COVID-19. And in this, of course, EMA had taken very early steps to establish certain regulatory reliance mechanisms, and this helped the industry and also various other international agencies. And of course, you are aware that the fast-track approvals were granted none other than even US FDA, which granted 2 approvals within 7 days, and within 2 months, the third vaccine got the approval. So I think this has been unprecedented. Next slide, please. India was not left behind. Indian NRA came to the challenge, and on 20th of March itself, they issued the instructions for expedited review and approval of vaccines. And as a result, we were able to register our first vaccine on 1st of January 21. And of course, in the month of April 21, the DCGI gave the permission based on the recommendation of the expert committee for usage of other vaccines based on reliance mechanism, which are approved by the stringent regulatory authorities worldwide. Next slide, please. Now, what has happened is that, of course, we skipped one slide, which was important from WHO perspective. The WHO had put in the reliance mechanism in place, and the result was very encouraging that over 5,000 regulatory approvals have been achieved in 175 countries till August 22. And I think this is a record of one kind. And the WHO is asking themselves now that if we could do this within this period of time, why not maintain this speed in future? And I'm very happy to share with you the new director of WHO regulatory processes is very convinced that we should take this as a routine matter, that we should be working very expeditiously on various approvals. Next slide, please. The story has not been very hunky-dory. There were several challenges. I will just give few of them to you. In terms of, there were shortages of critical raw materials because of certain export bans put by US administration. There were sustained investment and funding issues which came in the way, and it is not only for Indian manufacturers but also globally. Even Moderna did not get funding in the very initial stages. And of course, all of you are familiar that the mutations and variants have been playing havoc continuously over the period of time. Next slide, please. More than that, I think what was more important was the product development funding. And I think this is very important for all of you to really appreciate that 60% of the volumes came from developing country vaccine manufacturers, whereas only 5% of the investments into R&D funding came to this particular sector, whereas 95% was given to developed markets, including US and Europe. And US gave to US companies and Germany gave to German companies. So I think it is very important that we pay a little bit attention to this aspect so that there is a reflection on disproportionate allocation versus the number of doses delivered. Next slide, please. So in addition to this, I will very quickly go through that there have been certain challenges, regulatory challenges, particularly because the different legislative requirements for all countries are not same, and they require different requirements, different framework, different procedures and different timelines and maturity levels of different regulators all over the world are not same. So this is one of the big challenge. And then, of course, next slide, please. And then the second was the fast development and approval of COVID vaccines triggered a number of additional challenges like additional validation, processing data, optimized control strategies, additional data gathered from additional number of batches, and so on. Next slide, please. The challenge number 3 was absence of accelerated PAC approval, the post-approval changes, and this became a bottleneck while there was speed in giving regulatory approval EUA, but there was no provision for PACs. And regulatory collaboration and dependency became one of the big issue because most of these approvals were based on the on the tech transfers and small bridging clinical studies, and that itself pretty well, you come from that background, that it raises several other challenges associated with that. Next slide, please. Now, in addition to that, the R&D of new variant vaccines has been impacted due to various challenges, and from the regulatory pathway, I would say it is the conduct of efficacy study itself was unethical for 8 months in the 21-22 period, early 22. So the guidance from PQ team for pathway to license was requested to WHO, and of course I talked about financing and future scenario for the variant vaccine is because of the unpredictability of the demand and financing both. And of course, the yearly boosters, whether they have to be given to whom, which populations, and other things. Of course, the strategies are coming along with that, but there are challenges associated with everything. Next slide, please. So we need to now look at what have been the examples of the best practices. And I must tell you, the global regulatory collaboration has been definitely far superior from any other collaboration in terms of reliance mechanisms. And one factor which came out is the collaborative review process, which is CRP. And I think that going forward would be extremely helpful to the regulators worldwide. Of course, creation of the expert groups like Regulatory Advisory Group by CEPI, by ICMRA and several others has been extremely helpful. Next slide, please. Now, what is one thing that we should not do in the next pandemic is definitely let's not wait for the next pandemic to happen and then start searching for solutions. I think all of us here in this hall have to act now. And a good beginning has been done with this meeting, and thanks to Dr. Anup. Thank you very much.
Thank you very much, Shri Suri. Thank you so much. We will have comments after all the presentation. May I now invite Dr. Jitendra Sharma. He is the MD and founder CEO of Andhra Pradesh MedTech Zone to talk about bridging innovation and MedTech system. Jitendra.
Thank you very much, Professor Paul and Professor Y.K. Gupta ji. Happy to be here. Dr. Anup, you are celebrating 31 years of NIB. Congratulations to entire NIB family. AMTZ, the institution that I represent, we just completed 4 years, so we are babies, and we have a lot to catch up. I have no presentation. I have a video link which I will play in a while after I complete my very 4 small brief submissions. One, look at how we consume healthcare. Most developed evidence-based healthcare economies of the world consume diagnostics. From the diagnostics, they find out if there is a disease, so they go to a doctor, and then the doctor prescribes and then they go to the drugs. So it is DX, DR, and RX. In our case, and not just in our case, cases of many economies where healthcare is not equitably distributed fully yet, we first go to the chemist shop, we complete our RX part. If medicine works, fine, we report to duty, sick leaves are taken care. If we are not fine, we end up to a nice doctor, DX. And if doctor thinks it is appropriate, DR. And if the doctor thinks diagnostics are necessary, we go to DX. And a whole lot of problems, including antimicrobial resistance, pathogens, everything comes out of this anomaly. It has to be DX, DR, and RX. It cannot be other way. So the National Diagnostics Program of India, which was released by the then Health Minister, Mr. J.P. Nadda ji, right in this hall, 2015, when I was in the Ministry of Health, I designed the National Diagnostics Program of India. Today it operates in public-private partnership in 26 states and union territories of India, which is trying to correct this anomaly in public sector by bringing in strengths of diagnostics from the private sector. However, Indian healthcare is not just public healthcare. Particularly in diagnostics, where point-of-care diagnostics could mean pregnancy test, it could mean ultrasonography, it could mean anything. It could mean dengue, chikungunya, anything, let alone cancer markers, which is provided either in home or small nursing homes or large corporate hospitals. And therefore, my second point is, let's try to break diagnostics into pieces and see where India is strong and where India is not strong. So if you see diagnostics, as my friend Jatinji mentioned, we have the imaging and radiology diagnostics, which is now creeping into other forms of diagnostics. For example, now we have automated liquid-based cytology for cervical cancer screening. Automated. Image capturing and AI-based diagnosis of what was earlier simple swabs. Then we have instruments. Instruments is where India is extremely weak at this point in time, because instruments could require 500 components. We make close to 6 biochemistry, hematology analyzers a day in AMTZ. We make about close to 100 large instruments in a week, hematology, electrolyte analyzers, and so on, and over 1,000 components, right from a simple screw to 24-layer printed circuit board to lens, to arrays that are embedded in electronics. Those are things where India has to catch up. Thankfully, our country has been strong in pharmaceutical and biotechnology sciences, and therefore we have been able to do very well in kits. So for example, at the peak of COVID, when India was consuming about 12 lakh RT-PCR test kits in a day, in our AMTZ facility in Vizag, and thanks to the support of Niti Aayog and DBT, our capacity to produce RT-PCR kits in a day is 14 lakhs a day, 2 more than the country needs, just from one campus. Against 12 lakh RT-PCR test kits. We are today net exporters. But we are exporters because India was strong in biotechnology and pharmaceutical sciences. The same strength has to be tapped from the engineering sector. Can we make lenses? Today our microscope don't have lenses which is made in India. And therefore instrument is our weak link. And the fourth is of course reagents, chemicals and so on, where we are fairly strong as well. Which brings me to the third point. What is it that we can do together? Now, regulation, which is the topic of this session, is of two kinds, broadly. One, it is the horizontal regulation, the ISO, the NABL, horizontal. Then there are vertical regulations which are sector specific, IVD, medical technology, food and so on. And within this, there are super verticals, which is the category of the products. So with medical devices itself, regulation which is a super vertical for X-ray or CT scan will be different from dengue chikungunya kits. Now these vertical regulations, if we have to learn from our system, one of the best systems India has architected is food regulation of FSSAI. Look at the number of food products, packaged drinks, noodles, every restaurant that India has, it outnumbers by many degrees the number of drugs and medical devices and diagnostics we consume. But food is doing well. What can we learn from them? I think the one thing to learn from them is how they have tapped the private sector labs and empaneled labs to check food quality. Today, we even have mobile laboratories checking food, including seafood. I was shocked because we have a gamma irradiation plant where we irradiate even seafood. I was shocked that India is one of the best exporters of shrimp, and that is being tested by a mobile food testing lab under FSSAI banner. Now, if that could be tapped, then institutions like NIB could, which is my next point, like CDSCO, empanel large number of NABL accredited labs for kit validation. That will boost the number of diagnostic companies and their ability to bring kits in the market. Now, if this has to happen, if suppose NIB were to empanel laboratories across the country for testing and validating kits, we will need panels. Exactly as I think ma'am or somebody mentioned, panels is the problem. Thankfully, next 1 quarter in Visakhapatnam in AMTZ, we would have one of largest biobanks with a capability of 1 million samples, including a partnership with FIND for inter-exchange of samples across the world, which means we will be able to produce panels for almost 96% of the diseases for which global diagnostics exists right in India. So I think commercialization and productization of panels, which is the source material, empanelment of laboratories that can do kit validation under the umbrella of NIB across the country, and changing the practice from RX, DR, DX to DX, DR, and RX. I think these would be the 3 stepping stones that can change the value-based healthcare that the diagnostic sector is about to provide. Now, with that, I am keeping my points very brief because I have to show you guys, all of you, a 4-minute video, which I will ask the organizer to put in the link. This is about AMTZ, where we produce India's first MRI, India's first CT scan, 1 million RT-PCR kits in a day, 1,000 ventilators and oxygen concentrators in a day, world's largest medical devices cluster with 126 companies, 22 Government of India centers, a medtech city which is spread across 8 kilometers, just a campus where more than 6,000 people produce over 1,200 medical devices in India for the world. Thank you.
very much. If this is the performance of India's toddler at 4 years, what will happen at 31 years? Wow, thank you, very proud. May I now invite Shri Vijay Kumar, Head Portfolio Solutions and External Affairs, Roche India, to please talk about enabling policies in promoting diagnostic medical devices. Thank you.
Thank you, Dr. Paul. And I must congratulate Dr. Sharma for giving a very good feel of where India is going places. And again, congratulating NIB and Dr. Anvikar and team for completing this milestone. I don't have any slides, which means that you have to look at me. And I'm lying between you and lunch. I'm also a non-technical person. Mostly I work on the general management side. So excuse me if there is a little bit of story and less of scientific content in my talk. So most recently there have been a lot of news articles about population of India, and we have probably surpassed China. We are the most populous. But that obviously brings us to a point where we think more about from a healthcare standpoint also. And I'm really glad and excited to note that in all the sessions that we have had today, there is a lot of focus on that. We have covered about the importance of diagnostics also. It is a no-brainer. We have to look at the preventive part of it, and we have to look at all sorts of disease states and not just the common disease states, which means that we tackle the base, but we at the same time innovate for the next level of diseases, the next pandemics that might come across us. I want to share an interesting story. It's a very recent story. During the time of World Economic Forum, there was a strong India representation, and our Honorable Minister of Health and Family Welfare, as well as our Honorable Minister of Women and Child Development, both decided to pay a visit to our manufacturing facilities in Rotkreuz in Switzerland. And I'm really excited also to say that these posts also went on LinkedIn, and they generated thousands of likes and millions of. So what was behind these visits? I want to give a quick snapshot to it. Some of it was covered just now about instruments and reagents. We showcased our manufacturing facility for instruments. So mostly the high throughput instruments, 8800, which are basically powerhouse PCR instruments. These were shown to them, and the idea was to tell that, what is the complexity and what it takes to manufacture the instruments. Hopefully that kind of, exposure and that kind of experience will help Indian ecosystem also ramp to that level. So these instruments, surprisingly, 18,000 micro assemblies, micro arrays, chips, and whatnot. They are packed into a small size and through almost 6,000 different screws. That's the level of complexity in a private jet, if I can say. Though at a geographically smaller level. And each of these instruments is actually capable of taking care of almost the entire disease screening needs of entire state. So this is the kind of high throughput these instruments have, and the ministers, the honorable ministers, were very excited to see this. Also, we covered reagents. We basically covered the innovation part of reagents, where we are not approaching a stage where we have to detect microliters. We have to detect nanoliters and picoliters of small hormone levels in our bloodstream in order to understand what kind of disease might get prognosticated in our, healthcare reports. So, case in example being NT-proBNP, this I think many of you would know. Delta change in NT-proBNP will reflect heart disease risks in diabetes patients. And then we have tumor markers these days. So no biopsy, we are simply talking about tumor markers because there are small expressions of maybe few hundred copies of molecules of certain chemicals which can be enter into the bloodstream, and these are the molecules we are trying to detect through simple blood tests. So both reagent technology and instrument technology is definitely going places, and not to forget the base. We still have HIV, we have Prime Minister's vision to eliminate TB by 2025. We have HPV, high mortality, high impact on families kind of disease, which is cervical cancer. So all these we have to tackle also. Now, why I gave this background is because I'm coming to a point where maybe there are, Dr. Anvikar, I actually listened to this with a lot of interest, that so many progressive steps that you have taken at NIB under your leadership and the guidance at the Health Ministry. Just a couple of suggestions on what can take us to a further next level. At the industry side, we feel that some of the logistical challenges and lead times can probably be sorted if we have more labs empaneled across India to the same kind of prowess that NIB holds right now. The second is quality and accreditations. This is again a very, very exciting topic. India is ramping up with the quality requirements and standards. The only one thing that we always will need to be cognizant about is that we eventually have to gain global trust, which means that just like we have US FDA, some device has US FDA and we just believe, or some device is approved in Japan, we just believe. I think we need to have the same kind of positioning, and we are going there. This was included in the slide, but I think with a lot more focus, continued focus in the times to come on international acceptance and internationalization of our standards, that would be a great approach. And finally, international tie-ups. For example, some of the foremost research organizations in the world. We need to be shoulder to shoulder with them. We need to tell them, and they need to tell us, and there has to be a constant exchange. And this needs to be also conveyed and percolated so that we have the right positioning of our country. So that's all I have to say. Again, thank you very much, and congratulations for a great program.
Thank you, Vijay. Thank you so much. Can I now invite Dr. Shalini Menon? Shalini, you are Country Medical Director at Sanofi India with huge experience in this field. Over to you, and you're going to tell us about the strategies and challenges in biotherapeutics.
Thank you, sir. And I am the lunch now, not really standing between.
No, don't worry. Dr. Vaike Gupta ne bhi bolna hai.
So thank you. I'll try to make it brief, and because many of the stakeholders may have covered some of the aspects I'm going to be speaking about, which is on biotherapeutic strategies and challenges. Thank you, NIB, for the opportunity. Moving ahead, from an industry perspective, next slide. From an industry perspective, I think biotherapeutics are the fastest growing area, even compared to new chemical entities. Earlier restricted to niche areas and rare diseases, but now we see that it's also being explored in areas which could be of mass importance, and therefore the importance of having simpler regulations which will help us to come up with more of these molecules in the right safety profile to the patients. So if you go to the next slide, just to remember that insulin was the first If you go to the next slide, yeah, insulin was the first biologic which was way back in 1982, but since then there are multiple therapy areas, as you can see, and there are currently 30 monoclonal antibodies, which was in the area of oncology, which was one of the explored areas when it came to biologics. But today it's also gone beyond to osteoporosis, to thrombotic blood disorders and various other areas, marking the need for understanding these molecules and coming out with better diagnostics, as the two colleagues mentioned. If you go again to the next slide. This is just showing you the plethora of new biologics that have come up in the last few years. So I think 30 years for NIB, and in the last 30 years, the biologics also have been exponentially increasing. If you go to the next slide. There are challenges, and I think the stakeholders have already talked about the importance of identification of the right cell line, culture and media conditions, purification procedures, and also post-launch, the batch-to-batch variability, as some of you mentioned, are very key, and manufacturers need to be aware before they get into working with biologicals. Next slide, please. At all stages, there have been innovations which have come in terms of the manufacturers understanding the importance of anticipating the scale-up which is required and not entering into it unless they are able to understand the complexities of the need from a small batch being prepared to the complete scale-up. So that's something the industry is taking good care of to ensure that quality is maintained. Characterization studies are improving, which will help us to do our analytical procedures better. Manufacturing processes, the pharmacopoeial specifications to be followed, as well as the shelf life. So these aspects are being looked into in more detail so that we are able to overcome the challenges in biotechnology manufacturing. If you go to the next slide. Coming with biologics, we cannot but think about the biosimilars. In fact, biosimilars have prompted us to understand biologics better, because as we see with generics, where a bioequivalent study can help with launching a generic, we realize biologics are complex molecules, and therefore if we have to come with a generic of a biologic, which is now called a biosimilar or a follow-on biologic, we have to understand biologics in more detail. So if we go into the next slide, this is just talking about the same thing that I mentioned, that for licensing a biologic which is more complex, a biosimilar would also not follow the same pathway that would be followed by a new chemical entity or generic. Next slide, please. So it started off with EMA coming with its guideline in 2005, followed by various guidelines by WHO, and additionally, in parallel, a lot of countries also came up with their own guidelines, and we all know that the Indian biologics came up in 2012, and later on they revised and updated their guidelines in 2016, which has taken care and helped us to really bring biosimilars into the Indian market. If you go to the next slide. But one key difference between when you have to launch a biologic versus when you launch a biosimilar is the goal of the entire development process. The goal of the process when we develop a biologic is about demonstrating its de novo safety and efficacy. So you start with product quality and you do the animal studies, the clinical and the clinical safety and efficacy. But when it comes to biosimilars, as Rubina ma'am was also going to touch upon, it's a lot different. The goal itself is different. The goal is to establish similarity versus the existing biologic, which means that the processes may be longer in the analytical phase, which is the most important phase, which can detect a difference between these molecules. So while we ensure product quality, the analytical assessment techniques can be improved, and India being at the forefront of biotechnology can really master this and have more techniques to ensure that there is a similarity established at this level. Of course, the next levels are equally important, but they may not be in as much length as would be done for the original biologic, which would be the animal testing, but this is very important to be done in the right species, followed by the clinical testing of the PK/PD and the clinical trials. Having the totality of evidence, as has been said in all the guidelines, is very important and it continues to be followed. However, if you go to the next slide, the analyticals, I would like to re-emphasize, are the most sensitive when it comes to detecting difference between biosimilars and biologics, whether it is detecting the primary structure, the secondary structure of these molecules, or the glycosylation, or the product-related variations, or the process-related variations. So, in fact, there are various techniques and analytical platforms which are coming on an everyday basis to help us with this detection, and to ensure the similarity, there are also multiple ways to approach it so that you can reconfirm it by doing different tests, which gives you a better understanding of the similarity. So this is called an orthogonal technique, where we can use more than one test to identify whether there is a similarity or whether there is any difference between these two molecules. So if you go to the next slide, this is just showing you the plethora of techniques available for, and which are being developed for identifying the critical quality attributes of these molecules at various stages of development. Next slide, please. Coming to the challenges when it comes to biosimilars, I think some of them have been discussed today, that having a single window of approval is something which the industry looks forward to, considering we have to be going to various bodies, whether it is a genetic body or for the clinical trials to the CDSCO, etc. So that's one thing which would speed in the process. Second could be the harmonization of the guidelines with the Indian and across the world, which is already, I think, in discussions and would definitely help us to make it available in other markets faster. And there are newer and newer biologics. It started with a small peptide to now a monoclonal antibody. Therefore, there may be further restrictions on certain aspects when it comes to testing. And having a overarching guideline, yes, it's important not to confuse with multiple guidelines, but maybe having subsections on these kind of areas could help us to ensure that these are also of good safety and quality. If you go to the next slide. This is just showing you that how the biosimilar guidelines, unlike the new chemical entity, had to have multiple revisions to, till today. We see that it started with basic guidelines, but now there are product-specific instructions, there have been amendments, and even the biotechnical proteins, which were probably the first to come in biologics, now have a newer and revised guideline. Next slide, please. The last or second last point is the pharmacovigilance, which is very important. I think biosimilars are well understood by experts, but when it comes to doctors and physicians, I think it's the role of industry, the government, and everyone to educate them more on how to understand biosimilars when they prescribe, so that they are able to track the pharmacovigilance and report adverse events, and so that you can ensure that it's not only up to the pre-marketing phase, but the post-marketing phase, which is also taken care of. Next slide, please. Yeah, so I just would like to summarize that biologics are, rising exponentially, and with the advent of biosimilars, I think it makes it even more important to understand biologics better. The pre and the post-marketing area of biologics are important when it comes to regulatory requirements, and it is important for industry to take note and follow the process. Advancements in regulations are definitely looked forward to, considering the evolving space. And last but not the least, better education of the community at large on biosimilars and their appropriate use would be definitely a requirement in future. Thank you very much.
Thank you, Doctor. Thank you very much, Shalini. Thank you. Thank you, panelists, for very, very insightful presentations. May I request my distinguished colleague, Dr. Y.K. Gupta, for your wisdom and how you see the sector or the subsector growing with great reputation, great robustness, and to be able to realize the best potential that's really possible for India. Over to you, Dr. Gupta.
Thank you. Thank you very much, Professor Paul. I would say that when you talk at 2:15 or past 2 o'clock, generally the feeling is that there is a hunger for good food. But after listening to this discussion, and particularly the clip of Jitendra, I feel there's a more hunger to do more, to be able to do more for nation and to understand what are the gaps so that we can fill it. That hunger is, I think, more now, isn't it? That's why you're sitting here. So, so I won't say that when Dr. Paul will start talking about, anybody will feel any hunger for good food. So now, having said that, I would say that the biologics is the area of future. Entire future therapeutics is going to be resolved towards the biologics and diagnostics and instruments. And we have shown during COVID time our pride. And the success story is the COVID vaccines. I have seen the presentation of Professor Paul under his leadership, how the India has shown not only for our own country, but for Vasudhaiva Kutumbakam, that means we have been providing vaccine for the entire world, meaning thereby they have faith in our quality, they have faith in our efficacy, and they have faith thereby in our approval process through regulatory system. When we say regulatory system, I'm just putting it here. When you say regulation or regulatory system, the flexibility automatically goes off. When you say this is the regulation, this is a law, there is hardly any flexibility. It's a contradictory, it's a paradoxical phenomenon. But when the situation arises, when the situation demands that now you have to think differently with some caveats in it, with some pharmacovigilance, with some post-marketing surveillance, with certain restrictions, then the flexibility can be adopted. But remember this, the flexibility is never ever a permanent feature. The flexibility should be as early as possible, should be regularized by appropriate steps which is risk-free, or which you can say which is acceptable risk. And that is why when you talk of a complex situation like biologics, involving, which is a complex nature, is not a small molecule, is a larger molecule which has a complexity in quality, complexity in stability, complexity in each and every step for variability, and the infrastructure which requires for the optimum utilization of the scientific and infrastructure pool which we have. And all of these speakers have identified this, that we need to have perhaps a larger number of facilities which are empaneled, which are capable, and which are trustworthy, and which can be relied upon by the international agencies as well. What is the important thing required? I think everybody has said that the success story, but the challenges are equally important. And that is, should we have a regulatory scientific body who can constantly, continuously look for what could be the alternative scientific method which is equally valid for this? You cannot have a regulation which is 20 years old, even 10 years old, even 5 years old. Even today the regulation need to be changed tomorrow because the evidence which you generate today. And there is no need to have, say, if you want to do a trial in 1 or 10,000 patients, now there is a possibility that with the genetic understanding, with the situation which demands urgency, you can do it a much smaller number, provided you have a caveat there. Why there is a need to have multiple X number, three sets or maybe 10 sets of genetic manipulation, and then you do this? Can this be done in a much faster manner? If this has proved as an effective method in EUA, emergency use authorization system, you evaluate it. Can it not be adopted as a regular feature? If this can be done in 1 year, 3 months, or 15 days approval process, can it not be done in a regular manner? Why you call it as an emergency use? I think this requires scientific inputs, and this scientific input can happen through this type of forum where person like Neeti, Professor Paul, and all the industry and the academicians are present here. Hardly we do any scientific discussion. Why this? I was talking of sometimes when we revised the schedule, why rat and mouse sometimes might be in some time because of the typographical error, rat/mouse, and then it becomes rat and mouse, and then since it continues to be that. So this is, I think we have to be very critical when we put these things into our process. Because why I am saying? Because everything has a challenge. Biologics, biosimilars, monoclonal, gene therapy, or maybe you can think of any of these things. This is the process. 4 years. I would say Jitendra has a 4 years, and Professor Paul has said कि जब 4 साल में ये कर दिया तो 30 साल में क्या होगा? The same way we have to look for all these biologics which are developing regulations, and we cannot wait for 30 years to develop regulations. We cannot wait for 30 years and then we say कि हमें international collaboration की ज़रूरत है, तब जाके हमारे regulation बनेगा. अरे भाई क्यों? हम regulation अपना बनाएंगे, हम उसको validate करेंगे, फिर international वाले लोग हमसे सलाह लें कि आपके इतने लोगों में ये हो गया है, तो आपने क्या किया? And this is exactly, I was just telling you, when we develop the compensation policy, I chaired that, and we developed compensation policy for the first time in the world, in India, in clinical trial. Harvard and Oxford, the both university came, उन्होंने कहा कि नहीं ये तो नहीं हो सकता, इसमें हम कैसे compensation दे सकते हैं, हमारा TOTS law होता है. And now we are the only country we give compensation, and by mistake at that time I wrote that this is a provisionally final formula of compensation. एक बहुत बड़े इंग्लिश के ग्रामेटिक ने चैलेंज किया, ये कौन सी अंग्रेजी है, provisionally final. तो हमने उसको डिफेंड भी कर दिया बाद में कि प्रोविजनली फाइनल इसलिए है कि तब तक प्रोविजनल है जब तक कोई इसमें बेटर कमेंट नहीं कर दे, अभी तक नहीं किसी का. So till that it is final. So what I am trying to say, I think this type of forum must be used for something innovation in biologics regulation, and our regulator has shown flexibility, resilience, or adaptability, or fast. This should become the normal pace. This should be the normal pace. घोड़ा दौड़ता उसी स्पीड से है, जबकि उस चाबुक की जरूरत ही नहीं होनी चाहिए ना, जो वो दौड़ना चाहिए. तो आई थिंक we are at a very good stage. I am very happy. मुझे शुरू में डर लगा जब रुबीना ने अपनी स्लाइड दिखाई और हमारे डीजी साहब बैठे थे, DGHS, उन्होंने कहा जल्दी करो टाइम हो रहा है, लेकिन इतने सारे पैरामीटर्स थे उनके रेगुलेशंस के, माय गॉड, वो उनको एक जगह खुद ही कंफ्यूजन हुआ कि ये क्यों है. तो हमको जरूरत है कि उस रेगुलेशन में हम देखें कि भाई ये क्यों है, और है तो क्या इसका कोई दूसरा अल्टरनेटिव है, अगर नहीं जरूरत है तो क्यों, और अगर जरूरत है तो उस पर फ्लेक्सिबिलिटी क्यों. फ्लेक्सिबिलिटी नहीं हो सकती. अगर जरूरत है तो नो फ्लेक्सिबिलिटी, अगर जरूरत नहीं है तो फ्लेक्सिबिलिटी नहीं, फिर करो ही मत. So flexibility, I think we have to be slightly flexible in saying that there is a need of flexibility. Thank you very much, sir.
Thank you. Thank you, Dr. Gupta. Thank you, panelists, once again for very, very thoughtful, thought-provoking intervention that you have made. I would very briefly say only 4 points, and my first point relates to congratulating the National Institute of Biologicals for their stellar history, for being a great institution, I like to see the hands of NIB family so that we can congratulate you. Thank you. Thank you so much. Keep the banner flying. Your work is just starting. You are ahead of your times. अब जो revolution आ रही है इसमें अपने आप को position करिए और पूरा role अदा करिए, scale में भी, quality में भी और stature में भी. Thank you so much. I think it is evident from the presentation, and I'm sure presentations additionally from since morning, that it is India's destiny to be the leading nation in the space of biologicals. Ultimately, we will be leading the world. There's no doubt in my mind. Having seen the experience from COVID, and I'll come to that very briefly, though it has been spoken. So it's only a matter of time. Biotherapeutics, diagnostics, we shall be the hub. Only issue is how soon can we put it all together. And I like you to think how we can speed it up. And why we are all referring to COVID story is because the way we rose to the occasion. Vaccine is one part, but I think we didn't talk about diagnostics, or we talked indirectly. What I know is there were ultimately 280, let's say 300 diagnostics approved, RT-PCR and the other one, by ICMR, and 78% of them were India made. And within the first 100 days, roughly 100 diagnostics came up, Indian made diagnostics came up. Can you imagine? From nothing, you have that capacity. And Dr. Y.K. Gupta is right, वो रोज क्यों नहीं दिखाई दे रही हमें? वो HPV diagnostics में क्यों नहीं आ रही उतनी speed से? So we have allowed our potential to be capped. तो एक तो ये है कि it is our destiny. And please, NIB, you have a great role to play and to lead, and the industry has a great role to play, and we all have a role to facilitate it. And I believe Biopharma Mission, BIRAC, they're doing their job. What more can be done, please tell us. And I really believe it's only a matter of time. Having said that, in this particular session, I did not hear specific issues around the regulatory processes that you want to change or to modify. I leave that to discussions in other sessions. But if the changes are required, and I believe you had a discourse on this, please bring it to the fore. We are in the stage of revising the Drugs and Cosmetics Act. अगर उसमें कोई कमी है, wording में ही and लग गया है, slash लगना चाहिए था, इसके अलावा कोई missing चीज़ें हैं, उसको बताइए, आप देर मत करिए. Bring out a multi-stakeholder position paper and use this forum of today, where it's a churning, it's a melting pot, to give the reasons for it. CDSCO system, RCGM system, and wherever you think that change is required, please talk about it. If that is holding us back, that's not acceptable. And you know that the government, and particularly in the last few years, you have seen so many decisions that have been taken to make ease of business as a way of life, ease of life being a way of life as a policy of the government. And we have been talking about reducing the compliance burden and so on. So in the same light, direction would be taken in giving a regulatory system which is facilitatory and not obstructive at all. My second point relates to elaborating on the diagnostics, and I have a wish list here. I want us to progress on both the frugal as well as disruptive diagnostics. We have challenges on both. Frugal, affordable diagnostics, which we need on a daily basis, which should get less costly, become point of care, and particularly for our primary healthcare paradigm, where the hub now is a health and wellness center, where I'll get my tests done. That is a frontline area where I want help from you all. Because primary prevention as a part of primary healthcare is one part, but then there will be diseases and there will be new diseases. Secondary prevention means the earliest possible detection of the disease. That DX has to be earliest. And here, even in the current wish list of the diseases that you want to screen, we don't have optimum point of care, workable diagnostics. Let me say where we have it for malaria, We have it for pregnancy for long. We have for hemoglobin, but I'm not sure whether it's really optimum. Digital hemoglobinometer uptake has been low. I don't know whether we need a choice or not. But can we have, for sure, for something for anemia? Something which tells me anemia, not only anemia, but whether it's iron deficiency or not, because then the algorithm immediately changes. Because from nutrition to non-nutrition is a very important paradigm, and we are struggling with looking after nutrition by fortification, by prophylaxis, and by treatment. And in the bargain, we might be delaying other conditions and maybe interfering with them. So not only hemoglobin, if you are happy with current hemoglobinometers, no problem, but can we have something on ferritin so that this is sorted out? My second wish list is to have something for earliest marker of chronic kidney disease. What I know is creatinine on a chip. Is that possible? Or is there a better marker? I don't know. But can creatinine be something that can be done by in the home setting or the health and wellness center? Because that's only way we have blood sugar, we have hypertension screening by a different method. But the third disease where the deaths are 225,000 per annum, diabetes deaths are 255,000 per annum, not too far, and yet we don't have a screening tool for chronic kidney disease. And you look at the misery it causes. It devastates families. And likewise, liver disease. What is the best marker of chronic liver disease? I believe it must be one of those enzymes. I don't know which one, let's say SGPT. We don't have it on the chip. Why are we waiting other people to do it? Find or somebody else to do it, FIND, whoever they are. Please do it now. It cannot be difficult. If an unknown virus diagnosis can come in 30 days, 23 days, why will it not happen for these chemicals, these biologics that we understand for so long? So that's my third wish list. So hemoglobin, ferritin, the creatinine or equivalent, whatever you think is the best marker of chronic kidney disease, something for liver disease, possibly an enzyme, and then followed by HPV. I think we have some candidates, but I'm not sure they're optimum. So can you be in the game, be competitive in this regard? HPV for cancer cervix, we no more need pap smears, and of course we do not need to go in the direction of acetic acid related things. It's waiting to be done. There'll be one arm of HPV vaccination, which is hopefully will happen soon. But there is a whole cohort of women who have to be tested at a particular age. And pap smears don't work in primary health care. Acetic acid is very crude, doesn't work. And we are, blocked by not having a point of care diagnostic in order to invoke this screening procedure in a systematic way to create the loop for treatment and follow-up and so on. So HPV, DNA. Sickle cell disease is another one, going to be an initiative soon. We're trying to work out the details. We have a tool, point of care, but from other countries. Somebody else is doing it. Can we have something even more facilitatory, quicker, and so on? So ये 3-4 चीजें मैंने कही. Of course, there is a longest list. I overlooked hemoglobin A1C. So in my list, creatinine, SGPT, hemoglobin, ferritin, and HbA1c for diabetes control, because blood sugar based control is not going to be a good way of looking at the control. HPV DNA, sickle cell disease, and then you can add dengue and other things. So can we have point of care diagnostics in these areas? Please, can we work in mission mode? How can we do it? Please tell us. At the same time, the second track has to be disruptive, where our diagnostics are used in Germany and France and Canada. So both the tracks are important. And the cutting edge list that you saw from the first presentation, the AI algorithms and everything else that you showed, we must work in that, because then only we'll capture that market. But at the same time, we need to make these low cost, affordable point of care diagnostics for India and for much of the world. There again, we'll go long way. My last point is really to understand, and it's a very short point, it's more like a question. For biotherapeutics, do we have enough base or we don't have enough base? We seem to have technology or capabilities, but is it enough for us to go whole hog or not? What is it that we can do? Does our PLI scheme help you in that regard? What more can be done? Because biotherapy, diagnostics, I understand, can happen, I can see, but complex biotherapeutics, I don't know whether we have enough capacity in the country and how we can do it. And in this regard, I like to say, last but not the least, that Roche and Sanofi, please come and make in India, co-develop with us. We want this to be produced here on the soil of India, like we did those, billions of doses of vaccine of every platform on India. Make it on India soil. That will be my request. And last, जो भी हमने कोविड में देखा वो क्या देखा? You cannot become heroes from zero. One of my panelists made reference to this. We had the base, then only we could rise. You had MTEC, then only you could have, over 1 million tests possible, and so on. So the basic base of academia, national laboratories, regulatory ecosystem, industry ecosystem has to be there and must continuously improve and be ready with time, and tell us which policies will enable that. To become hero, we cannot rise from zero. That doesn't happen. With these words, my deep gratitude for the opportunity and my sincerest apologies for the delay that I caused in access to your lunch. But I really enjoyed being here, and thanks, and a big hand to the panelists and my co-chair.
Just few more moments before we leave for lunch. I would now like to request Professor Paul to kindly felicitate the speakers one by one. I'll take their name. We can start with our only lady speaker on the dais, Dr. Shalini Menon. We have Shri Jatin Mahajan. I'll go by that. We'll have Shri Rajendra Kumar Suri, CEO, DCVMA. Dr. Jitendra Sharma, CEO, AMTZ. And Shri Vijay Kumar, Head Portfolio Solutions and External Affairs, Roche India Private Limited. May I request Dr. Harish Chandra to come up here. Dr. Harish is our Deputy Director, Quality Control. Sir, if you have come, I may request you to kindly come on stage and present a small token to Professor Paul, Professor Y.K. Gupta. Professor Gupta. Thank you all. Thank you for this excellent session, guys. And now I guess we are already running ahead of time. So we'll see all of you at sharp 3:30. We'll gather here again at sharp 3:30 for our academia session. Thank you. Good afternoon, everyone. Welcome back for our last session of the day. Like I'm telling you all these while that we have 3 sessions, 2 are over, and now is the turn of academicians. The session is Step Towards Atmanirbharta, Connecting Industry with Academia. And I would now request our chair of the session, Ms. S. Aparna, Secretary, Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India. Ma'am is Secretary to the Government of India in the Department of Pharmaceuticals since 1st October 2020. A postgraduate in zoology, MPhil in Energy and Environment from India, an MBA from Melbourne, Australia. Ms. Aparna is a member of Indian Administrative Service for past 3 decades with a huge experience in the national, state and local governments. I would like to invite her on the dais, ma'am. Yes, ma'am. To start the session with her, I would like to invite Dr. Pramod Kumar Garg, Executive Director, THSTI, to chair the session. And I would request all the distinguished speakers for the session to kindly come up on the stage. We have Dr. Amulya Panda, Associate Director, Panacea Biotech Limited. We have Dr. Alka Sharma, Senior Advisor, DBT and MD, BIRAC. We have Professor Anurag Singh Rathore, IIT Delhi se. And we have Dr. Harish Chandra, Deputy Director, Quality Control, NIB, talking in the end. And with this, I would hand over session to Madam Secretary.
So why don't you speak earlier in the session, so that you can tell us how we should go.
Over to you, ma'am.
Thank you.
Thank you.
What I'm going to do is, first of all, welcome all our panelists. We have a very varied panel here with experts from the clinical side, the research side, in the public sector, in the private sector. We have a range of disciplines who are represented here. I was inclined to accept and join this event because of the emphasis on quality, which I believe is going to be the make or break deal for the Indian pharmaceutical industry, which I use as a broader terminology to cover all the medical countermeasures based on clinical research. So covering the range of products like therapeutics, diagnostics, preventive products like vaccines. I'm just using pharmaceutical as a broad category. And I do believe that while India has justly earned the name of being the pharmacy of the world, and we do take great pride that we contribute about 20 to 22% of the generic drugs produced and consumed globally, there is now a stage has been reached wherein we need to differentiate ourselves on the basis of quality. And this, I believe, is something which is an imperative. It is a precursor of the trends that will be seen over the next 2 decades or so. And there is, I think, a growing understanding that we need to forge a new path now for the entire sector. So it is, of course, our privilege that we have experts on the panel. We have been given a tight timeline, so I will not speak at length. I will try to sum up at the end. And since Professor Garg has to leave for another meeting by 4:30, you said? So I think we can start with hearing his remarks and then move to the panel. Is that okay with everybody? So we can go. I'm happy that the room is now filling up. a very warm welcome to the delegates. I hope you found about 15-20 minutes to have a very quick lunch, but it's all your fault. You must have asked lots of questions to the last panels. So over to you, Professor Garg.
Thank you, Madam. I'd like to thank the organizers for having invited me here for this very important session on connecting industry with academia towards Atmanirbhar Bharat. Now, I mean, for time immemorial, we have been talking about connection between academia and industry, and both as a clinician and researcher, I have seen the gap for far too long in this country. But as COVID has taught us many things, it has also taught us how to connect the two. And in fact, during this last 2 years, we have seen successful tie-up between academia and industry at multiple levels. And vaccine, for example, was the best outcome of that collaborative research. Currently, I'm at HSTI, where also we are in close partnership with industry developing vaccines and also coming out with newer therapeutic molecules against various diseases, including tuberculosis, dengue, and other infectious diseases. So there is no doubt that today the environment is ripe for very fruitful collaboration between academia and industry, and there are government agencies which support this kind of collaboration. For example, BIRAC, Dr. Alka Sharma, MD, BIRAC is here with us. So whether you are in academic centers or you are in industry or regulatory space or other institutions, I think today is the time when all of us have to make more and more efforts to connect the two and come out with very useful products against various diseases. So with these initial remarks, we can invite the first speaker, Dr. Amulya Panda, who is going to speak on translating innovation in biotechnology. Dr. Panda has been earlier Director, National Institute of Immunology in Delhi, another DBT institution, and currently he's Associate Director, Panacea Biotech. In fact, we have at HST ongoing collaboration with Panacea. So Dr. Panda is going to talk about this connection in a more, Dr. Panda, please.
Thank you. At the outset, I would like to congratulate NIB for the 31st annual day, and thanks to Dr. Anup for giving me this opportunity. As Dr. Garg told, I am predominantly an academician. I worked in National Institute of Immunology for 32 years, and for the last 2 years, I am at Panacea Biotech. So I believe I know both sides of the coin a little bit. And what I will tell you is that from the morning we are just listening to the industry. So this time, थोड़ा पढ़ाई लिखाई भी हो जाए. That is important because हम लोग वही बोलते हैं. And I think please look very carefully about this title, Translating Innovation and Biotechnology. If you see every day, even in newspaper talk about translation, newspaper talk about innovation, newspaper talk about biotechnology. I'm sure everybody, you know about the term. I have my own definition. I hope you don't mind. This is a very preliminary definition. But all I want to know that let's understand what is biotechnology, what is translation, what is innovation. Then only we can talk. Next slide, please. Okay, first, as I told you, I'll tell term and terminology. And again, I apologize to all of you. I know all of you know these terms. No big deal. What is innovation, what is biotechnology? But there is my way of thinking. I think I'll put my words. And second, I'll talk about translation. I mean, during BIRAC, I think Dr. Alka is here. I have that chance, as well as I was very lucky to get associated with Dr. Bhan. I learned something from him. What is translation? Basically, all you know, Dr. Bhan is the person who initiated translation. I'll tell you certain rules of translation. If you follow, I can give you in writing, you will succeed 100%. Third, I'll tell about synergy between academia and industry. I think Dr. Garg, this is what we have been talking for the last 30 years. When we were students, we were also talking. You go to academics, they say, industry wale chor hai, ye to paisa kamate, kuch karte nahi. You come to industry, they say, what you are teaching, you are only publishing paper, impact factor, only for your promotion, it has no impact. Itna bura nahi hai. I think we need to understand. I'll show you some example. It is time has come, we must understand our own strength and utilize it for the country's development. हो सकता है कि there are shortcomings. So I'll talk about that, about academics and industry collaboration, because that is important. So with this, let me go to the terms and terminology. I again apologize. These are very preliminary, fundamental school level terms. Next slide, please. This is the proper definition of biotechnology, and if you see, this was defined by the International Biotechnology Symposium in 1984 in IIT Delhi. This is technological exploitation of living organism, biological. It has biology and it has technology. And when you say technology, it is tech and logy. It is engineering and science. Without that, no technology. People keep telling this is biotech, this is bio, this is B. No, this is complete biotechnology. Let me tell you how I think about biotechnology. I'm sure everybody must be telling to your kids, study biotechnology, medical name mila, biotechnology pad lo. I have a different way of. Next slide, please. If you see, these are the 4 things we operate in biotechnology. When people ask, how do you define biotechnology? I say biotechnology, I'm an engineer by profession. Anurag will agree. For me, biotechnology is like mathematics. Mathematics is very, very simple. I tell you, what you do in mathematics, you do only 4 things. Addition, multiplication, division, subtraction. That's all. Very simple. You look at these 4 molecules. We only manipulate these 4 things. I'm sure someday some biologists will tell nucleic acid replicate, carbohydrate degrades, something divide. So biotechnology is all about manipulating these 4 things. It's nothing, simply mathematics. The very large scale integration somebody does to send a supersonic jet, or how do you integrate things, or high speed computation, these are based on simple principle of addition, multiplication, subtraction, division. If your fundamentals are clear, that's clear. This is biotech. Next one, innovation. I think we all studied innovation. बचपन में पढ़ाया गया. Columbus discovered America, Galileo invented, Edison innovated electricity. Please remember, in research lab like THSTI or NIE, we put money to create knowledge. That is invention. When knowledge creates money, that is innovation. So you can say innovation is reverse of invention. Never get confused about this. In all public sector institutes, we put money because our aim is to generate knowledge. If that knowledge can be used to generate money, that is innovation. And कोई जरूरी नहीं है कि सारे चीज innovation भी हो. No need, not that everybody has to be Edison or Louis Pasteur. We need Niels Bohr, we need Chadwick, we need fundamental science also. So this is innovation. Next slide. I can give a list of innovation, but I'll ask you, nowadays people talk about disruptive innovation also. You must have heard it. Very disruptive innovation. The number 10 is disruptive innovation. I ask any one of you, can you purify protein without using chromatography? Can anybody think? Come to my lab, I will show you how to purify protein without chromatography. That is called disruptive innovation. So please be sure, what is innovation? What is disruptive innovation? See, if you go to Punjab, I have an industry, we have a setup in Landu. If you travel, you see in Punjab, they make lassi using a washing machine. That is not innovation. That is simply logical extension of centrifugal principle. That is jugaad. No innovation. Innovation needs serious contemplation. You need to understand, innovate so that you create money. Next one. This is translation. I'm sure this is a stupid way of explaining translation. Nucleic acid to protein, right? Everybody know this? But please think about this term. Nucleic acid go to protein, that is translation. What is happening? Nucleic acid, protein is amino acid, so there is a chemical change, right? Nucleic acid, I'm not saying predominantly has no conformation, there is conformation, but we know in the morning we are listening, protein works on conformation, right? Without conformation, protein will not work. So when nucleic acid go to protein, there is a chemical change and there is a physical change, right? We have taught in the school, chemical change are permanent change, physical change are visible change. So if your discovery bring permanent visible change in society, that is translation. Somebody develop a clone, somebody develop a sequence, these are only leads, these are not translation, these are not technology. That's why we fail. I'll show you how translation can go. Next slide. look, morning you are saying, look, these are the 10 commandments of making vaccine. Look, from proof of concept to expression, process development, facility, manufacturing, supply chain, assay, clinical, regulatory. In academics, except one or two, we don't understand this. No academics understand last 8 points. How do you expect translation? This is what is essential. I think, as I said, I was in NI. Somebody asked me what you have contributed. I calculated, we have produced 500 wonderful PhD. All our PhD students are placed in Merck, Pfizer, all the places. Then I realized out of these 500, only 50 are in academics. 450 are in industry. If that is the way our PhD or our education system is producing for industry, why not teach them the rule of industry? We don't teach them. I don't know how many education them GMP, GLP, QA, QC. They don't. I think we need to learn that. Next one. This is what I picked up from Dr. Bhan. If you want to translate anything, please follow these 5 rules. This is like Lipinski's 5. Is it useful? Is it science reproducible? Has the proof of principle has been made in all appropriate model? Is it scalable? Is it competitive or sustainable? I can go on talking on these 5 points all together. I'll not tell anything. I'll just tell about the scalability. I think you must have read in the school how to make oxygen. Take potassium chloride, add manganese dioxide, heat it, you get oxygen. Have you ever seen such plant operating anywhere in the world? No way. Today morning, I think in the medtech we saw oxygen is produced by purifying or liquefying the gas. So we must change the way we are taught to the student. We must change what is the role of the scale. Then only you can translate. But I tell you, anything you want to do, anything you will discover, ask these 5 questions. If you 5 questions are yes, you can go to stage number 2, like TRL 4 or 5. But this is important because it has to be useful, it has to be reproducible. Like we do experiment in academics. I have an antigen, I immunize 4 different way to a mice. I'll get 4 different results. 1 time tolerance, 1 time immunity, 1 time energy. I'll publish 3 papers. But will it produce anything? No. I need reproducible things. So if you follow this, probably you'll do the translation. Next one. Now I'll talk about academia and industry interaction a little bit. We all have been talking that academia is different, industry is different, but look very carefully, there is no difference between academia and industry. Next slide. Look at the education, our system. We memorize, we understand, we analyze, we apply, evaluate, create. It's a different story that we are in the first step. Our education system only we write, mug and reproduce. We don't do that. But look at the manufacturing, same way. We lead, we express, we analyze, we supply, quality and product. In education system, the product is a live human being, like I am a product of IIT. Same way, a product is a biological thing. If you think very carefully, there is not much difference between the academia and industry. No, the whole aim is to produce something, a quality human being or a quality product for making that human being. I think we need to understand in this aspect, then only both can talk each other and discuss. Otherwise, accusing somebody something will not give you a solution. The other point I want to tell you that in spite of all these things, we have a wonderful education system, let me tell you. You may say we are not, our numbers are less, no 500 universities are there, we are below 500 of different evaluation, but we are pretty good. Let me tell you 2 points about the beauty of our education system and our manufacturing system. I give an example. Take a PhD student who do PhD with me, 5 lakh per annum, maybe 5 years, 25 lakh we spend. Government of India spend 25 lakhs. If the same student go to US university, he spend 2.5 crore, $50,000-$60,000 per year tuition fee. So the cost of a PhD student in USA is 10 times higher than the PhD of NII or IIT. But I tell you, of my personal experience, the quality of both the students are same. Next slide. India is a developing country, but it is a developed country as far as intellectual infrastructure is concerned. We produce the highest intellectual capital per dollar here. This is not I am saying, this is John Welch, the GE chief is saying. That means with the same amount of money, we produce maximum intellectual capital. So to all industry friends, academic is not that bad in India. We have brains, we have people. I can tell you my students, because I was in NI, it is one of the elite institute. All my students are everywhere. They are in GSK, they are in Novartis, they are in J&J, they are in Merck & Sharp, everywhere. No difference. That means we have the capability to produce world-class students. So let's forget that ki हमारे यहाँ ये नहीं है, वो नहीं है, वो नहीं है. We have capability. There may be cluster around IIT and other place, but we did have capability. Talk about Indian industry. Next one. You see, from the morning we are talking, during the pandemic, we produced 3 billion doses of vaccine here. Forget about who has the technology. We made supply chain. We have all platform developed in this country, including help of BIRAC, DBT. We can make wholesale vaccine. We can make VLP-based vaccine. We can make recombinant vaccine. We have nasal vaccines. We have plasmid vaccines. We have our own mRNA vaccines. Everything we could do. This was possible because we have a synergy between industry, academia, and regulatory that we have been talking from the morning. And in fact, I didn't know that Dr. Garg will be there. Panacea and THSI is in a collaboration, a strong collaboration of developing vaccines for future. So all I want to say that when we talk about academia, industry, let's not find fault who is what, who is not. This COVID has a great teacher. It has taught us we can deliver. That's all. Next slide. All I want to say, we have wings. All we need is to have the intent to fly, and we should fly. Thank you very much.
Yeah, thank you very much, Dr. Amulya Panda, for a very insightful talk. I think we'll have time for discussion later, and audience can also ask questions later. Now I invite Dr. Alka Sharma, who's a Senior Advisor, DBT and MD BIRAC. She's going to talk about boosting biotechnology, building India as an innovation nation. And she's someone who's actually funding the academia-industry partnership to develop products. Dr. Alka Sharma, please.
Thank you, Dr. Garg. Namaskar. Good afternoon, everyone. Respected chairs and dignitaries on the dais, speakers, and also all the participants. It's my pleasure to be part of this event, and also I'm going to talk about boosting biotechnology and building India as innovation nation. So I don't have to define biotechnology the way Dr. Amul Panda has already given such a nice, clear understanding of biotechnology here. So somebody is operating my slides? Okay. So this actually, many of you may be aware that during past few years, now global biotech industry has grown by 5%, more than 5%, and India contributes 3% of its market share to global biotech industry. And India is ranked 12th in the world and 3rd in Asia Pacific in biotech sector. And we are the largest vaccine manufacturer and second producer of BT cotton globally. In order to strengthen and empower the public-private partnership and also to spur innovation, Department of Biotechnology established Biotechnology Industry Research Assistance Council 10 years back. And here I would like to remember Dr. M.K. Bhan. This was the vision of Professor M.K. Bhan to set up BIRAC as an entity. This is first of its kind, unique organization in the country to promote public-private partnership in biotech sector. And you can see on your right side that the number of biotech companies, startups, products reached in the market, bioincubators, and teaching and research institutes in biotech sector across the country. So currently that bioeconomy has crossed $80 billion in December 2021. 2022 data is being analyzed. So the target is to achieve $150 billion bioeconomy by 2025. Here I would like to emphasize that the efforts being made by the government on laying strategy creating ecosystem to facilitate end-to-end processes, and also creating critical mass of desired skills and addressing regulatory issues from time to time. Government is playing a very important role in all these and creating a vibrant ecosystem across the country. Next slide, please. So this actually I would like to just share very briefly. So keeping global and national scenario. So in Department of Biotechnology formulated National Biotechnology Development Strategy in 2007, and after that it is being revised from time to time in consultation with all stakeholders, and the focus is on all components of biotech sector which are essential for driving inclusive growth for industry, bioeconomy. And you can see in the last column, nurturing innovation and entrepreneurship is one of the key components of this strategy. Next, please. So before I go forward, I would like to just highlight the key factors which are very, very important for successful technology innovation. And basically, there are technology pull and push factors for technology innovation. This is very important to understand when we all say innovation. So it should actually be clearly understood what exactly innovation and how to go about in this. So that there are these two factors, pull and push. So technology push is a simple linear sequential process, and this starts with basic research leading to new invention, creation of intellectual properties, and also development of technology. So here you can see the emphasis is on research and development, and we require people with good imagination power and also an ecosystem to promote and support invention. Whereas in technology pull, the emphasis is on commercialization and marketing. So here the market is the source of ideas for directing research and development. And what you require for that, right kind of policy framework, adequate resources, and also proper networking for commercialization of the product both at national and international levels. So this is the basis of all kind of product and technology development, and we have to be very clear right from the beginning in what manner we have, since this entire pipeline that I'm going to discuss very briefly because I will just keep time in mind. So next please. So taking this actually that how to connect it pull and pull mechanism in this, so how to have a balance in these two, because we can't only have the push mechanism or only pull, because it has to be in a synergy, there should be a synergy, so there should be a balance. So in order to just to strengthen and empower emerging biotech enterprise to undertake strategic research and innovation addressing nationally relevant product development needs, so Department of Biotechnology has established in 2012 Biotechnology Industry Research Association Council. As I said, it's a BIREC, and the mandate is to nurture innovation across entire product development chain. And that starts from ideation, that is idea, just to have an idea, and then go for proof of concept, early and late translation research, validation, testing, and up to commercialization. So various schemes are available with BIREC for each kind of support that anybody wants, the innovators, startups, entrepreneurs. And under Entrepreneurship Development Initiative, that 75 bioincubators have been established across the country to give the opportunities to the innovators, startups to test their ideas, to run their operations, and it's a landing space for the newcomers who want to just have some kind of high-end equipment, infrastructure just to use that. So just to provide a landing space to all the innovators, startups, bioincubators have been supported across the country. So in fact, you can just see that how these activities has contributed significantly towards development of biotech innovation ecosystem across the country. So as of now, if you really see, more than 3,700 startups, biotech startups are being supported by BIREC. Next. So this is very important, just slide to have an impact of this. So as you all know, the biotech sector, which mainly includes healthcare, industrial biotech, agriculture, waste management, and clean energy, so that requires heavy investment, long gestation period with high risk and high reward also. So the contribution from biotech innovation ecosystem to the bioeconomy of the country, that you can just see in terms of number of startups, bioincubators, number of products, job created in that specific domain, specific areas. So that has actually now in last 10 years, if you can just see the number, and next please, and also the target which we are now looking forward to have in coming years, that is up to 2025. So this is the impact that we are now having. That system which was created by the government 10 years back, we started, and now we are seeing fruits and bioeconomic contribution by that whole entire ecosystem, and we are hoping that we are going to get that target very soon, and we are hoping that before 2025 also in some of the cases. Next please. Yes, this is very important because just everybody should know that yes, this slide shows that biopharma still accounts the largest share of bioeconomy that you can see here. And at the same time, I would like to share with all of you that there is a huge jump in the index, Global Innovation Index 2022, and the India was in 81 in 2015, now we are at 40. So in 2022. So this is actually showing that whatever government is doing in terms of strategy, in terms of any other activities, and what are kind of policies, guidelines, is contributing towards that bioeconomy, and its a value addition is of course is there. That's why we have now a huge jump from 81 to 40. Next please. So this is the funding opportunity. I'm going to keep this very, very brief because the details are available on the website. So BIOREC actually is continuously supporting, promoting this biotech startup ecosystem through its various schemes and programs, and including funds of funds and biotech angel networks. And this is providing, as I earlier said, that end-to-end processes. At any stage, there is a scheme for any kind of startup in biotech sector in a broad area that I mentioned: healthcare, agriculture, waste management, clean energy, and all. So this is the actually this, you can just see the ladders, and these are the names of the schemes, and details are provided on the website. Next please. Yeah, so I would like to just say that a number of reforms have been now taken place as far as regulatory reforms are concerned, and many of you are maybe aware, and today morning sessions there were some talks also from the many speakers that new drugs and clinical trials rule notified in 2019, national medical device rules rolled out in 2017 and now operational from January 2018, national medical device policies, so it is now driven by Department of Pharmaceutical, and then we all have now part of this. We have already provided comments on the approach paper. And this is something that DBT has done very recently under the guidance of Dr. Gokhale. He is now Secretary, Department of Biotechnology, so he has taken this challenge, and now this is in place, the gene editing guidelines and standard operating procedures for regulating reviews of genome editing plants under SDN1 and SDN2. Earlier for this also RCGM approval was required. Now this can be done at the level of institutional biosafety committee unless it's having this exogenous DNA. So basically this is very simple now for the researchers who all are doing now agriculture-based research. And first half I am going to cover in my next slide. This is something that you all must know, that what government is facilitating and promoting. Next slide, please. So this is something that this is a platform, and though this platform is now maintained by BIOREC, but we have representatives from all ministries and departments concerned, that is DBT, BIOREC, DST, NIB, CDCSO, GEM. So actually weekly, earlier it was weekly during COVID, but not fortnightly. They all sit together and they address queries of innovators, startups. It may be through messages, through phone, through online, emails. So they are free to send any query at any for anything related to technology development, product development, getting regulatory approvals and all. They get the clear answer from the person concerned who all are dealing with the subject matter. Next, please. Similarly, National Biopharma Mission is a collaborative mission between Department of Biotechnology and World Bank, and this is the industry academia collaboration mission, and the focus is on biosimilars, medical devices, and also vaccine development. So overall aim of this mission is to make India a hub for design and development of novel, affordable, and effective biopharmaceutical products and solutions. So this is now ongoing mission, and lot of now efforts have been made so far, and there are leads available in many of the projects under this mission. So I will just take you through this, some few examples where this is now, has been shown that an integrated innovative approach was adopted by all the stakeholders to just combat this COVID-19 pandemic, and that these are the examples. There are many more, but I would like just briefly, I'll just go through that, all these examples where all came together and worked. So that since this session is on steps for Atmanirbhar Bharat, so these all schemes, programs, activities are related to Atmanirbhar Bharat only. So I just wanted to just share a few slides. So this is a multi-pronged research strategy was formulated by Department of Biotechnology with all its 16 autonomous institutions and also PSU BioRec. And this for building and leveraging ecosystem for development and manufacturing of vaccine, the support which we actually provided to few of the industries by BioRec and many infrastructures by support from DBT. So for example, that this testing I'm going to cover in one separate slide, so I'm not going to cover here. So the establishing biorepository, COVID-19 biorepository, and also INSAGOG network, where this whole this government agencies and department concerned are the partners in this. Developing immunoassay labs, animal challenge facility, Professor Garg is here. So this was done at THSTI with other partners from other autonomous institutions, antiviral screening, and also vaccine testing facility. That two autonomous institutions, and one is in Pune, NCCS, because Serum Institute was there for manufacturing of vaccine, and similarly Bharat Biotech was in Hyderabad. So DBT was asked to establish these two national testing facilities, so that was also done. Next, please. So this slide shows the diagnostic portfolio. The beauty of this slide is most of these products which have been shown, and there are many more actually. So these were the products which are now reached to the market, and these products have been developed by the startups, biotech startups, and during COVID only. So these products were not there before COVID, and they all startups came together, and these products are now in the market, and large number of testing has been done utilizing these products. So similarly, that the efforts were also made by DBT for scaling up of COVID-19 testing, and in a hub and spoke model, all autonomous institutes of Department of Biotechnology along with national institutes across the country, they came together and they were the hub, and then spoke model, there were some other institutions, then they worked together. And then one thing I would like to highlight here, the Make in India initiative, that deployment of iLab was done to facilitate testing in rural areas, because that was the challenge that was faced at the crisis of pandemic. So these, all the states are listed here, and these iLabs are now actually available in all these states. So these things, actually what I am trying to say, these are the examples, the real examples of Atmanirbhar Bharat. Next, please. So Mission COVID Suraksha, that was actually launched under Atmanirbhar Bharat 3.0 package, and that led by DBT and implemented by BioRec, and that was a mission from Government of India. And under this, you can just see the vaccines which are listed here. The DNA vaccine, as you all know, is a world first and indigenously developed vaccine. Similarly, nasal vaccine, which was launched yesterday, that was also world first and indigenously developed vaccine, and then Corbevax and mRNA vaccine. So all under Mission COVID Suraksha, these 4 vaccines got emergency use authorization, and they are now in the use. And then similarly, clinical trial sites, animal challenge facilities, immunoassay labs, they all will establish during that COVID period. And that actually, that was discussed with all the stakeholders within the government, outside the government, with the experts, and that was felt that country needs these kind of facilities, and we have to do this. And now these all facilities are fully functional. And I'm happy to share that THSTI lab has been recognized as the Global CEPI Network Lab with expertise in T-cell immunoassays. So the scientists at THSTI, they also work very hard. And then later on also, the few augmentation projects were also supported under Mission COVID Suraksha. Next, please. So as I mentioned, INSACOG was actually, in order to assess SARS-CoV-2 variants from Indian patients, the INSACOG was established by DBT, is actually coordinating this with Ministry of Health and CSIR. And so far, more than 3 lakhs genome sequence has been analyzed and has been sequenced and analyzed by this. And there are 59 Indian genome sequencing laboratories where these activities are being undertaken. Next, please. So this is a glimpse of the products which are already reached in the market, and they are developed by the biotech startups, and this ranging from this diagnostic kits, ventilators, monitoring device, remote consultation, PPE kits, and all. So this again is Atmanirbhar Bharat. They were not there before our pandemic. So this is a glimpse, actually, overall impact of the BIRAC in a decade now. And as per the mandate, ignite, innovate, and incubate, that BIRAC has contributed tremendously in the biotech innovation ecosystem. You can see that in terms of national international partnership, establishment of bioincubators across the country to promote and support public-private partnership, and having a pool of mentors. And the details are available on the website, but the biotech startup all are now getting hand-holding and also financial and technical support from BIRAC. Next, please. So this actually, as I mentioned, that our target is to have $150 billion bioeconomy by 2025. So the enablers, we can see the key drivers are that we have to take this nurturing, innovation, and entrepreneurship to the next level through public-private partnership. And also, whatever now ecosystem has been created, there is a need to just expand that further and promote entrepreneurship, and that the key actually component is to foster bed-to-bedside research, and also to have specialized services, research resources for translating research leads. And the last but very, very important, to build a strong, transparent, and efficient regulatory system to promote innovation through public-private partnership. So I'm under now only few slides just to run through that what exactly the impact that we are seeing sitting here in after a decade, that the activities and programs which the government actually supported and the regulatory reforms undertaken and the enabling biotech ecosystem have boosted the overall growth of biotech sector, which has facilitated a large number of innovators to pursue entrepreneurship. So that the impact that we are seeing, that they themselves have converted themselves from job seekers to job creators. So that, for example, the young innovators and the doctors, engineers, product designers, they turn into entrepreneurs to form a startup developing globally relevant innovative medical devices. Similarly, next please. Young scientists, they came out from their lab and turned into entrepreneurs to pursue their passion for developing innovative products, and right from conceptualization to commercialization. Next please. And the leaders, that is very unique actually. Some of the people, they have worked 20 years in the company, they came out from their own comfort zone, and they came actually, and then they actually following their passion through developing various products. And now their products are already in the market. Took some time to bring those products in the market, but now they are there. And now this year you can just see that social entrepreneurs are also there. For example, a cancer biologist left his job to find a cost-effective and reliable solution for breast cancer survivors. So these are the examples of Atmanirbhar Bharat, and this is all the entire product development chain. So this brings me to just close my talk here, and I am happy to take any questions. Thank you.
Thank you, Dr. Sharma. Yeah, we are a little bit behind schedule, so may I invite the next speaker, Professor Anurag Singh Rathod, who is from IIT Delhi. He is going to talk about India as a global biomanufacturing destination, challenges and solutions. Dr. Rathod, please.
Thank you. Slides, please. Okay. So the topic I was asked to talk about was what India, or how India can be a global biomanufacturing destination. Like Aparna Madam said, that we are already regarded as a pharmaceutical producer, major pharmaceutical producer. The question is, can we do this, right? So in terms of, you can just keep going, yeah, in the interest of time. So I just wanted to say that in India, we are known as a producer of safe, efficacious pharmaceutical products across the world, and I think India has played a major role in not just supplying these drugs to Indians, but also to a large part of the world which otherwise would not have accessible pharmaceutical products. The question is, can we do this? Can we repeat that success for biopharmaceutical? And like Alka ji just presented, we are still at, I would say, single-digit percentage, in terms of global biopharmaceutical market. So we have a long way to go. Next slide, please. We can keep going. Yeah. So yeah, you can just keep going and just make it the whole slide. Yeah. So I just wanted to say that if you look at affordability of biotherapeutic products, and I don't know if anybody has used these products, personally, you'll see that there's a huge gap between the pricing and treatment using pharmaceutical products versus using biotherapeutic products. Typically. Anywhere from, I would say, a 10 to even 1,000-fold difference, right? Which means that biotherapeutic products, as of today, are actually not affordable to majority of Indians. Next slide, please. And this slide, this is actually something we did a few years ago for Nature Biotechnology, where we showed that if you look at the pricing of these products across the world, India actually has one of the lowest prices. So it's not like this is as good as it gets in terms of technology today. Next slide, please. So how do you improve the situation, right? How do you make these things more affordable? Keep going. Yeah. Next. So I think the opportunity here, if you look at the left side graph, the trick is that if we can have more and more indigenous companies that are making these products, the prices keep coming down, right? In the case of GCSF, which was the first biosimilar approved in India, prices are now roughly 80% below the price that it was at the time. Yeah, and on the right-hand side, you can see how the volumes increase. Obviously, as things become cheaper, more people can afford them, right? So from a business perspective, that opportunity does exist. Next slide, please. So why is it so difficult? I mean, why is biopharma different from pharma, basically? Next slide. Next slide. So we did a review in which we looked at, okay, what are the most common reasons of, why people fail or companies fail to, do products. And these are the 5 biggest reasons, or most common reasons of failure, right? One is comparability, which means that, if I'm making a biosimilar molecule, people fail to show that it is actually comparable to the innovator molecule, in which case, obviously, you don't get approval. Getting a reference standard, validation, GMP, right? So these are things that I'm sure if some of you are from biopharma companies, you know that these are major issues that we continue to battle with, right? These are things we have to deal with if we want to be globally successful. Next slide, please. Also, these products are very complex. So I am not going to go deep into it, but compared to pharmaceutical drugs, say aspirin, or any of these statins, right, where you basically have one molecule. Here you have a heterogeneous molecule. You have a bunch of different species that together make your product. So it's much more harder to make, much harder to characterize as well. Next slide, please. Our processes are complex. If you do an audit of a biotech facility, right, much more difficult to defend the audit as well to actually do the audit because of the complexity of these processes. Next slide, please. This is just to show you the number of tests we do. So typically for a biotherapeutic product, a company has to do at least 30, 40 different tests to release that product, right? So different level of complexity compared to a pharmaceutical product. Next slide. Next slide. I just wanted to show, so time to time, our lab has published on the quality of drugs that are on Indian market. This is something we did for rituximab biosimilars. These are all products that are selling in the market today. And what we find is that, there are differences. Now, it's obviously, I'm not saying these drugs are not good enough. Obviously, they may be good enough. But the point is that when you look analytically, there are areas where, there are substantial differences between the innovator drug and the biosimilar drug. And that's a challenge for the regulatory agency, because ultimately the regulator has to decide, whether to approve or not to approve a drug. And this just shows that with this class of products, it's a tough, tough decision, right? Where do you draw that line? How much differences are, can be ignored? Next slide, please. So I'll just, in the last part of my talk, I'll just give you a few things that we need to focus as a country. Next slide, please. One is innovation in manufacturing, and I'm really glad that both Alka ji and Aparna ji are here because they lead two major agencies of our country, and so I'm happy to be here for them. Next slide, please. Keep going, yeah. So this is, I just wanted to say that majority, in fact not majority, all of our manufacturing, yes, we made 3 billion doses, right? That was obviously a huge task ahead of us. But any company in India today, you look at, we are all following, and I'm talking about biotherapeutics in particular, we are all following the same technology that, say the companies or the innovators in the US and Europe are doing. We are replicating the same thing, essentially, right? There is hardly any innovation. There's hardly any innovation. And that is an issue, right? So you can't reduce the price of these things beyond a limit, unless you change, you change the way you are making them, right? And that innovation is kind of dismal as of today. Next slide. One of the major trends that is happening in the industry is continuous manufacturing. So this is something which is happening quite widely across the world. Not as much in India. In India, again, our companies are not doing this as of now. But this is something we ought to look at, because if you can do it, you can reduce your cost by 50 to 75%. And I can increase my productivity by 10 to 15 times, right? So it's a major game changer in the sense of how you do manufacturing and then how you can make these things affordable. Next slide, please. And this is kind of a facility that we have built in India, in our lab in IIT Delhi. Next slide, please. And we have shown, we have these platforms where end-to-end, we have shown that you can do this. So there are, I remember last time there was a meeting that Aparna ji had called where Professor Charles Cooney, who's a very famous professor from MIT, presented on the same topic. And coincidentally, we also are working on the same topic. We have the technology that we can give to industry if industry wants. Next slide, please. And also for monoclonal antibody products. So this is something that if the industry takes in, this could take the Indian industry, way above, or at least at par with the best in across the globe. Next slide, please. Next slide. I think you probably, I just picked on very recent news. I'm sure all of you have seen this cough syrup, problems in the newspaper. And I'm not going to go into whether that's real or not. That's not for me to decide. But I just wanted to show that, next slide, please, that, that there is something that we published that just using something as simple as FTIR, you can do fingerprinting of these products, right? So fingerprinting, if I can do this, an FTIR, by the way, is a very simple analytical tool, right? It's not a complex tool compared to Raman or NIR or any other spectroscopy method. So just using FTIR, I can fingerprint my my product, and I can use this. We showed this in our publication. That next slide, please. That I can use this to actually check any counterfeiting, right? So someone like, say, NIB, in fact, so NIB is organizing it. That's why I picked on this particular case. You can have this FTIR in a mobile van. You can go to a pharmacist, buy a drug randomly, test it on the FTIR. Right away, you can catch whether there is counterfeiting, whether there's dilution, whether the drug is spurious, right? So instantly, you can do. So these technologies exist. Now it's for, obviously, the country to use these technologies to prevent, this kind of stuff from happening. So just my final slide. That yes, I think this is we need to invest more in technology. The Indian industry has not, in my opinion at least, has not invested that much in innovation, particularly in manufacturing, and of course academia has a role to play, and so do the government agencies, whether Department of Pharmaceuticals or DBT. Thank you.
Thank you, Dr. Rathod, for a very nice talk, very insightful. May I now invite the last speaker of this session, Dr. Harish Chandra, Deputy Director, Quality Control, NIB, and he's going to talk about scientific advances in the quality and research of biologicals. Dr. Chandra, please.
Yeah, thanks to the organizing team and thanks to the chair for invitation for the talk. I'm basically a laboratory worker, so I'm going to talk a little bit more about the things in the laboratory, what's happening. So many of you might be very excited to know what NIB is doing inside their labs, right? So let's see. So this is where I come from. I'm working as a deputy director for quality control in that institute. And today morning, Madam Minister has said that NIB is a very unique institute. It's really a very unique institute in the country because we develop and validate standards for quality of testing of biologicals, develop linkages with other national and international institutions for scientific research. We also provide training facilities for quality control testing, conduct systemic examination of the quality of biological products in order to facilitate the release of indigenous or imported products. We also develop and advise on pharmacopoeial specifications appropriate for biologicals and verification of proposed monographs. We also do a research on finding new, easy, less expensive and faster methods for quality testing of biologicals. So these are the NIB laboratories. I will not go into detail because our director, Dr. Anup Anvikar, have already covered this thing in the morning. So regarding the types of biologicals, we do 302 different type of biological testing in our institute. And regarding the scientific we are able to do quality testing because of our collaboration and collaborative studies with all these agencies in the world. And we will do a different kind of collaborative studies and also prepare reference standards for those molecules listed on the other side. And these are all the institutes where we collaborate and we do our work of the quality testing of biological and also the research on those biologicals. So among the scientific advances, for example, like in a simple immunodiagnostic kit lab, there is a shift from ELISA to CLIA. CLIA is required because it's a high throughput platform and it's good for a country like India because we have a huge population. So CLIA include high throughput and it's diagnosed without a human error. It's more sensitive and a faster way of detection. So another small thing to do in that case is like in case of a rapid testing, we all use point of care testing, like rapid testing for various diagnostic markers, right? So what the problems come sometimes is that sometimes that line, that band is very faint. It's difficult to diagnose. It's like, positive or negative. Sometimes it's really strong or very thick band, right? Then again, it becomes difficult to, make a decision whether it's a positive or a negative. So we need to have a method like a densitometric studies, which can assess the density of those lines that appear in the rapid diagnostics. Then we have a blood reagent laboratory, very small laboratory, but do a very important job in testing of 98 different kind of, is this not working? Yeah, involving various collaborative and validation studies of blood grouping reagents and gel cards for safe blood transfusions. And also they also do the comparative study for viability of cryopreserved red blood cells when thawed and resuspended in elsewhere solutions for a period of 4 weeks. So next is like we have a biochemical kit laboratory. They do a quality control evaluation of blood glucose test strips, fully automated analyzer-based glucose reagents. The laboratory also tests the glucometer samples. So the COVID-19 kit laboratory does the testing of the COVID-19 kits along with the VTM and antibody, rapid and ELISA kits. We also do the harmonization of all those biologicals. There's a project funded by ICMR, harmonization of COVID-19 RT-PCR kits efficiency data towards enhancing their specificity and sensitivity for different strains of SARS-CoV-2. Then we also did a COVID-19 vaccine testing in our laboratories. So COVID-19 has really impacted our lives because every talk since the morning has touched upon the COVID-19. So here we have a different easy and alternative method for the detection of a spike protein of coronavirus by immunoblotting. So and also we did the infectivity assay as per the manufacturer. So when I was doing that assay, I was saying that I'm unable to see those things, but that infectivity assay worked, but I thought like we should have a different alternative method for this. So we did an immunofluorescence assay, and where we found that this immunofluorescence assay is way cleaner and way easier to do. On the this right side panel, you can see that red marks. That is the expression of a spike protein of the coronavirus inside the cells. So you can easily see that this protein is expressing. That means the vaccine has a good quality. It has an immunodominant antigen in the vaccine. So there are other testing, quality testing of biotherapeutics. Like in sterility laboratory, they also like trying to have a rapid microbiological method for sterility testing of biotherapeutics on CAM-based assays. So we also have a recombinant product and enzyme hormone laboratory. They do an alternative method to an animal testing for quality control testing of erythropoietin. They also try to develop and validate of cell line based assays to determine biological activity of recombinant follicle stimulating hormone, and estimation of potency of urokinase assay by chromogenic method for replacement of existing bioassay. So in a blood product laboratory, they do a different kind of a testing. So they are working on an alternate kits based method for potency assay and human rabies immunoglobulin. They are also trying to work to how to replace barbital buffer in one of the assay. They also work on a Willer-Windermere factor content in a factor 8. And they also evaluate, do SDS page for determination of protein composition in human albumin A. So therapeutic anti-laboratory does the analytical and functional characterization of anti-CD20 monoclonal antibody biosimilars marketed in India, analytical profiling of anti-HER2 monoclonal antibody biosimilars that are marketed in India. They also do the spectrophotometric evaluation of biological activity of vedolizumab as an alternative to FACS. So this is the thing that we are working on. It's pretty interesting to me, as I agree with Dr. Rathod that biosimilars and innovator molecules are different, not only analytically, but also in their biological activity. Here in this graph, you can see that innovator molecule on BT474 cells gives you a reading, EC50 at a different concentration. When this innovator molecule we are using on a SKBR3, it gives a EC50 reading on a different at a different concentration. So similar is the case with the biosimilars. So that indicates there is certainly a difference between the biosimilar and an innovator molecule. So these kind of things can be addressed when academia and an industry comes together and do that those projects together. So this is pretty interesting. And so this we had that final observation, and this project has been funded by ICMR. And our final aim is to have a recombinant cell line that covers the variations in the biological activity of biosimilars for the bioassay of trastuzumab, which is an anti-breast cancer antibody. So hopefully, that recombinant cell line can be used by all the indigenous manufacturers who are producing biosimilars of trastuzumab. So that will be a very good thing to do, have a recombinant cell line. So at NIB, we also do research, and we have recently started a program, a PhD program in a quality testing of biologicals, and also we do a 6-month dissertation program for MSc students. So, so we also have an extramural research funding of, from different funding agencies, and we do have different research fellows like a DBT Ramalingaswamy fellow and a research associates. So take-home message is that all the stakeholders, research institutes, academia, industry, and regulatory bodies come together on one platform to interact, develop, and harmonize the quality testing of biologicals in order to safeguard public health. So National Institute of Biologicals is ready to cooperate and collaborate. Thank you.
So thank you, Dr. Harish Chandar. I think we can open up the house for questions. We have the 4 panelists here. So I would welcome questions, request you to put up your hands, and then also mention the panelists to whom the query or the comment or the question is directed. any comments, if not questions, from your own experience, from challenges that you have faced? Yes.
Yeah, we can hear you. Please go ahead.
उसको भी चालू करो and I had abbreviated an IB as a naturally industrial brothers. Please consider me as your brother. I'm not here as a police officer, as a police inspector. And whatever, perchance I had training in the PGI Chandigarh as a how to do the, write the paper, present the paper. So in that they had taught my professor that whenever you are doing audit, don't say that it is your fault. You say that it is a fact. And the deviation of the fact may be a fault, but I'm not saying that. Then they got convinced, and I said, please, you help me yourself. Because we are not having any, Indian Pharmacopoeia Commission was not available at that time. And no parameters were available in the Indian Pharmacopoeia. So, perchance, Dr. Irade Madam was the director, and she gave me another post, the activity manager of the WHO program for training in the throughout the India. Then I made a special committee, had approval, and said whosoever is manufacturer, they should give their protocol, and as per their protocol, the SOP will be developed. I think all the paper were there, but from shifting from the Dandewala to Noida might be missing. And I think I will request Dr. Anup that he should find out the paper, because until unless you do not know the basic fundamentals, hi-fi will go away. And suggestions. Then to Dr. Amul K. Panda, since I know, basically at that time when anti-sida were being raised by our own hand, anti-sida was not marketed in the country. It was Lalit Mahajan who imported anti-sida for blood grouping reagent. Then we had to do the quality assurance of anti-sida. I know that certain terminology must be very fundamental. I appreciate your lecture, but please do not change the terminology for the biotechnology, for innovation. It is standard technology, standard term must be used that one. Because my way is not the highway. A very fundamental point. You are a academician, you are a professor, that's very good. But for the manufacturer or for the researcher, very fundamental sticking to the terminology is must. With my all this comment, I will again thank to Dr. Anvikar and his whole team, those who have bring the institute to this level within, I think, 2 years only. Yes, I will request all of you to give a special hand to Dr. Anvikar and Farooq.
Anybody else would like to give some comments, or yes, at the back?
Thank you. My question is to Dr. Alka Sharma. I'm Dr. Saini, Associate Professor at AIIMS. Ma'am, is there any preference that BIRAC funds and at what level of the product? And secondly, if a product is already developed Does BIRAC or DBT provides any sort of assistance? Thank you.
There are various schemes available. You just find out on the website. If not, then you can just send me the mail alka.dbt@nic.in. And MD BIRAC also there is a email, and then I can just tell you what exactly, if you will not find that detail on that. The first question is that how much, and that depends on your objective actually of the project. So it's not that up to this and all, but even in that scheme, if there is some limit for the funds, so that also is mentioned in the scheme. But other than that, I would like to tell you that if the scientific merit of the project is there, then I don't think that any committee will deny that to fund. So it's just because nowadays the projects which we are receiving a very large number of projects for each and every, against each and every call. So it depends on the merit of the project. But if yes, there is no cap as such, but it depends on your objectives which you are going to propose for the project. Thank you.
There is one more gentleman at the back who wanted to speak. Jawab aa gaya shayad.
Okay, good evening. My question is to you, Dr. Alka Sharma. Actually, under National Biopharma Mission, do you fund for process development or only for product development?
It's both actually. So it's the entire product development chain, and that focus area which I mentioned in my presentation also is a vaccine, medical devices and biosimilars. So that completes the entire chain. So it's not process or product development, but mainly the overall objective is to product development, but you have to have a process development also. Without that, you can't develop a product.
So I had submitted one proposal, and I got a response that it does not fall under the scope
So
of
that maybe depend on the call, the nature of call, which call was there. So we have to find out. But there are always specific calls again which we take that. But as I said, the overall aim is to develop product and bring the product to the country. Thank you.
Thank you.
Anybody else? Fingerprinting. Expansion.
The expansion, the Fourier transformation infrared spectroscopy. So it's a spectroscopy tool. Yeah.
Just like NMR.
Okay. So I think if there are no more comments, just a last look around, we can summarize this session. As I mentioned in the beginning, I was attracted to this event because of the topic, that is quality in biologics, and I think biologicals, let's say, and that is something which is not only very important, but it is also very topical. Professor Rathod also made mention about these issues. Today we heard a range of perspectives. We heard from Professor Panda, who has both public and private sector experience, and he showed that in his opinion there are fewer obstacles than we believe to innovation and taking it to the market. That is, that entire cycle of innovating, translating it, commercializing it, and taking it to the market. There are clarity of purpose, I believe, from his talk we could gather is central to be able to translate innovation and, as he said, make money out of science. We also heard a very, very good excellent presentation from my colleague, Dr. Alka Sharma. What exactly the government has been doing to promote and support the entire bioeconomy, not just focused on biologicals in the therapeutic space, but across all sectors, and ensuring that government is able to facilitate innovation in the sector. And again, from her from her talk, I think all of you could appreciate that a huge transformation has taken place over the last 10 years in this technological sector, and that is as it should be, because this is going to be one of the drivers of economy in the future. As more and more we will see environmental issues around use of chemicals, as we see more and more AMR, as we see greater demand for precision medicines, tailored medicines, personalized medicine, you will see that there would be most of the growth and value capture will be in this space, and it is important that India should be prepared for that phase of the sector or the industry. We also heard Professor Anurag Singh Rathod, and he was I think extremely pithy, very, very well chosen words. But again, his talk also was, very, very clear that we need to be looking at new technology to help us to transform the industry. And he talked both about, continuous flow technology, which is not really the norm yet, and the fact that we have modern methods of testing that we can use to ensure quality is delivered at the last mile. And finally, of course, we heard Professor or Dr. Harish Chandar from NIB, who again gave us a glimpse of the wealth of expertise and facilities that is available for ensuring the integrity of lab work. I would like to, with the permission of the moderator, to just wrap up with a few words, if that's okay. I'm not disturbing the flow of events.
No.
All right. So, as I mentioned, in India, if you look at the entire pharmaceutical sector, so I'm only going to speak about biotechnology as it impacts the pharmaceutical sector. And pharmaceuticals, as I mentioned, is a broader subject that covers the vaccines, the diagnostics, the medical devices, and the therapeutics. For us, the goals are access, affordability, and quality. These are the triple goals for the Indian sector. So as a developing country, we have to necessarily ensure that the products that come into the market are affordable for the average patient or the consumer. We have to ensure that access is a given, the advanced therapeutics should not be limited to a small elite section of society, but there should be universal access to therapeutics as well as diagnostics and vaccines. And lastly, I think both of this cannot be achieved without ensuring quality of the entire supply chain and the value chain. So quality, access, and affordability are our triple goals, and we have to keep that at the front of the mind in any role that we may have in this entire sector. So whether it is as a researcher, whether it is as a private sector player, whether it is as a regulator, I just would request all of you that we must remember that these three are the cardinal goals for the sector. The pharmaceutical is a manufacturing sector like any other sector, right? So I could be manufacturing automobiles, I could be manufacturing, soft drinks, but I have chosen to manufacture pharmaceuticals. And therefore, there are certain additional responsibilities to the sector, not only in terms of the industry, but also all the stakeholders who enable and ensure that the industry functions in the manner that it should. And this is where I believe Indian industry has really proved itself. And as I like to say, we have built a triple advantage of low cost, large scale, and assured quality, except of course for when we see these unfortunate incidents, which I will come to a little later. So this triple advantage has enabled us to reach a size of about $50 billion for the pharmaceutical sector alone, and another $12–13 billion for the medical device sector. And you saw the scale at which the vaccine industry performed, specifically a couple of companies. You saw the scale at which the pharmaceutical sector performed. So throughout this entire 2.5 years of the COVID pandemic, which were challenging times, not just for India, but across the globe, I think there were probably just 2 or 3 weeks in which we faced any disruption or price extraction in medicines. So remdesivir, you would remember, all of you, right? So that was just for 2 or 3 weeks when we faced that, and that was the time taken to ramp up the production, because as Pandaji will also know, there is a cycle of 3 weeks. When I put in the first batch and start production, and I'm able to bring the batch to the market after testing for sterility because it was an injectable, it does take 3 weeks. Like, I cannot ask somebody to produce a baby in 3 months. It will take that 9 months, right? So that is the lag that is there, because these are biochemical reactions or biological processes, if you're talking about MAB. Apart from that, I think we were able to maintain a fairly well-managed supply of therapeutics. Vaccines, all of you did see that once the plants stabilized, they were able to ramp up. So scale is a given as far as India is concerned. Similarly, we talk about cost. You would again agree that even today, 3rd year of pandemic, if you go into a Western economy like the US or EU, even adjusting for purchasing power parity, you would be paying much more for an RT-PCR test than you pay in India. Today it's probably 400 rupees for a test. I mean, I can get tested every week if I have to go to the cabinet or meet some higher ups every week, you see. So cost also we have been able to conquer in our own domain. And the third is quality. Quality in the pharmaceutical sector, and I was quite shocked when I came into the sector a couple of years ago. Although there is we call the sector a regulated sector. You're making me nervous. Should I wind up?
No, I'm listening.
Okay. I feel I'm making you stand. So you see, although this is a regulated sector, all of us know it, and there are some doctors, professors from the clinical side also in the gathering here today. The quality is to be maintained at various levels. So the quality of the raw material, quality of the excipients, quality of the processes that are used, and then the quality in the supply chain, where you store the medicine, where you store the vaccine, how you deliver it to the patient, etc. There are different stages. However, regulation is a one-time effort. So when you're putting up a manufacturing facility, you have an inspection, you submit your documentation, your dossier, etc., and you get your drug license. Subsequent testing of every batch, except in the case of vaccines, which was being done batch to batch, is actually being done by the industry. And thereafter, there are some sporadic tests for quality by picking up samples through the drug administration from the retail level or the storage level. There are some sporadic inspections when the WHO GMP license is completed, etc., of industry. So quality in the sector has to be assured by making quality an integral part of product development, manufacturing, distribution, and consumption. So I think even consumption has a part to play in quality. If we as consumers are going to indiscriminately pop azithromycin for every sore throat, we are contributing to increasing the ineffectiveness of these products by increasing resistance in the population. So all of us have to understand that maintaining quality is is a challenge or an issue across the supply chain and the value chain. In India, for example, again, this shocked me. If as a manufacturer, I change the supplier of my salt, I don't need to take revised permission from the regulator, from CDSCO, who incidentally is not with the DOP, is with the Department of Health, but we're all part of the same government system, so we can talk about all these things. Whereas I know that if you need to take an approval for a product in the US FDA or a WHO pre-approved vaccine, and you're changing your suppliers not only of the salt if it is a medicine, but even of, say, the consumables. If you are making a vaccine, for example, you have to go back to the regulator to get approval, where the regulator will be ensuring or checking that change in your supply chain, change in your supplier will not adversely impact your quality of the product. Talk about product development, which is done. We were talking about translation in innovation. This product development is done in laboratories. It is done in public research institutions. It is done in private laboratories of manufacturers. How many are actually trained to view the success of their product development in terms of how it will perform in the field? It is like clinical trials, right? So you are in a product development cycle, you are working in a closed environment, closed ecosystem. And how much are we trained to think in terms of the risks that would be becoming manifest when the same process is replicated in an industry, not in a large industry like, $2 billion industry or something company, but in the scores and dozens and hundreds of MSMEs which will be producing that generic drug or that biosimilar down the line, 5 years down the line. What would be the risks when that product is being produced or manufactured at that level, where it will be a completely uncontrolled environment, ecosystem, with very different capacities and capabilities in terms of manpower. I think I'm saying all this because in India, we have a challenge of maintaining quality in the face of our avowed objective or goal of affordability. That's why I started with our goals. Somehow, there is a trade-off that is sought to be built between quality and affordability. Now, this is a trade-off that we should not accept. We need quality medicines at affordable prices. This should be the goal of all of us present here, and it is a goal that we can certainly achieve. As we have seen, right, in whether it is in the the vaccine space or in the therapeutic space, Remdesivir, for example, which was being sold at 5,300 when we got the license, and 7 companies came forward to make it in India. They were asked that, can you reduce the price? And voluntarily, all of them brought it down from 5,000 plus to below 3,500. One company even brought it down to something like 2,500 or so. So we know that quality and affordability don't have to be traded off against each other. And therefore, within that paradigm, it's our responsibility, whether one is working as a researcher, whether one is working in a private company as a manufacturer, whether one is a regulator, to be having that single focus on quality of the product that we are putting out in the market. Particularly because here, again, the market is fairly unregulated. These pharmacists, we have 9.5 lakh retail outlets in India, 9.5 lakh retail outlets being licensed and ostensibly supervised by 32 different regulators of varying capabilities and capacities. So therefore, one has to remember that the patient who actually consumes the product is subject to so many risks related to quality along the entire supply chain. And this needs us to invest in innovating. You talked about Jugaad, which has been given respectable name of frugal innovation by Anil Gupta and others. I would say that for us, we have to invest in robust innovation. That innovation of a process product which can withstand the pressures of all these risks in eventually reaching the patient, risk along space and time. Right? So because the product that you make stays in the market for some time, right, before it is replaced by some other product. Similarly, in terms of space, it moves from the laboratory to the factory to the distributors and so on till it reaches the consumer. Every particular batch I'm talking about. So I do think that there is a need to inculcate this risk awareness when we actually are being enthusiastic about developing new processes, new products, etc. There is a need for incessant and extremely, how shall I put it, extremely demanding testing of our processes and the final product that emerges from it. I totally agree with the panelists, particularly Dr. Rathod also. In terms of manufacturing, I think there has been less attention to innovation in manufacturing. There is a lot of attention to innovation in product development. And I think most of your startups also, you will find, they focus more on getting a new product into the market, differentiating it, providing some cosmetic or therapeutic benefit, etc. But as regards improvement in the manufacturing process, I believe there is opportunity, and I do hope researchers and scientists present here would like to think about what kind of innovations we can bring into manufacturing to again meet those goals of assured quality and affordability. So this is also something where I do believe that there is a lot that requires to be done in this country. Finally, I will come to quality assurance. There is also, I think, a lot of potential to develop the quality-related infrastructure in the country. Right now, we have, since 1st October 2022, rolled out regulation for medical devices, 2 categories of medical devices. And one of the concerns raised by the industry is as to whether there are adequate testing capabilities available in the country to ensure the integrity of the device vis-a-vis the standards and norms that have been prescribed for it. And you will find the same challenge will soon become manifest in the biologic space also. So as a country, we are very well equipped in terms of what I call traditional therapeutics, that is the chemistry and biochemistry based therapeutics. But the moment we move in a larger way into biologics, which inevitably will happen, we would face this shortage or dearth of testing infrastructure. And when I say infrastructure, I mean not only the brick and mortar labs, but the processes, the IP for the biologics, standardization of testing processes, norms, and the manpower to actually be populating these testing infrastructure. So you can understand that there are still a large number of gaps that we should be addressing if we want to truly achieve, as Alka mentioned, that figure of bioeconomy much by 2025? $150 billion size from the current $80 size. If we have to really achieve that, or as pharmaceuticals say, to go from $50 billion, we want to achieve $130 billion size by 2030. So all this growth is going to demand that we pay attention to every part of this entire ecosystem, and I'm very happy that today we have people from different organizations, different sectoral segments talking, and you had very rich discussion in the morning also when you talked with industry, you talked with Dr. V.K. Paul, who kind of guides all of us, and that at the end of the seminar or at the end of this workshop, if all of us are filled with this understanding that whatever we do in our daily working place, that has to be geared towards the larger objective, larger noble objectives. And there is still one item which I would just mention in passing, and I talked about the environmental impact of chemical-based therapeutics and diagnostics. So when we move into genetic manipulation, when we go into applying technology to biological processes, there are ethical implications, right? So those will also become a huge area of study, analysis, and regulation that we have to start preparing ourselves for. And therefore, I do believe that each one of us, while working on this sector, may like to keep the larger picture in mind, may like to have that perspective that at the end of the day, what we are trying to do is provide solutions, whether they are in the form of therapeutics or vaccines or diagnostics, to our people and eventually to the entire world, provide solutions that are safe, efficacious, adequate, not excessive, and affordable for the masses, for everybody. So access will continue to drive this sector. All of you are aware of the Sustainable Development Goals. So SDG 03 talks about universal health coverage or universal health. And one of the indicators or targets within that SDG 03 is safe and affordable medicines and diagnostics for all. So we would be working in that huge global endeavor. And this is not a science like any other science. It is a science that affects human life, well-being, and therefore, society at large. So I always feel that, anybody who has chosen to come into this sector would have a sense of purpose which is higher than just earning your livelihood or, getting your PhDs or promotions, etc. We have a duty. There is a noble moral duty to the society that we have in this sector, and maintaining quality through the various, processes or various channels that have been discussed here, including that of innovation, is probably the most fundamental duty that we need to keep in mind. It is not a question of simply, if there is an incident, you react, you respond, and you move ahead. No, I think there is learning to be taken from each one of this, and this learning consists of appreciating challenges, being able to take tough decisions, whether it is in enforcement or in regulation, and trying to weed out unnecessary, how shall I put it, chaff, unnecessary elements from the entire ecosystem, so that we can have an ecosystem that is fully aligned towards maintaining quality. This will be difficult to do, but I do believe that if there is a common purpose and all of us are aligned in terms of objectives, it would be possible. So I would like to thank all the panelists for very enlightening, illuminating, and thoughtful remarks. Certainly I learned a lot in the last 1 hour, 15 minutes that was here, and I hope that everybody here also enjoyed it. So I would not like to add more. Thank you.
We couldn't have anticipated the better ending of this session, ma'am. Thanks a lot for your all valuable words. With this, I would like to request Madam to felicitate the fellow speakers. I'll request people to come. Okay, we'll start with Dr. Alka. Dr. Amolya Panda. Professor Anurag Rathore. Last but not the least, Dr. Harish Chandra. I would like to request you to stay back and you can take your seats. I would like to invite
Anup,
our director, Dr. Anup Anvikar, on the stage, please. And Dr. Charu Merakama, can you please join us on the stage? I would like to request first our director, Dr. Anup Anvikar, to kindly felicitate Madam. Before moving to our closing session, I would quickly request all of you to kindly fill up the feedback forms. It's there in your folders. We would love to listen from your side. And you can give us, anyone who has this orange tag, you can hand it over to them or to the registration desk. With this, I'll request our director, sir, to come and address the gathering for conclusion and way forward remarks.
Madam S. Aparna, Secretary, Pharmaceuticals, and my dear friends. Now we have all become friends. I don't have to go into those formalities. Madam abhi aayi hai, but then I don't know how she came to know what we discussed during the whole day. She summarized the whole thing which we had been discussing since morning. So I will just take 1.5 minutes to summarize the whole day and what we have discussed. So, Madam, today, Honorable Minister Sir and Honorable Minister of State Madam, so they inaugurated, they could not physically come over to this place, and Secretary Health Sir was also here with us, and Professor Vinod Paul also chaired the previous session. I won't summarize this session. And since morning we have been talking about the SDG goals and the target 3 point of that, and affordable commodities, and that too quality. It's not that there is a poor medicine for poor people. So that has been the mainstay of today's discussion, and everybody of us was on the same page, be it the researchers, the manufacturers, or the academia, or the policy makers. And in fact, we had excellent representation from the procurement agencies as well, because these are the ones who are the link between the public health system and the industry. And we have discussed many issues, and we have also decided some way forward, and my team has noted down all the discussions, and we will soon be circulating the proceedings of this meeting. So I again thank you everyone for being here, and it was really good to share experiences with diverse types of colleagues, and that will definitely help us help India to strengthen the quality assurance system, not only for biologicals but also for other commodities. So thank you all.
Can we request ma'am for few more lines?
आपका कुछ closing remark?
Okay, then I would like to request Dr. Charu to come forward and present the vote of thanks for all of us.
No duty is more urgent than that of returning thanks. Good evening to you all. I, Dr. Charu Mehra Kamal, feel honored to propose this vote of thanks on the behalf of the National Institute of Biologicals. I wholeheartedly thank our chief guest, Dr. Mansukh Mandaviya ji, Honorable Union Minister for Health and Family Welfare, guest of honors, Dr. Bharti Praveen Pawar ji, Minister of State, Mr. Rajesh Bhushan ji, Secretary Health and Family Welfare, our chairs of the 3 sessions, Professor Atul Goyal, Dr. V.K. Paul, Dr. Y.K. Gupta, Ms. S. Aparna, Secretary Pharma Madam, Professor Garg, eminent speakers, and all the dignitaries present here for sparing their valuable time and sharing with us valuable knowledge and information. An event like this cannot happen overnight. The wheels start rolling months ago. It requires planning and a bird's eye for details. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues of NIB who know their job and are result-oriented. I take this opportunity to thank our director, Dr. Anup Anvikar-ji, for his valuable guidance and unhindered support. I would like to express my thanks and heartfelt gratitude to all NIB family, our super seniors also, all known and unknown hands who have contributed to the success of this event. Thank you all.
She's closing.
Just a last point that we would now have come to the last point. We'll have a high tea ready for all of us, and please collect your books from the counter as well. Have a nice time. Thank you.
हम नीचे आ जाते हैं, हम नीचे आए।